It's working. It's working.
मैं रखूँ कहाँ नीचे I'll start. So, good morning everyone. Welcome to the 7th Molecular Virology Meeting. Without further ado, please let me welcome on stage Dr. Usha Vijay Raghavan to declare the meet open. Dr. Raghavan is the Dean of our Biological Sciences Division here at IISc, and she has had an illustrious career, and her research on the gene regulatory mechanisms driving cell fate and differentiation have been very well recognized and awarded. So please, let us welcome her on stage.
Good morning, everybody, and welcome to IISc. It must feel so nice to come back to Bangalore. Many of you have been here before on one or the other forum, and it's my absolute pleasure to say a few words, although it is not necessary, to just get this meeting going. The Molecular Virology meeting, the 7th in the series, if I'm not mistaken. So for those of you who are very young and don't know the history of IISc, we are about 114 years old and started with just 2 departments with an equal emphasis in science and engineering. One science department which was in electrochemical technologies and the other on electrical technologies. And we've grown now to 42 departments organized in 6 divisions. The Division of Biological Sciences houses 6 academic departments and 2 centers. Now, the research on virology actually traces back to very many years when people studied infectious diseases and infectious disease models in the then microbiology and cell biology department and its predecessor department, which was on fermentation technology and pharmacology. So understanding the processes which cause diseases, the pathogens, their interactions, and so on, has been a tradition of deep research at IISc. This group of molecular virology itself traces its history back to the early 1990s, and we have people with huge experience. You know, we have Vijaya here who brought in her expertise in Japanese encephalitis virus and several flaviviruses. We have Soumitra here who works on many different types of viruses, hepatitis, Coxsackie. We have Where is Rangarajan, P.N. Rangarajan? Somewhere I saw, oh there, right, JEV. If I miss a few names, please forgive me. This has only grown from strength to strength, and we now have Kesavarna Sanula and Shashank Tripathi. So you're all welcome here. You're going to have deep insights, both on the research which is going on here, and our team, our students, our faculty and postdocs will learn a lot from the research that's going on at your institute. Enjoy the coming 2 days of rich academic discussions.
Thank you, ma'am. Now please let me welcome on stage Professor Soumitra Das to give the opening remarks. Professor Das has been an asset to IISc, and he has contributed immensely to the field of virology in all the years spanning his career. So we are really quite fortunate to have had his guidance in organizing this meeting. Please welcome him on stage.
Thank you, Usha, for gracing this occasion, and we wanted to keep it very informal because it's a molecular virology group meeting rather than a conference. I was just telling people that you enjoy more in interactions and very informal gathering, so you can always feel free to ask questions, run into anybody and then discuss your problems. This whole group concept started way back in 2009 when Institute was celebrating centenary at that year, and Professor Balaram at the time director and the dean at that time we used to call Division of Biological Science chair D.N. Rao. He asked me to somehow convene a virology meeting for the conferences. And as usual, there was a challenge for the funds, and then Institute gave us some funds, and then Professor M.R.S. Rao from JNCASR stood forward and said that you please go ahead and then have this meeting combined with JNCASR. So that's the beginning of this. And it has come a long way. This is the 7th meeting, and in between we had this is the 3rd time we are hosting this meeting. Because of this COVID, we had a gap in between, but we found this 3 and 3.5 years, there are a lot of virologists and young virologists are doing excellent research. So in this particular meeting, we try to highlight that talent, which actually, will actually carry the mantle to the future. And all the seniors, we have decided to actually stay back and then participate, or we try to accommodate them as a chairs, panelists, and the poster judges. But the unfortunate situation is because of several constraints, we couldn't accommodate all the posters. There was a huge enthusiastic response, but we could only select some of them to attend physically, and apologies for them who could not attend physically, but we are planning to have some live streaming to somehow at least they can listen to the talks. As I said that this meeting are actually the strength is the students. In fact, in 2009, there were 2 student participants, Dr. Shashank Tripathi from ICGEB and Keshav Sanula from Indian Institute of Science. They are now established virologists, like several of you others who actually were in the first meeting and presented poster or presented a short talk are now established virologists, and that's the tradition. We wanted to continue that tradition and move forward, and I will just try to see that how as a group we can help each other and actually strengthen the virology research in India. But most of this work is done by the conveners. That time there were participants Shashank Tripathi and Keshav Sanula, and I just said that, as Usha has mentioned, this whole virology group thing, the inspiration was actually there was a strong virology group in our institute, starting from Professor M.S. Shaila, Vijaya, Professor Anthony, and we had at least 8 of them. And now in between we didn't have that many people, but now it's good that we have to revive the virology group, and that is possible because of new recruitments. Same is for the other institutes also. I'm sure that with the new blood and the new rigor, I think we'll be able to move forward and then take this virus research in India to a newer height. I would like to request Dr. Shashank Tripathi and after that, Keshav Sanula, the conveners, to address few words to you. And we wanted to make it very informal, and that's how I think you enjoy this stay, and any problem, you blame us. Okay, but I think it's anywhere there will be some gaps, we try to cover it. That's what Shashank is going to highlight, but still we'll try to solve it together, like the way we are actually solving the virology problem in India. Thank you.
Good morning and welcome. Professor Das mentioned already, stole my line, that I was here as a student in 2009 and now I'm part of the organizing team, so I feel privileged. And for those who we couldn't accommodate for several restrictions in the meeting for their physical presence, we have the event live on YouTube. That is on the conference website. You must share with your colleagues and your lab members back in your lab so that they can also attend along with you. And because of curating the talks and posters, one good thing has happened, that the quality of science has gone really high. You can browse the abstract book and you will see how difficult it was for anybody to reject anything. There is fantastic science curated here on the poster sessions as well as talks. So please immerse yourself in good science and enjoy. Thank you.
Good morning everyone and welcome you all for this meeting, and we thank you all for attending this meeting because we are excited to conduct this meeting at this point of time. And along with the meeting and talks, and we think that accommodation, transport and food is important. I think we took care of everything, but if we face any difficulty, please let us know. We'll be here all the time. And I'd like to thank Professor Usha Vijayaraghavan and Professor Samitra Das for inaugurating this session. It's a fantastic start for this meeting. And I thank all the speakers, students, poster presenters, and panelists, and poster judges, and the chairs for attending this meeting. And we welcome you all for this meeting, and I hope you have a good time in this meeting. Thank you.
Right. Thank you, sir. So the first session of the day is on virus structure and replication, and it will be chaired by Professor Vijaya Sachidanandanam and Dr. Priya Abraham. Dr. Vijaya is an expert on the molecular pathogenesis of flaviviruses. She is recently retired from professorship at the microbiology and cell biology department here at IISc. However, she continues to inspire us. Welcome, ma'am, to the conference. And Dr. Priya Abraham is currently the director of the National Institute of Virology and a professor of clinical virology at CMC Vellore. She has served as a consultant for the World Health Organization on various viral infections. She has also served on the Scientific Advisory Committee of the Indian Council of Medical Research. She took over the role of director at NIV Pune just 2 months before the outbreak of COVID in India. And it is because of her team and her that India was able to make significant progress in containing the spread of COVID in the country. And very recently, as of September 2023, she has also been elected as a fellow of the Indian National Science Academy. Thank you for joining us today, ma'am. I would now request the chairs to begin the session. And just a small reminder to the speakers, there will be an 18-minute timer on the screen while your talk is ongoing. And then there will be a 2-minute grace period followed by a 5-minute Q&A session. Thank you. Please.
She Hi, good morning, everyone. And it's a great pleasure to be at this meeting and in IISc Bangalore. Thank the organizers for inviting me. So I'm going to talk about one of the ongoing themes in my laboratory, which is trying to understand the role of virally encoded membrane active proteins in the life cycle of viruses. And the virus that I'll be focusing on is chikungunya virus. So, viroporins, as the name suggests, are membrane-active proteins or peptides that are encoded by viruses. Sometimes they're components of the virus capsid, and sometimes these are non-structural proteins that are generated during viral replication. And typically, the aspects of the virus life cycle where these membrane-active proteins are involved are during membrane penetration, which makes sense because the cellular membrane needs to be disrupted in order for the viral genome to be positioned in the cellular cytoplasm. But there are supposed to be some very essential roles for these viroporins in viral assembly and egress, and this part is relatively less understood. So what I'm going to tell you is what we have found so far, but we really do not have a definitive answer as to what these viroporins do or these membrane active proteins do and how do they assist in viral egress or viral replication. So these membrane proteins, a subset of them are ion channel forming proteins. All of them exist in some kind of oligomeric form. Compared to regular ion channels that we are familiar with, viroporins are a little bit different because their association typically has a lot of dynamic character. So the oligomeric status is usually not fixed. And this has been shown recently by structural analysis of some viroporins in SARS-CoV-2 and also in dengue virus, that the association between monomers to form oligomers, even though the oligomeric forms all appear to be functional, the association is relatively fluid and dynamic, which makes it challenging to carry out structural studies. And as I mentioned, the way that they help in virus propagation is not really established. So the mechanistic details of how the ion channel activity is helping in viral egress or viral propagation is not very clear. So chikungunya virus, I think, requires no introduction to this crowd. It's an alpha virus, it's mosquito-borne, and it causes infections in almost 100 countries in the world, including India. Now, if we look at the proteins that are produced by chikungunya virus, so on this side we have the structural proteins which make up the viral capsid, and then these are the non-structural proteins that are generated during virus infection, but they are not part of the capsid. Now here, there's a very small peptide. It's a 6kD peptide, which without much imagination is just called 6K. And so it is not really very clear whether this peptide is part of the virus capsid or not. So it kind of straddles the space between structural and non-structural protein. It is very, very hydrophobic. It's a membrane protein. And on studies on other alpha viruses, such as Semliki forest virus and Syndebis virus, has established that if you just remove this gene from chickpea, from other alpha viruses, the viruses that are produced are kind of different looking, and I'll show you what I mean by different looking in a minute. And the particles that are produced are not very infectious. They get very quickly cleared out from animals. So there was some thought that this could potentially give rise to some sort of attenuated strain, which would be virus particles not terribly infectious to animals or humans. Now, the story of 6K is further complicated by the fact that during infection, viral infection, 6K is produced also in a trans frame format, or this is the TF protein of 6K as it is called. So 6K itself is very, very hydrophobic. It also has a ribosome, a slippery sequence where there is a -1 ribosome frame shift, which gives rise to a variant that has a C-terminal tail which is different from that of 6K. So the TF that is produced during infections is about 2/3 identical to 6K, but the C-terminal region is different and it is hydrophilic in nature. It is a charged protein. This is a minor product which is produced in about 18 to 20% of the cases, but when TF is being produced, then the capsid protein E1, which is part of the glycoprotein spikes on the surface, cannot be produced. But studies on chickpea as well as other alpha viruses have shown that both 6K and TF are definitely produced during viral infections. So our question was, what do these two different proteins do in the chickpea life cycle? So we were able to generate both of these proteins, but we can only generate them in conjunction with a partner like GST or MBP, and in conjunction with detergents such as DDM. It is hard to stabilize these and keep these in solution. Some structure prediction softwares show that the C-terminal tail of TF kind of extends out, which is the hydrophilic region, and while 6K has a little bit of a kink, but the middle helix, which is common to both 6K and TF, is expected to span the entire region of a bilayer. We showed that both 6K and TF form ion channels in artificial membranes, although the nature of the ion channels are a little bit different. Both of these typically have a fairly high average conductance, and as I said, these viroporins form ion channels which are different than regular ion channels that we have. The gating mechanism is not absolutely clear, but both of these are sort of non-specific cation conducting channels. And if we just look at some kind of an oligomeric assembly for both 6K and TF, what we see is, okay, so this is on the right we have the TF ion channel, on the left this is the 6K formed ion channel. Again, these are predicted structures. But what we find is that in the oligomeric assembly, the central helix actually twists around in case of 6K, whereas because of the presence of the C-terminal extension in case of TF, the helices are fairly straight while they're forming the channel. And TF helices have a lot more interaction between monomers in the construction of the oligomer. Again, if we just look at the nature of the channel which is produced, we find that the channels are different in terms of the size as well as the charge. Particularly TF channel is highly charged. It has more of a negative charge component, whereas 6K ion channel is not as highly charged. Also, there is a size difference, so the gating mechanisms in both of these cases are expected to be different. So with this in mind, what we have been trying to do is to try to resolve structures of 6K and TF using cryo-electron microscopy. We started first with 6K, and this is data that we've collected at the cryo facility in IISc Bangalore. And what we found was that the particles we get are significantly heterogeneous. So this is what comes down to the conformational heterogeneity and the dynamicity of the particles. So if we just do a 2D classification out of the images that we collect, we find that where we get particles that are relatively, we can tell that these are maybe tetramers or hexamers. In other particles that we see highlighted in yellow in the 2D classes, definitely there are some higher order oligomers, and at the 2D stage it becomes computationally very, very challenging if there's a significant amount of heterogeneity to try to sort out that heterogeneity. Because of what we saw in the computational prediction, that the TF interactions among the TF helices are supposed to be more intense and more comprehensive, we thought that perhaps TF would give us a better idea about how the ion channels would form. It would be structurally more amenable. So we started working on TF, and we collected a data set at the cryo facility in IIT Delhi. And what we find is relatively more homogeneous particles, as we can see here, which all appear to be of a specific oligomeric form. And we get a fairly decent 2D classification. This is just the 3D initial model. We've got a small data set, now we're collecting a large data set and we're trying to solve the structure. But what we find is that it appears to be a trimer of dimers. Each one of these lobes that you see here, MBP is out there, and TF comes here and forms that open channel that we see. and there appears to be at least 6 particles. The majority of the population appears to be hexameric, and there would be 6 TF helices which make up the central channel. And hopefully a higher resolution structure would give us a better idea about the gating that we are trying to conjecture. But while we try to understand what the ion channels formed by 6K and TF look like, the other question is functionally what do they do and why are they important? If we remove 6K, why does it affect the assembly or the infectivity of the particles? So initially we tried to figure out an inhibitor which would make our job easier, and we found that amantadine, which is an inhibitor, a generic ion channel inhibitor, it was I think initially identified to inhibit the M2 ion channel of influenza virus, that also inhibits the ion channel activity of 6K. And with amantadine, we tried to see how it affects virus assembly. So we used just the structural cassette to generate virus-like particles in the absence and presence of amantadine. And what we found, where in the presence of amantadine, which inhibits 6K activity in vitro, we find that the particles that form are actually significantly different from wild-type chickpea particles. So this is a cryo-EM image, and what we find are things that appear to be multi-core, or particles which are really not covered by glycoprotein. In comparison, you see these beautiful chickpea particles from where one can do a beautiful reconstruction. But with these particles that are produced when 6K is inhibited, they look very, very different, and they're functionally different. So this is work that we did in collaboration with Dr. Soma Chattopadhyay at IISc Bhubaneswar, and her lab found that these particles are not really very good in terms of infectivity. So they don't look good and they don't behave well either. So to try to find an answer as to what could 6K be doing which is affecting virus assembly, we tried to see how it localizes in cells in conjunction with other viral glycoproteins. And we found that while 6K typically is located in the endoplasmic reticulum, when other viral glycoproteins, specifically E2, is expressed, it moves to the cell membrane in conjunction with E2. And recently, Joyce, Jose, and Richard Kuhn's group has shown that in syndipus virus, they see a similar phenomenon where 6K and E2 together go on through cytopathic vesicles to the cell membrane. So the proposal right now is that 6K could be functioning as a chaperone for virus assembly, and its presence is really needed for the viral proteins or the structural proteins to move together and get packaged in the same particle in a correct fashion. Otherwise, their assembly is affected, as well as the infectivity of the misassembled particles is definitely compromised. Now, and this again, I don't have an answer as to why the ion channel activity would be required for this, but this is something that many groups are trying to work out. Now we come to TF, which again does form ion channels like 6K. The gating properties are supposed to be different from 6K ion channels, but again we do not know what it really does. So when we were looking at the sequence of TF, particularly the unique region, what we found was the presence of a very nicely defined PDZ domain binding motif, which is highlighted in pink here. And some prediction studies indicated that one of the binding partners for this particular PBM could be human scribble. Scribble is a cell polarity protein. It is present at tight junctions in conjunction with some other components like DLG1 and LGL. And together they form the cell polarity complex. Scribble is also a pro-apoptotic protein, and it is targeted by various, the one viral protein whose interaction with Scribble is very, very well defined is influenza NS1, but it does interact with other viral non-structural proteins as well. So we tried to see whether Scribble could be a binding partner for TF, and we initially started off by doing some computational analysis using empty simulation, and we found that Scribble PDZ domains 1 and 4 show potential tight bindings with TF. We then overexpressed TF in cells, and we tried to see where does TF go. So this is TF overexpressed in conjunction with EGFP, and this is live cell imaging. So after about 24 hours, what we find is that sort of associates in puncta. It localizes in the form of puncta in cells. And when we co-stain for scribble, we find that the TF co-localizes with the puncta that also have scribble. So TF appears to be mislocalizing scribble. It moves away from its normal localization pattern and it gets localized in the form of these puncta. Now, when we mutate the PDZ binding motif in TF, so the PDZ binding motif is HGG ATV, and when we mutate by introducing 2 alanines or 4 alanines, we find that the localization pattern of TF is changed and there is no co-localization with scribble. So the sequestration of scribble in punctate form in cells is likely being guided by the PDZ domain binding motif of TF. Next, we tried to generate an antibody against specifically the unique region of TF, because we wanted to monitor TF during chickpea infection and without trying to label 6K. There are no commercial antibodies available for either 6K or TF. But we generated an antibody in conjunction with Dr. Shomin Basaket and II, and we found that this antibody, which was generated against the unique region of TF, actually does only bind to TF, not to 6K, in Western blot as well as in confocal microscopy assays. So what we found again, that during viral infections, so infections of HEK293 T cells with chikungunya, again TF goes into these punctate forms where scribble is also localized, or rather mislocalized after chickpea infection. Interestingly, after about 48 hours of chickpea infection, we find that scribble is almost completely gone. It is down-regulated, and the scribble which is mislocalized in the puncta along with TF also gets heavily ubiquitinated. So somehow the expression of TF during chickpea infection seems to be targeting scribble for degradation. And the puncta we also tried to analyze using electron microscopy of cellular sections, using specific scribble antibodies that are labeled with nanogold, and we do see these beautiful puncta that form in cells upon chickpea infection. Now, this particular mislocalization of scribble appears to be unique to TF, not to any other chickpea component, because even in transfection models where we just have TF EGFP, we find that scribble does get mislocalized in the puncta where it is ubiquitinated. So the question here was, why is scribble important? If scribble is down-regulated, then what would happen to chickpea infections? So we knocked down scribble using an shRNA method, and we tried to see what happens to chickpea infections. And what we found that upon about 50% knockdown of scribble, the chickpea infectivity or the propagation of chickpea increases to about 3-fold in cells. So what we suggest is this TF, in addition to its ion channel activity, appears to have another activity which is localized in its PDZ domain binding motif, which it uses to downregulate scribble, and this seems to potentiate chikungunya infection by probably downregulating the pro-apoptotic functionality of scribble. These PDZ binding motifs are found in TF sequences of many other alpha viruses. Igbo-Obra and Ong-Wong virus has exactly the same PDZ domain binding motif, whereas some other alpha viruses, the TF sequences, have type 2 or type 3 PDZ domain binding motifs, and it could be possible that these are actually host interacting factors which carry out similar functions. So in conclusion, it appears from our studies as well as those of others that 6K and TF both have important roles in alpha virus assembly as well as propagation. Our work is primarily on chikungunya, but there has been work done on Sindbis and semliquiforous virus and Ross River virus. We think that 6K and TF both have very important functions, but these are different functionalities. 6K may just be involved in the assembly part, whereas TF may have roles in terms of host factor regulation. We see that inhibition of 6K definitely leads to assembly defects and reduces viral propagation. On the other hand, TF seems to have an important role in in sequestering scribble in punctate form, which sort of abrogates its pro-apoptotic activity and allows the virus to propagate further. And this interaction is mediated by the PDZ domain of scribble and the PBM motif of TF. Now, there are many unanswered questions, and I have listed some of them here. But the primary question for my group has been to try to answer how is the ion channel activity at all connected to what these proteins are supposed to be doing. How is this connected to virus assembly, for example, or whether ion channel activity of TF has any role to play in host-virus interaction. Another very important question to us is, are 6K or TF incorporated actually in virus particles? So this, in context of synthesis virus, it has been shown that TF actually does get incorporated in particles. We do not know whether TF is also part of chickpea, and we are trying to figure that out by trying to solve a high resolution structure of chickpea without application of symmetry. And then the last question that we are trying to answer is, can we sort of separate out the assembly, the role of 6K or TF in assembly with their role in potentiating chickpea infection? And by making judicious mutations, can we generate attenuated particles which would look like chickpea, but it would not be as infectious? So with this, I would just like to thank my lab members who did all this work. Primarily, Kirti is involved in solving the structures of 6K and TF. Pushkar and Ramesh are the two students who are working on the interaction of TF with scribble, and Johnson is a very recent joinee who is doing the prediction algorithms and the MD simulation studies. Our collaborators are Professor Shubhendu Ghosh from IISc, with whom we are doing the ion channel assays, Dr. Soma Chattopadhyay from IISc Bhubaneswar, and Dr. Shomin Basar from NII, with whom we're working on chickpea cell culture and animal models, and the data collection have been carried out in all of these different facilities. Thank you for your attention.
Is it possible that 6K has a role in maintaining the ER action levels? And that's how protein bonding and the virus?
That is a very good question. It may be entirely possible. But it has also been shown to localize to the plasma membrane when E2 is expressed in cells. So it could be that a population stays in the ER and a population goes to the plasma membrane. We do not know why or how. It could also be that some activation of inflammasome through formation of ion channels could be involved in chickpea pathophysiology. We have not worked that out as yet.
Is it possible that this concept should clearly be a spectrum of heterooligomers between 6K and TF?
Yeah.
And how does it affect the viral function?
Yes, absolutely. That's a very good question. So in the ion channel studies, we see different types of channels that are produced, and they are all functional in terms of ion channel activity. But it would be good to tease out if we can separate out the oligomeric forms and see what happens. Unfortunately, there appears to be some kind of dynamicity. So even if we try very hard and we work out, we're just taking one population from size exclusion chromatography, we do see some conversion if we keep them for some time. So that part has been challenging, but this certainly would help in identifying what they do.
Thank you very much. My name is Varun Patel. I work at ICMR and IISc. I had a question regarding the involvement of this protein in what is recently in respect to the essential process in long spreading of the virus during
Right.
TNT.
Yes.
So
Yes.
is anything known, or are you planning to look at how this essentially facilitates infection?
Yes.
So whether tunneling nanotubules and their assembly have anything to do with this protein?
No, that's a very, very good point, and this is something that we are trying to understand in terms of long-term, pathophysiology. But we have several things lined up in terms of cell culture as well as animal models, actually. But we hope to address those questions in future.
Hi,
Yeah,
Thank you for the very nice talk.
thank you.
So I'm curious to know why did it take TF 24 hours to on this program? So you didn't state in the earlier time.
In terms of infection versus transfection. So in transfection, it always takes a little bit more time. Plus in transfection, so in the transfection model, it could also be, but then another thing is that the expression of TF that we have seen in bacteria as well as mammalian cells is not very healthy for those cells because it's a membrane protein. So we see, to some extent, a lot of cells do not express these. So what I'm trying to say is that TF and 6K expression seems, it's no longer it's working. Seem to work better in the infection model as compared to the transfection model. In transfection model, it always takes a while, but we start seeing the puncta within, I would say, 12 hours.
So how relevant is the transfection model?
So the transfection model was for us to have a relatively cleaner system where we could see what is happening to scribble if there is only TF and no other chickpea proteins. So it's kind of like a supporting model. And also initially we did not have the antibody against TF. So then the transfection model was the only way that we could try to address some of the biological problems. But the infection model is way more relevant, absolutely. Probably, yes. Thank you.
And we move on to Ajit Chande. Ajit works on his postdoctoral work identifying cellular antiviral protein cell 5, and he's continuing to have the same interest now that he's at IISc. Welcome, Ajit. And he will be talking to us about retroviruses and RNA surfaces.
Just a second. Can go. So, okay, just a second. Just one.
Thank you very much. It's both nostalgic and privilege to be here. My mentor, Professor Soumitra Das, is there, my long-term friend, and stalwarts like Professor Vijaya and all. Thank you very much for inviting me. So I was part of Molecular Virology Meeting when I was PhD student, and since then I wanted to continue this association. So at IISc Bhopal, we are interested in understanding how retroviruses evolve. So these are RNA viruses. And we try to understand how these viruses replicate in different host range that they have, and why and how they are restricting themselves to the host range that they target. So it is important because in the past sometimes they wanted to jump from one species to the other, and that has not been successful in most of the cases, because thanks to our immune system that they are kept in check from jumping from one species to the other and crossing the species barrier. But some of them, like HIV, outsmarted us obviously, and we are trying to understand what makes this pathogen so successful compared to other retroviruses. So if you compare retrovirus genome of a simple retrovirus that has canonical retroviral gene, it has GAC, pol and on, and in case of complex retroviruses like HIV, it can complicate really things to express accessory proteins by various mechanisms that it introduced to express them. So over the past 15 years, 15 to 20 years, it was thought that these accessory proteins are not required for virus replication and pathogenesis. But it turns out that in fact they are very much needed by the virus, and HIV invested a lot of time in evolving them to antagonize the restriction factors that targets various steps of retrovirus life cycle, right from entry to reverse transcription. Even in the release of the virus particle is targeted by tethrin, and HIV has VPU that antagonizes this restriction mechanism. More recently, we introduced this serine family of proteins that works very early on at the stage of virus fusion with the target cell, and it is efficiently antagonized by NEF protein. But I am not talking about NEF and serine today because it's an old story. What is interesting and not in this picture is this protein called VPR, which remained a mystery for about 30 years. This protein is required for maintaining the viral replication kinetics. This protein is also called as an immediate early protein, obviously required during very early steps of virus replication. And for that reason, probably virus incorporates this protein into the virion. So there are several copies of VPR gets incorporated, and that makes sense. Probably this protein is required for the immediate early process that virus needs, but the molecular basis of that activity remained unknown for so far. We were really not working on VPR when we dissected this mechanism. We were working on something that bizarre gel that we got led to this entire story. We were studying how different exons combine during the process of alternative splicing, and the way you would look at it is by using convergent pair of primers. So that will tell you the combination of exons that appear during the process of transcription and gene expression. What we added additionally, out of curiosity, that we had a pair of primer in the freezer that were divergent in nature, and they are not supposed to give you amplification. And when we looked at the gel, we got shock of our life. The divergent primer got the amplifications. So not throwing this gel, very important for students as a message that if you have something like this, keep it, Sanger sequence it. We did it, and what was happening, these exons were non-canonically joining, right? What we call back splicing. We didn't have any idea of something called circular RNA exists then, but out of curiosity we found out that this exon was joining non-canonically, and in a back splice manner this ligation was happening, and that's why we are getting this kind of amplification with the divergent primer. So really got interested in these kind of molecules because they are very, very stable inside the cells, because they don't have the free ends, so that that can targeted by the that the endonucleases can target and destabilize. So they are very stable structures. What is challenging in infection condition is to really analyze them and characterize them in the native form. What I mean by native form is you cannot really imagine the fraction of circular RNA characterizing using direct RNA nanopore sequencing, because this fraction constitutes less than 1% in total RNA, right? So what you need is essentially a protocol that would allow you to efficiently enrich this kind of species by depleting the most abundant component, ribosomal RNA component, and subsequently poly-A RNA was depleted, and with maintaining the abundance of circular RNA in these fractions, we successively depleted the ribosomal RNA and then poly-A RNA and also structured RNA, and we got a fraction of circular RNA that was enriched up to 50 to 80 fold, enabling us to look at them using direct RNA sequencing approaches like nanopore sequencing. Only to realize that nanopore was not yet ready to sequence circular RNA. Nanopore allowed so far the sequencing of poly-A RNA. So we thought, let's trick the machine and make this enriched circular RNA as a linear one, attach poly A tail, and so that the nanopore can sequence it, considering that it is a poly A RNA. We were very happy to achieve this feat, and only to realize that such kind of data that is generated through nanopore sequencing, there is no computational pipeline to analyze it. So another student of mine, Aman, who was trained by my colleague, bioinformatician colleague, so he did exhaustive work in terms of generating a pipeline that enabled us to finally capture the circular RNA that are associated with infection from such kind of data. So we were very happy. We had a list of circular RNAs that were infection specific. So we characterized them, validated using conventional PCR methods, and we found that 10 of them were really significantly expressed and upregulated in the HIV infection. And our most obvious way is to look at them functionally is to knock down them. So we used CRISPR-Cas based knockdown approach. Only the problem here is in circular RNA. What is unique is that back splicing junction. The rest of the sequence is also shared with the parental RNA or the cognate RNA, linear RNA. So in some cases, some of these genes were really critical for cell survival. So if anywhere we touch other than the back splice junction, the cell was not surviving. So we were restricted to this back splice junction. So we designed guide RNAs and performed knockdown. It was not very good knockdown that you would otherwise get, up to 90%, but we were okay with this condition, and in this condition we found one circular RNA that was really influencing the HIV replication. We called it as citron. So next obvious step was, what is it doing really, right? So when virus infect, the first step it happens is it reverse transcribes the RNA genome into the cDNA. So we looked at the RT product in the context of the circular RNA knockdown and the control conditions. We saw there was no difference. Right? We used appropriate control, as it is a reverse transcriptase inhibitor, and the assay was working fine. We looked at the subsequent step, that is integration of provirus. When it is integrated in the proviral DNA form the chromatin. So we thought there is a difference, but there was no difference at the integration step either. What was interesting that the transcription from this integrated provirus, there was a difference when we detected, and all these steps we done from the subcellular fractions that we collected, the cytoplasmic fraction, nuclear fraction, and so on. So the cytoplasmic mRNA that is transcribed from the integrated provirus, there was difference in condition when the circular RNA is knocked down. Now this circular RNA has nothing to do with HIV. It is not from HIV. It is not known to have any link with the HIV in the literature, and we were really surprised to know that an independent event that is happening, a circular RNA, when we knock down, there is an effect on HIV transcription. We went back to the literature, and the way the circular RNAs are most abundantly known to regulate gene expression is by sequestering microRNAs. So they can sequester a lot of microRNAs, and that way they can regulate the gene expression. But then it was not true, because what we are seeing, the knockdown of the circular RNA was leading to defect in the HIV transcription. And also the circular RNA that we found, it is very small, 280 or 260 nucleotides, and it was there was no any microRNA binding site. So we were very pleased to see the emerging functions of this circular RNA, that these can actually sequester proteins also, RNA binding proteins, and obviously they can regulate the functions. They can even bring substrate and enzyme together, and they can regulate gene expression, and they even get translated with the help of IIRES, right? So we were very pleased to see these kind of roles coming up. So we hypothesized that there is a proteinaceous factor that is actually being regulated by the circular RNA. And to understand what protein actually is interacting with the circular RNA, we employed this method called CARPET. Essentially what we are doing is we are placing, we are installing catalytically dead Cas, which is having BASU, that is biotin ligase. So when this catalytically dead Cas is interacting with the circular RNA, the biotin ligase is going to biotinylate the proteins that are in proximity to the circular RNA and interacting with the circular RNA. We used a conventional approach, and in silver staining gel, in the case of biotin pulse fraction, what we saw that there was a clear distinct band. When we subjected it to mass spec, we found SRSF1 as a top hit here. So this protein was essentially interacting with the circular RNA. That's what the result told us. So we needed to validate this mass spec data, so we used Park-Lip method. So Park-Lip method is essentially what you do, you cross-link all the RNA and proteins, and then you use protein-specific antibody and see whether that RNA is interacting with the protein. So when we use SRSF-specific antibody, we saw that SRSF was significantly interacting with the circular RNA. Now SRSF is known for about 10 years that it inhibits HIV transcription, only in condition when you overexpress it. So this protein levels don't change when there is infection or versus normal conditions, right? So this is a constitutively expressed protein, so virus is having a mechanism to counteract or antagonize this constitutively expressed protein, the effects of which are only seen when you overexpress, right? So you can see the difference vector versus SRSF overexpression. What is interesting here, we are bringing a new player that these kind of effects can be phenocopied by knocking down a circular RNA, which is not encoded by the SRSF transcript, neither by HIV nor it has any link with these two proteins. So independent events are happening. A circular RNA, if you downregulate, it phenocopies the effects of SRSF overexpression. Now question is, the knockdowns can be non-specific, right? So we proved it by overexpressing the circular RNA. So you are here basically, SRSF is inhibiting the HIV transcription, and you can rescue the viral infectivity just by synthetically overexpressing the circular RNA, right? Suggesting that this event is specific and there is some kind of competition. The more levels of protein are balanced by more levels of circular RNA, so that you can achieve in these kind of experimental conditions. So essentially we found this interplay with respect to SRSF1 and the circular RNA which we named citrin. How relevant it is for primary infection? When the PBMCs that are infected with HIV, we saw that the circular RNA is upregulated, and the CD4 cells isolated from these healthy donors and infected with HIV, we saw that circular RNA is significantly upregulated. Now a constitutively expressed protein is being antagonized, so there must be a mechanism that virus uses to induce this circular RNA and use this circular RNA or hijack this circular RNA for its own replication. So we suspected an involvement of viral protein, and the way you would look at it is by using mutants that are unable to express these viral accessory proteins and other viral proteins. And guess what? The virus that is defective in VPR was not able to induce the same circular RNA. And not just that, if you take VPR and clone it in pcDNA and transfect it, you are able to see much better circular RNA induction alone with VPR. That means VPR protein is alone sufficient for inducing this circular RNA. And this VPR we used NL43 backbone, which is B-type clade B isolate. So we used, and we have extensive panel. Just in the interest of time, I am not going through it. The primary VPR from primary isolates are also doing same thing, but the VPX, which happened during the gene duplication event from HIV2 lineage, is not able to do. So it is HIV1 specific adaptation that has happened. So I mentioned that VPR gets incorporated into virions, right? So we were interested to know whether virion packaged protein itself is capable of inducing this circular RNA. So we fused VPR with GFP, checked its incorporation in virions, and this is not the VPR that is encoded by virus. We are packaging in trans. So this encapsulated VPR was enabling this circular RNA induction and also in macrophages, and we checked in primary cells also. So how relevant it is for the primary isolates that are in clinic and that are in field. So we checked that SRSF binding site in the LTR promoters of field isolate is quite conserved, right? So this prompted us to clone this LTR from clinical isolates and check whether this really citrin and SRSF axis is relevant for the clinical isolates. So whenever we express the SRSF1 and do this luciferase assay, which is driven by LTR and transactivated by TAD, whenever you had SRSF, there was suppression of luciferase expression, and that was in every case invariably antagonized by, rescued by circular RNA expression, suggesting that more SRSF you can actually titrate out with the help of this circular RNA. To cut the story short, so basically relevant for primary isolate, we have checked from patients also that data I am just not showing with the To cut the story short, essentially what you have is VPR that is encoded by virus and also incorporated by virus that induces the circular RNA expression. And this circular RNA then sequesters a protein which otherwise inhibits the HIV transcription. So it binds to the HIV promoter in the absence of this circular RNA, and that is the condition where virus didn't have VPR. So in the absence of VPR, circular RNA levels are low. This protein is free to bind to HIV transcription complex and inhibit the HIV transcription. And the way it does it by displacing the TAT, which is really required for HIV transcription. So the first paper in this series of circular RNA, and incidentally this is the first circular RNA that is characterized in HIV infection and known to have effects like effects on transcription. So everything was done mostly by my student. Experimental pipelines were developed and the protocols we had to start everything from scratch because we are no RNA biologists, and we had to develop all the reagents that to express circular RNA, synthetically make them, all those things were done by my student Vipin. Aman developed pipeline. He is co-supervised by my colleague Dr. Nagarjun. We got reagent help because we use a lot of reagents and that was supported by IISc's reagent program, and Massimo was my postdoc mentor who was very helpful and providing the crucial reagents that we needed. And it was not possible perhaps without India Alliance, because these kind of crazy ideas India Alliance supported, and we were very lucky to have funding that was enabling us. Thank you very much.
Very nice work, Pradeep.
Thank you.
I have two quick questions. First one is SRSF1 is a part of the nucleus speckles. Now, what happens in the viral infection? You think of all the circular RNA if you overexpress.
Yeah.
They get destabilized and there is a reprogramming of nucleus speckles. The second question is, do you think the circular RNA can contribute to the HIV latents?
Both are very interesting questions. So we tried to just manage to see this circular RNA. So again, the problem was signal is not very strong because we are targeting the backsplice junction. So we are enhancing the signal. But we see that in condition, whenever there is infection, we see strong co-localization of the circular RNA and SRSF in speckles, right? And those are distinct ones from where the HIV is transcribed. So that's what we are optimizing now. We are trying to see the HIV transcription and this localization of SRSF and circular RNA, whether these signals are merging. The initial experiment says they don't merge, and that makes sense probably. But we need to do more work and refine these techniques. To address second question, that most of the simple retroviruses prefer to become latent and become endogenous down the line, right? And probably the goal of these retroviruses is also to become latent eventually. And these kind of mechanisms are there to make it silence. We see more pronounced phenotype from the macrophages. And that makes sense. They make latent reservoirs of for HIV. But we don't have strong experimental data to really say that it is true. Yeah.
So I have a question. So in your circular RNA sequencing study, did you identify any virus-derived circles? Because I think there is some evidence now of virus-derived circles also.
Correct. We have, but we don't know what they are doing actually. Right? So why virus is making circular RNA? First of all, we get a lot of noisy data, and that is obvious because you have RNA-seq, and we were lucky to have found these circular RNAs because most of the reads that in the infected cells are represented by the virus, right? So in that condition, it becomes very tricky, and you have a lot of complications like transplicing, missplicing. So that is why the primary reason why we decided to go with direct RNA nanopore sequencing, so that whatever we see is true, is not an artifact of library preparation or the infection-induced changes that we see. But yes, we need to characterize that. Yeah.
Thank you for the nice talk. So, you mentioned that SRFS, SRF1 is a avalent protein.
Yeah.
So, I'm assuming so should be side tram And in those lines, do you have any idea of the stoichiometry, how many molecules of SLSF1 does that?
We managed to get the number of molecules, although I mean we had to devise a way to do it. About some 30 or 50 molecules per cell we detected for this circular RNA, and we can go up to 400 by overexpression. And the SRSF levels are not changing. So when we overexpress, we see very clean phenotypes. So, but stoichiometry in terms of the SRSF binding on the circular RNA, there are 7 binding sites. So in theory, that 7 SRSF molecule, 1 circular RNA can sequester. And as Professor Samitra Das mentioned, that it is part of speckles. So these things happen much more pronounced in these kind of compartments. Milan.
Probably misstate, where is the location of this circular RNA?
We find it in nucleus and some fraction in cytoplasm also. So this is encoded by a gene called SMARCA5, which is part of this YSNF complex. Chromatin, yes, it is in the coding region. Exons 16 and 17 are forming this circular RNA. And the way we, I forgot to mention, we call it citron because this is the first circular RNA that is known to regulate HIV transcription. So that's why we are calling it citron.
It has no translation regulatory element.
We don't find any signature of IRES or M6 modification so that it gets translated or, yeah, neither microRNA binding site.
Can I have the last
Yeah.
question? In your data, you did not show the effect on the 2LDR circuits. So do you know whether there's any effect on non-integrated circuits?
Yes, so non-integrated circles, there is transcription that is going on, and we sort of recapitulated those events when we transfect the molecular clones. In that condition also we see these effects when we overexpress SRSF, and that can be titrated out. So with 2 LTR circles also it will be true. Only I don't know how it works in the case of context of IISc that actually modulates the 2 LTR circle.
How does it circularize? Are there overlapping and hybridizing sequences?
It has 7 binding motifs for this SRSF.
So the protein circularizes.
No, the protein binds to the circular RNA.
I know, but the circular RNA, does it have sequences that are complementary to facilitate circularization?
For circular, yeah, that's a good question. That I didn't show the data. We know that it is actually circularized with the help of this protein QKI. So in fact, SRSF1 is well-known splicing factor, right? So we initially thought the splicing of the circular RNA or back splicing is regulated by SRSF, but that's not true. In SRSF knockdown cells, we don't see the levels going down. In fact, they go up much more. So we suspected another protein, and we did partly to find out QKI was there. That was binding to the flanking introns, and that was mediating the circularization, bringing these 2 exons together. So if 280 nucleotides, just very small circular.
So no LTR sequences?
No LTR sequence. No. It's very interesting that the abundance of circular RNA sequesters SRSF. SRSF also binds to HIV LTR in the TAR region, replacing the TAT. But then what determines these affinities? I mean, we need help from structural biologists actually at this point to understand.
So I will thank Rajat Chandu now, and we will move on to
Thank you.
the next speakers, which is Dr.
Our third speaker for this session is Dr. Indranil Banerjee from IISc. And he has been working with human pathogenic viruses that have a pandemic potential, particularly influenza A virus. This group has been particularly interested in targeting host factors which viruses manipulate with their molecular hack codes to design novel therapeutic strategies. And his topic or his talk is entitled Cracking Open a Hard Nut, How Host Factors such as TRIM62, and taxonomy promote influenza A virus nucleocapsid uncoating
This is how it will go.
Over to you,
Do you want this or just this
This fine. Yes. Thank you for the nice introduction. And first of all, I'll begin with thanking the organizers for inviting me to this excellent meeting. This is my first molecular virology meeting. Although it's the seventh, many people here attended the first one. But I must tell that I was introduced to the field of virology right here at IISc. When I was a master's student at BHU, I came here for a summer internship in the lab of Professor K.P. Gopinathan. And that was my introduction to virology, and still I'm continuing in this field. Today I'll be talking about, in general, how viruses uncoat during their entry into the host cell. And that's a paradox. It is called assembly-disassembly paradox. And here we have found that two cellular proteins, TXLNA and TRIM62, help in this uncoating process, but in a distinct ways. Advancing. Okay, so coming to the virus assembly-disassembly paradox, like the question is, why a virus particle which is extraordinarily stable in the outside environment suddenly falls apart when it enters a host cell? In the environment, it has to endure different harsh conditions like varying temperature, mechanical stress, desiccation. And when it enters the cell, it has also to encounter different conditions like hostile environment in the cell, immune response, then actions of proteases, and other factors. So the question is, this virus particle, when it enters, it falls apart because it has to transmit its genome into the cytosol, and this genome goes to the nucleus for influenza viruses, and for other viruses it can transcribe. But after a few hours, the same virus produces progenies which are extremely stable in the same cell. When they go out, they remain stable, and when they come back to the cell, they again crumble, they fall apart, and they unpack. So in the outside environment, the virus travels in a transport configuration where the genome is condensed and the virus particle is in a very solid state. It is very compact, and the same is reversed when the virus comes to the cell. So two highly motivated and talented students, Kajal and Gagan, they decided to work on this Lego ball, and especially to study what it takes to crumble and crush this Lego balls into smaller pieces. And they asked this question, like, how does influenza A virus exploit cellular factors for capsid disassembly and genome unpacking? So this is the overall structure of influenza virus. It has a spike glycoprotein hemagglutinin, neuraminidase, which is responsible for the cleavage of sialic acids, and also it is responsible for productive binding of the virus during entry. It has its genome segmented into 8 segments, which are then complexed with the viral nucleoprotein, or NP, and then it has N2 ion channel present on the membrane, which allows the passage of monovalent cations. And it also has this M1 capsid protein, which makes the shell of the virus. During the assembly process, the viral proteins are assembled at the plasma membrane in a hierarchical manner, and the interactions happen between the spike glycoproteins with the M1 protein, and then the VRNPs come and they bind. But when the viruses enter the cell, it has to undergo the similar reactions but in a reverse order. And that is a question that we ask, that whether the viruses are self-capable in doing so. Recent studies showed that chemical cues are required for this unpacking to happen efficiently. It was long known that the viruses, once they enter the endosomes, they'll get acidified and there'll be conformational changes. But in vitro, it has been very elegantly shown by Miyake that if you subject this virus through this glycerol gradient at a neutral pH, the viral nucleoproteins, or the VRNPs, are unable to dissociate from the M1 capsid. But if you do the same experiment at pH 5.4, you can get very nicely the VRNPs which are clustered together and which are dissociated from the core. And Sarah Stauffer showed that not only the protons, but if you treat the viruses with high level of potassium, then the viruses are primed for this dissociation event to occur. With this, we'll start with the strategy that we have used. So we wanted to look at the virus entry at every step. It's a multi-step entry process. So we first started looking at the virus at early endosome, which is RAB1 and EA1 positive vesicle structures, and the viruses bind to different receptors like EGFR, nucleolin, etc. And using a high content screening approach, we could differentiate the viruses which are internalized and the viruses which are surface bound. Here you can see the green dots are the viruses which are internalized, and the red dots are the ones which are still outside. The next step is a virus acidification. While it's journeying through the endosomes of virus, take the acid birth in the late endosome, and that triggers a conformational change in the spike glycoproteins, which exposes a fusion peptide, and this fusion peptide anchors to the endosomal membrane, and the viruses, and this acidification event is very nicely probed by an antibody which detects the acid conformation of the hemagglutinin. This is followed by virus fusion, where the viral membrane fuses with the endosomal membrane. And this is where we're interested in. This is uncoating. Once the virus fuses at the plasma membrane, the capsid protein disassembles and it disperses throughout the cytosol. And if we use an antibody against this capsid protein, M1 protein, then you can see the dispersal of M1, whereas if you block the acidification, the viruses are restricted to late endosomal compartments, and you do not see this dispersed signal of M1. That is our assay which we employed to address the questions of virus assembly disassembly paradox. So talking about uncoating, it was believed till 2013 that upon entry the virus is exposed to the acid environment in the late endosome, and then fusion happens, and after that the viral capsid uncoats. But things got complicated since 2013. Many groups showed that it is not only the chemical cues which are required for uncoating, but cellular factors are also critical for this uncoating to happen efficiently. The first clue came from this group, Lai, who showed that each is required. It's an E3 ubiquitin ligase for efficient uncoating of influenza. Then we showed that while its entry, influenza uses agrosome processing machinery where it employs various proteins like HDAC6, actin, myosin, tubulins, etc. Then Yohei Yamaguchi's lab showed that upon uncoating, the debundling of the VRNPs happen with the aid of transprotein 1, and recently EPS8, another protein which has also been implicated in this encoding process. We have recently identified that two other proteins, TRIM62 and TXLNA, are also involved in this process. How did we get into these proteins? So in collaboration with 3B Biosciences, we conducted a druggable genome screen where we screened for the cellular factors responsible for infection, and we found about 177 factors. And out of these factors, TXLNA and TRIM62 were two of the best hits. So it's a brief introduction about TRIM62 and TXLNA. TRIM62 is a member of a TRIM protein, which is containing an E3 ubiquitin ligase domain. Here it's called the ring domain. It's followed by a B-box domain, which is responsible for substrate targeting. Then a coil-coil domain, this is responsible for homomeric or heteromeric interactions with other proteins, and a PRI, SPRI domain, and is involved in different functions like development and differentiation, autophagy, innate immunity, and antiviral response. However, the interesting fact is most of the TREAM proteins which have been implicated in viral infections are restriction factors, whereas TREAM62 we have found is a supporting factor for influenza. And TXLNA is a very simple molecule. It has a very long coil-coil domain, and then it has two disordered regions at the C terminal and the N terminal, and it is implicated in regulating trafficking, vesicular trafficking, also the release of HBV and HCV, etc. And it is also a supporting factor. So coming to the functions of TREAM62, we depleted TREAM62 by siRNAs, with using two independent siRNAs. I could find that the virus infection was substantially blocked. Unfortunately, we could not get an authentic antibody against TREAM62, so we had to rely on RT-PCR to look at the knockdown level. We also generated CRISPR-Cas9 knockout cells of TREAM62, and we found that influenza infection was significantly attenuated. We saw also in the plaque assay that the infection was down. When we overexpressed TREAM62, we observed a post-infection, further confirming that TREAM62 is indeed a supporting factor. We then tried to rescue the phenotype by expressing TREAM62 back into the knockout cells, and we could partially rescue this infection phenotype. And this is TXLNA data. Very similarly, we found that upon knocking down of TXLNA, influenza infection was attenuated. And when you add back the TXLNA in the TXLNA knockout cells, infection was restored. With this, we addressed that which step of virus infection these two proteins are involved in. We first started with endocytosis assay. We saw there's no difference in acidification. There was little difference for TREAM62, but not major difference for TXLNA. Fusion was almost unperturbed for both, but we surprisingly found that both the proteins have function in the virus uncoding. And we also tested the subsequent step, that is virus nuclear import. There's the import of the VRNPs into the nucleus. That is also impacted upon knocking down or in the TREAM62 or TXLNA deficient cells, confirming that uncoding is a major step that these proteins are involved in. Next, we addressed that what is the mechanism. So we looked at the cells which are expressing LAMP1. These are fixed cells, and you can see we stain the late endosomal or lysosomal vesicles with LAMP1 antibody. And in the red, you can see the influenza M1 signal, which is supposed to be dispersed during 2 or 2.5 hours of infection. You can see this dispersal signal here. However, strikingly, we found that in TREAM62 knockout cells, these vesicles are rather enlarged, and the viruses are restricted. They're trapped in this late endosomal compartment. On the other side, in the TXLNA knockout, the phenomenon was different. We observed that late endosomal compartments are rather collapsed, but uncoding did not take place in both the cells. So that prompted us to think that maybe TXLNA and TREAM62, although they are both involved, both are involved in uncoding, but they're following two distinct mechanisms for this uncoding event. So we firstly checked the transfer and recycling and addressed like whether this effect in vesicular trafficking is virus specific or it is in general. It's a general effect. So we performed a transfer and recycling assay in the TREAM62 knockout cells, and we found that transfer and recycling was significantly delayed. We also performed EGF degradation assay. We found that EGF degradation was delayed in TREAM62 knockout cells. We then pulled down TREAM62 and performed a mass spec analysis, and we surprisingly found a high enrichment of actin-related genes, actin and myosin-related genes. And very recently, it was shown that one of the TREAM proteins, the TREAM27, is involved in regulating vesicular trafficking, especially in regulating the actins around the endosomes. So these are endosomal actins. And there are other proteins like capping proteins which are involved in the in restricting the length of the actin proteins. And we also showed that actin and myosin and tubulin, so these proteins are involved and they're critical for the uncoating event to happen. So we then looked at the cells which lacked TRIM62, and this is how a normal A549, this is a lung epithelial cells would look like, and this is a top view where you can see the branched actin, and this is a ventral part of the cell where you can see the long thread-like structures. Very strikingly, when we looked at the TRIM60 cell, we found that there's a high upregulation of the polymerization of actin, and we could find the actin bundles running parallel, and that is true for both the top and the bottom of the cell. And you can see here, this is the color-coded actin for height, and in the top you can see there's a branched actin which are mainly catalyzed by ARP2C complex, and at the bottom you can see the ventral filaments and the actin arcs which are kind of surrounding this region. So then we moved on, and what we observed that in the wild-type cells, while the LAMP1 positive vesicles are present both in the perinuclear region as well as to the other parts, and this is a live video, in the TRIM60 knockout cells, the LAMP1 positive vesicles are restricted only to this region where the actin branching is happening, but they are kind of barred from the region which are having this long threads of actins running parallel. So it appears that they are kind of caged, and these actins are barricading this LAMP1. So I'll have to move fast here. So then we found out that these actin regulators around the endosomes are involved. That is, we pull down a WASH1 complex, WASH1, tubulin, and CABC proteins, and surprisingly we found that all these endosomal actin proteins are pulled down by TRIM62, and we did the live cell imaging. You can see here that WASH1 is very nicely recruited on the endosomes in the wild-type cells, whereas in the TRIM60 knockout cells, they're kind of dispersed. So this tells us that WASH1 complex is probably regulating the endosomal actin, which is lacking in TRIM62 knockout cells. And I'm just cutting this story short here. We also knocked down CABC proteins, and we found that CABC proteins, which are limiting the length of actin, these are also important uncoating factor. If you knock them down, uncoating is affected. Now very quickly coming to TXLNA, and what we also found that in the TXLNA cells, knockout cells, the LAMP1 positive vesicles are collapsed, and also the early endosomes are enlarged. As you can see here, this is a wild type cell, and this is a knockout cell. You can see the enlarged early endosomes, which is reversed when we add back TXLNA in the knockout cells, and this we quantified. And then we address that what causes this endosomal maturation defect. Gagan then carried out the screens against all the proteins of Corvette and Hobbs complex, which are responsible for endosome to endosome fusion, and also the ESCOT machinery. And this is an Easter hybrid screen, and from which we saw that there's a protein called VPS22, which is a member of the ESCOT2 complex, is a potential interactive. And then we looked at the endocytosis and acidification status in the VPS22 knockdown cells, and we found a similar phenotype like TXLNA or TRIM62 knockdown cells, that the uncoating is blocked if you knock down VPS22, which is a ESCOT2 protein. And we also found that EGFR localization is perturbed in the TXLNA knockout cells, and EGF and EGFR residency in the early and the late endosomes is increased, and this is reversed if you add back TXLNA. And I'm skipping this part, but very recently it was shown that these two degradative pathways, endosomal pathway and autophagy pathway, they are cross-talk to each other, and there could be implications then that TXLNA would be involved in this autophagic process. And when we looked at TXLNA knockout cells, we found enlargement of the autophagic vacuoles and upregulation of a LC3 level, which is a marker for autophagy. And we observed that there's a significant difference in the LC3 levels between the TXLNA knockout and the wild type cells. In the interest of time, I'm just skipping some part. I just show here that in TXLNA knockout cells, autophagy per se is downregulated. And we also use different autophagic markers, autophagic inhibitors, chloroquine, monensin, watmanin, rapamycin, and we saw that influenza uncoating was perturbed upon addition of these compounds, especially chloroquine, monensin, watmanin. But if you increase autophagy, so these are all blockers of autophagy. They induce the formation of autophagosome, but they block the fusion of autophagosomes with lysosomes. But if you use rapamycin, that is an inducer of autophagy, then we also see that the uncoating was down by 40 to 50%. And I'll just skip this part. So coming to the summary, we found that TRIM62 recruits wash complex, which is a mediator of actin assembly around the endosomes, and that is how it is responsible for efficient uncoating. And TXLNA is interacting with VPS22 so that it aids in the maturation of endosomes, and also it regulates autophagy, and this ensures uncoating at the end. With this, I come to the acknowledgement. This is my group. Some people have left, some new people have joined, and this study was mostly done by Kajal and Gagan, who are also presently posters here. Tejal is also here. She is also presently posted, but her work is not shown here. And thanks to all my granting agencies, and thank you for your attention.
Thank you, Dr. Banerjee. It's open for questions. Yes.
Nice talk. I have a question regarding, you have shown that the nuclear import is also getting downregulated. Is it because of the inhibition of the uncoating, or is there any specific role that institute can play in the import as well? Because other members of the family has been shown to have played a role in the import. Are you looking into that as well?
Yes, so the reason we think that uncoating is a reason that we see a block in nuclear import, because uncoating is a step ahead of nuclear import. So if you block uncoating, that is expected that nuclear import would be blocked. It also depends on the extent of uncoating that you're seeing and the extent of nuclear import that is happening. If the uncoating is happening, say 50% and nuclear import 25%, then there could be some reason that the nuclear import, or the TRIM62 could be involved in nuclear import. But we see the levels are almost the same. It is about 25 to 30%. So it's correlating. So we believe that it's uncoating. That's the reason the nuclear import is blocked.
Hi, very nice talk. I just had one question. So the two host factors, they are in general what could be involved in the uncoating of any virus which goes through the endosomal pathway, right? I mean, it's not specific to
Absolutely. We have not tested other viruses, but would love to test.
So these could be general?
This could be general. This could be general.
And also the capsid protein for influenza virus. So this isn't icosahexaene, it's a pleomorphic virus, so the coat is not icosahexaene.
No, the code is not icosahexaene.
So
it's
pleomorphic. Very stable, you are saying in general about viruses.
In general, yeah, that was a very general statement. In general, the viruses are stable, but influenza, we know that once it enters a endosome, it takes an acid bath and it becomes metastable. Right. And this priming is important for uncoating. If the viruses are not primed, they are unable to uncoat even if the presence of low pH.
Because in general, viruses with icosahexaene coats are
they're most
stable.
Yes. Yes.
Thank you.
Yes.
Very nice talk.
Thank you.
So in light of your data, how critical is a low pH now for uncoating compared to these? Would a low pH still be as critical?
Absolutely. Low pH is an essential requirement for uncoating. Without low pH, the interaction between the spike glycoproteins and the capsid proteins will not be abrogated, and also the low pH ensures the dissociation of the capsid proteins from the VRNPs. So low pH would be required.
Combinations can be involved in the capsid degradation, or would it just be low pH?
So before 2013, people believed that low pH would be sufficient for the fusion and uncoating to happen. But then we saw that even at the low pH, the viruses can fuse, but somehow they're unable to break open. And for this even to happen, the virus would require the participation of the host factors, which are gradually being identified like there are 4 or 5 host factors which have been identified. I'm pretty sure there'll be more. And maybe some of the host factors will work in concert with each other for this efficient uncoating. Very good question. Yes, we have observed, there's no paper as such, but we have observed that after 30 minutes of this dispersal, the capsid protein is indeed getting degraded. Yes.
So you mentioned that potassium also destabilizes your capsid.
It primes the capsid. It helps in the dissociation. So people generally believed that it was a proton or acidity in the lead endosome that would cause this dissociation or that would prime the virus for the dissociation. Sarah Stauffer showed that if you add potassium to the virus, you enhance the efficiency of this dissociation. And it is now believed that during its journey or during its residency in the endosomes, the virus is exposed to both protons as well as potassium. And this potassium is entering the viral code along with the protons through the M2 channel. And this is further helping in the dissociation of the proteins, dissociation of the capsid protein from VRNPs as well as from the spike glycoproteins. So it's enhancing the effect of dissociation.
Just one more question.
Yeah, just general question. Whatever you have shown, is it cell type dependent, and you are actually relying all your data based on the cell type reason, or do you think this could change to actually go to a different cell?
It's for some of the experiments we tried out some other cells like HEC293 or HeLa cells, but most of the experiments were done in A549 cells. But we believe that it is not a cell type specific or virus specific. We also used different viral strains, and we saw that the effect remains the same for both the proteins.
You did not use NDCK cell lines?
We use NDCK cell lines for propagation of the viruses, but this is a canine cell line, and these are human proteins. So we did not have the reagents to really create a knockout in the NDCK cells. Thank you.
Thank you so much, Dr. Banerjee. Move on to the next talk, which is a talk on phosphomimetic adaptations leading to fine-tuning of influenza virus RNA synthesis and virus replication. This is by Dr. Arindam Mondal from IIT Kharagpur. And he's going to be talking about focusing a lot on the role of those factors that will drive viral RNA synthesis.
Thank you. And first of all, thank you, the organizers, Professor Das, Shashank, Keshav, and all the research scholars and the students who are working tirelessly to make our stay convenient at the IISc. It's great to be back at the IISc campus. And today I'm going to talk about how phosphomimetic adaptations, fine-tuning the influenza virus RNA synthesis and eventually virus replication. So before I get into the influenza virus biology, I would like to talk about the phosphomimetic adaptation. So we all know about the serine or threonine residues, which are basically polar uncharged residues and can be phosphorylated by the kinases in order to get themselves converted into a negatively charged phosphoserine or phosphothreonine residues. Now these phosphoserine or threonine can well mimic the structure of the negatively charged glutamic acid or aspartic acid residues. That's why in biochemistry we oftentimes change the phosphoserine or phosphothreonine residues with the glutamic acid or aspartic acid in order to have an understanding of constitutive phosphorylation at a particular position of the protein. Now today what I'm going to show you is how influenza viruses are changing from serine into glutamic acid as a part of the phosphomimetic adaptation in order to fine tune the replication of the virus and hence shape up the virus evolution. Okay, so influenza viruses falls in the Orthomyxovidae family. There are 4 different kinds of influenza viruses, influenza A, B, C and D viruses, amongst which the influenza A viruses or avian influenza viruses are the most widely varied one. Now there are 18 different subtypes of the surface glycoprotein hemagglutinin and the 11 different subtypes of the neuramidases, which together defines the different subtypes of the virus, for example H1N1, H3N2, etc. Now among these different subtypes, all of the viruses including from H1 to 16 and N1 to 9 are circulating in the wild aquatic birds, which are the natural reservoir for these viruses, and from there it can acquire adaptive mutations in order to cross the species barrier to infect a wide variety of host species. Now in recent past, during 2012 and 13, 2 newer subtypes of influenza viruses has been isolated from bats. These are basically H17N10 and H18N11 viruses, which has never been isolated from any other species so far. And post their discovery, a lot of concern were raised about their zoonotic or epizootic potentials. That means whether these viruses can cross the species barrier in order to infect humans or other animals. But that doesn't seem to be the case. So, it seems from the phylogenetic analysis, the bat influenza viruses has been classified as an isolated clade which remain isolated within the bats throughout the course of the history of influenza viruses. They have never been able to mix up with the classical strains which are here. In our lab, we study the influenza virus replication machinery. So 2 of the students, so this is Shoptoshi was the first PhD student from the lab, who initiated this project back in 2018 in order to look into the viral RNA polymerase and whether there are specific differences in the viral RNA polymerase of the bat viruses which make them restricted into their specific hosts. Now the project is being carried out by Arotvika, who is the 3rd year PhD student. Now you look into the influenza virus replication machinery. This is a negative sense RNA virus which contains 8 different segments of the viral genomic RNA, and each of these negative sense RNA remains enwrapped with multiple copies of viral nucleoprotein which forms these kind of double helical hairpin-like structures. And each of these hairpins contains a single copy of RNA-dependent RNA polymerase. We call them a resident RNA polymerase, which is constituted of 3 different viral proteins, the PB1, PB2 and PA viral proteins. So this is the self-sufficient machinery for the virus which can perform different modes of viral RNA synthesis including transcription and replication. So at the early stage of replication, the viral RNPs perform transcription whereby the viral RNA-dependent RNA polymerase snatches the cap structure from the host pre-mRNA and then utilize it in order to initiate the transcription so that they can produce a capped and polyadenylated mRNA indistinguishable from the host mRNA so that they can be easily translated by the host ribosomes. Now at the later stages, when you have sufficient amounts of the viral RDRP and the nucleoprotein got synthesized, they actually support replication whereby the newly synthesized RNA pol forms a dimer with the resident RNA pol, and then this dimer then performs the viral RNA replication in order to produce the positive sense RNA, which then gets concomitantly encapsulated by the viral nucleoprotein in order to synthesize a complementary viral RNP. Now this positive sense RNP then can act as a template for the synthesis of the negative sense RNPs, which then gets used for subsequent rounds of transcription or can be packaged into the virion particles. So you can see that the viral RDRP can execute transcription at the early phase, replication at the later phase. How this transition from transcription to replication occurs. So this is the structure of the actively transcribing polymerase bound to the negative sense RNA promoter. And here you see 3 of the different subunits constitute a U-shaped structure where PB1 constitutes the active core of the polymerase, and in this structure the PA endonuclease domain faces the PB2 cap binding domain so that the PB2 can bind to the cap structure of the host pMRNA and then PA endonuclease can cleave it off 15 to 20 nucleotide downstream so that it can generate a capped RNA primer. Now in this structure, this PB2 627 domain, which is a well-known determinant of the host tropism, remains distant apart from the PA endonuclease domain.
Now, in the replicase structure, there is a major structural rearrangement between the 627 domain and the cap binding domain, whereby they undergo a 62-degree rotation, and hence now the 627 domain is closely juxtaposed with the PA endonuclease domain so that it can block the cap snatching activity. Okay, rather it triggers the dimer formation of the polymerase, which is the active replication form of the polymerase. So you can see there is a structured transition of the PB2 subunit of the polymerase, which can transition from the transcriptase to replicase mode. Now, in order to study the transcription and replication of the virus without having an infection, in the lab we have generated a series of viral reporter RNA constructs where we have changed the viral open reading frame with the luciferase gene, keeping the 3' and 5' UTR constant. And once you transfect these reporter RNA constructs along with the viral protein, along with the plasmids which can express the viral proteins, the NP, PB1, PB2 and PA, they together form the reporter RNPs within the cell so that they can express the luciferase as a result of viral gene expression. So you measure the luciferase in order to have an overall viral gene expression pattern, or you can isolate the RNA in order to measure the viral transcription or replication product. So we have utilized these primer extension analysis in order to show that actually the H17N10 polymerase, which is the bat influenza virus polymerase, replicates poorly with respect to the H1N1 polymerase inside the human cell. So next, our students performed an extensive bioinformatic analysis coupled with function screening in order to identify the specific residues which are different in these two polymerases and might be responsible for this particular attenuation. And by doing so, I am not getting into all those bioinformatic analysis, but that screening helped us to identify this particular motif, SES motif present in the PB2 subunit of the polymerase, which is not only conserved throughout all classical influenza A viruses, but also for the influenza C viruses. If you look into the influenza B and D viruses, there is not the conserved SES motif, but both of them have two consecutive glutamic acid residues, the negatively charged residues at the first two positions of the motif. So this suggests that the negative charge, at least one negative charge at these motif is important for the RNA synthesis. Now in the case of the bat influenza virus, they do not have any negative charge, rather they have a SST motif in the same position. So, in order to test the importance of the motif in the context of the human adapted H1N1 virus, we have reconstituted the polymerase activity assay both in human as well as chicken cell line. DF1 is the chicken cell line. And we have introduced mutations in the motif. You see, when we have completely abrogated the motif by introducing a triple alanine mutation, or have introduced a positively charged lysine in presence of the glutamic acid, we completely lost the polymerase activity. And that's in a post-independent manner. Furthermore, when we have changed the SES motif, which is a human virus specific motif, to a bat virus specific motif, for example SST or SSS, there are also about like 60 to 70% decrease in the polymerase activity, suggesting that the SES motif is important for supporting viral RNA synthesis, at least in the context of the polymerase activity assay. But what about the viral infection, right? So in order to test it in the context of virus infection, we have generated recombinant H1N1 influenza viruses. So in the wild type H1N1, you have the SES motif. We have changed the central E to S in order to constitute a bat-like SSS motif in the context of H1N1 virus. And these viruses showed smaller plaque phenotype. Not only that, they replicated poorly with respect to the wild type virus, not only in the human cells but also in the bat cell lines. So together we concluded the first story in this section, where we showed that the SES motif present in the H1N1 polymerase is essential for polymerase activity and virus replication. H17N10 polymerase harbors a SST motif, and if you introduce a SST motif in the H1N1 virus, the polymerase becomes defective, the virus becomes attenuated. But then the question was, what would happen if we introduce a human-specific glutamic acid, human virus-specific glutamic acid in the bat influenza virus, that is H17N10. So in order to answer this question, we started working with the bat influenza virus polymerase. Okay, and here you have the wild type motif. To our surprise, when we completely abrogated the motif by introducing a triple alanine mutation, there is only 64% reduction, which is in contrast to the 94% reduction, which in case of the H1N1 polymerase. So we looked into the structure of these two polymerases. So structurally they are conserved, okay. But then You see in the case of the H1N1 polymerase, because you have a SES motif, you have a surface exposed negatively charged patch, which is completely destroyed when we introduce the triple alanine mutation. In the case of the H17N10 polymerase, you have the SST, polar uncharged residues, right? That's why you don't have a negatively charged patch here. So when we are changing it to the triple alanine, there is not much surface charge alteration. Possibly that's why the effect is not so drastic. So based upon that, what we proposed is if we can introduce this negatively charged patch in the surface of the H17N10 polymerase, it should increase its activity. And that's what we saw. That when we introduced a human-specific glutamic acid at the center of the bat influenza virus polymerase, SST motif, there is twofold increase in the polymerase activity. Not only that, when we have introduced the motif at each of the portions, glutamic acid at each portions of the motif, 79, 82 and 86, you see there is a gradient of increase, right? In the case of the 279, maximum increase, 282, moderate increase, 286, almost no increase. So this shows that the negative, it's not only the negative charge, the proper positioning of the negative charge with respect to the surface of the polymerase is critical for the optimal activity. So based upon this, what we have introduced is the RNA meter. In the case of the H1N1 polymerase, because it has a SES motif, the RNA meter is already at the good side. You introduce a bat specific motif, triple S, it will come to the fair side. You introduce a positively charged lysine, it will go to the poor or very poor side. In the case of the H17 polymerase, it has the wild type SST motif. So that's why the RNA meter is already at the fair side. If you introduce a human specific motif, human virus specific negatively charged residue, it will go to the good side. If you introduce to the positively charged amino acid, it will go to the poor side. You see, a single negative charge at the surface of the polymerase can fine tune the influenza virus replication or viral RNA synthesis, right? But the question that still remains open is how this SES motif is regulating the RNA synthesis at the molecular level. In order to understand that, we performed MD simulation analysis with the bat influenza virus polymerase, okay, which is harboring the wild type SST motif. And here at each position of the motif, we have in silico introduced mutations. Mutations like the positively charged lysine, negatively charged glutamic acid or uncharged alanine residues. And to our surprise, what we saw is lysine, which is functionally the least active one, is having the maximum stability or improvising the maximum stability to the polymerase, right? So the maximum stability of the polymerase, least it is active. We predicted that it's not actually the stability, it's the rigidity of the polymerase which is getting imparted by the lysine residue. So in order to explain this better, we looked into the structure of the PB2 subunit of the polymerase, and this is how it looks like. So you have the 627 domain here and the gap binding domain here, and this is intermediated by this mid-link region or mid-link domain, which is constituted by the mid and the linker region together. Now it is the mid-link region across which the 627 domain and the cap binding domain is undergoing this 62-degree rotation so that it can transition from the transcriptase to replicase mode. Okay. Now if you look further closer, that's when you see that the SST motif is closely situated with two of the glutamic acid residues which is present in the 627 domain. And these two glutamic acid residues remains completely conserved for all of the influenza viruses 1 to 18 and NA 1 to 11. So there is no change in these glutamic acid residues, completely 100% conserved. Okay. So based on that, what we predicted that if you are having a SST motif, as in the case of the bat influenza viruses, there is about 6 angstrom distance between the SST motif and these two glutamic acid residues present in the 627 domain. But when we are having a glutamic acid, negatively charged glutamic acid in the motif, that means reconstituting a EST or ACT motif, that's when the negatively charged side chain of the glutamic acid is coming in close proximity to the negatively charged side chain of the E576. And that's when they start having a repulsion. Actually, this repulsion is good for the structural flexibility of the PB2 subunit of the polymerase, because that can drive the rotation of the 627 domain across this motif. Okay. Now, if we artificially introduce a lysine residue, the side chain of the lysine is big enough to come within 2.4 Armstrong distance from this conserved glutamic acid present in the 627 domain, and hence it's forming a salt bridge. Okay, and hence locking the 627 domain in the transcriptase structure. So based upon that, what we have hypothesized is basically this SES motif imparts higher flexibility to the polymerase, which is essential for smoother transition from the transcriptase to replicase mode, and hence maintain the balance between the transcription and replication. To validate this hypothesis, we again performed the primer extension analysis. And here you see, this is the H1N1 polymerase, this is the mRNA, positive sense complementary RNA, and negative sense viral progeny viral RNA. Okay. Now in case of the wild type H17N10 polymerase, which is having the SST motif, there is not much decrease in the transcript level, but the positive sense complementary RNA is completely gone significantly, and the negative sense vRNA synthesis is also significantly impacted, suggesting that this polymerase is to some extent reluctant for the transition from the transcriptase to replicase. Now, when we have introduced the glutamic acid either at the 279, 282 or 286 position, you see there is an increase in the complementary RNA and the viral RNA. Now, when we perform the ratio of replicon, positive sense replicon by transcript and negative sense replicon by transcript, this is how the ratio should look like, transcription to replication by transcription. Okay. In the case of the SST motif, that is H17N10 polymerase, this ratio is completely skewed up. And if you introduce a EST motif, that means introduce a glutamic acid at the 279 position, again the ratio has been rebalanced. Okay, actually you see there is a slight decrease in the case of the transcription. So it is reducing the transcription, increasing the replication in order to rebalance the transcription replication balance. Okay. So so not only that, when we have performed the primer extension analysis with the wild type H1N1 polymerase, which is having the SES motif, this is the mRNA, cRNA, complementary RNA, and the viral RNA, negative sense genomic RNA. When we have introduced the triple allele mutation, the transcript levels remains the same, replication is significantly impacted. Introduce the positively charged amino acid, replication levels are significantly impacted. Again, supporting the same hypothesis that this SES motif or the negative charge in the SES motif is important for transition from the transcription to replication. So based upon all these data, finally we are proposing our model, which is we are saying that it's a spring switch model, where this mid-link region in between the cap binding 627 domain and the cap binding domain is acting as a molecular spring. At one end of the spring you have the SES or ESS motif or SST motif. Now if you have a SES motif, you have a glutamic acid, so that you have glutamic acid at the two end of the spring. One is E576, which remains conserved, and this remains variable. If you have a glutamic acid at this particular position, your spring becomes highly flexible. So there is an easy transition from the transcriptase to replicase mode, and hence ensures high RNA synthesis. Why? Because once you have more replication, you have more template for transcription. You block the replication, the transcription will also be impacted. Now in case of the H17N10 polymerase, where you have the triple S or SST motif, this is less prone towards the switch because the spring is less flexible, and hence there is a decrease in the RNA synthesis because it's impacting the switch. And if by artificial means we are introducing a lysine, that's when it's making the spring completely locked into the transcriptase complex. No switch, no RNA synthesis, completely abrogating the RNA synthesis. So based upon that, what we are concluding is basically the SES motif is important for maintaining the transcription replication balance. So that's why all of the classical influenza viruses is maintaining at least one glutamic acid in this region. In the case of the bat influenza virus, it doesn't have a negatively charged glutamic acid, so that's why the transcription replication balance is skewed up, and that might be one of the responsible factor for the restriction of these viruses into a particular host species. Now, if there is a phosphomimetic adaptation from serine to glutamic acid, there is a chance that virus to some extent will be able to overcome this restriction. So with that, I would stop by acknowledging the students. I have already mentioned Shoptoshi has completed his PhD, is all set to go out for his postdoc, and then Arotrika is performing experiments right now with this project. Other students are also working. Indrani and Shahan will be presenting poster today. Yes, and also our collaborators, funding agencies, and thank you for your patience.
We'll take just 2 burning questions. We are a little behind time.
Very general question. Very general question. In the bats, does it cause a chronic infection?
In bats, it's more kind of a sublethal chronic infections, mild infections. Yeah, it's kind of attenuated virus. It's well established.
So nice talk, no problem. So very silly question like this. So whether before transitioning from a barb or swine to human, whether the mutation has an effect of this SCS or this SST?
So, in the case of the classical influenza viruses, there is no change in the case of the ACS mode. So far, we have been able to analyze about 30,000 sequences for the classical influenza viruses. We will not be able to see any change. Always there is a ACS mode. Okay, so that's why influenza virus possibly doesn't like to have tolerate any mutations in this particular mode, because it's essential for the balance.
This has been a very interesting talk, and I'm sure there are at least a couple more questions. We'll keep that for the coffee break. Thank you for your patient listening, and thank you very much for that wonderful talk. Thank you for the coffee break.
Just a short announcement for the poster presenters. Their poster list is present on the registration desk, so you can look at your poster number and start putting up posters if you want. I also request the speakers for the next session to kindly give their presentations to Rajesh, and I thank the chairs for the first session. Thank you, everyone.
We go out. Yeah? No, we have. Yeah, I have, your name is ma'am? Okay, I have to check it online. It's better if you have it in a presentation.
That one was the latest one. That is why I wanted to give that, but it is okay.
No, but if that's the latest one, I can download it. That's okay.
See, if you open the mail, can
Sure,
you download?
sure. Just a second.
Otherwise, I'll get this one. I think there was some spelling mistake, so I changed that.
Okay. So I have.
Actually, they told to send in mail, so I
Yeah,
thought
so
that,
it's there on mail only. Just wanted to check if it is not. You have sent it to molecular biology.
Yes. Yeah, this one.
I'll just present it also. Can you just check once? Just download it. This is the one, right?
Yeah.
For some reason it didn't come here. Okay, let me check.
It must be in downloads, I think.
Keep it here. Yeah. Okay. Can you just check if this is fine? Good. Thank you. Dude, we have next session is there are 3 people who have shared the presentation: M. Chawla, Manjula, and someone called MS. Who is a pending person? Wait. Anantika. Guys, you just take the session chair down. Just leave it on the table, that will be easier. Cool.
He said take two. Take both. This one and this
So
one.
I have it online. If that's online, we can just take it from there only. His name is Venkat Raman. अबे यार इसमें बहुत सारे मेल में से yeah this fellow but there's no mail dude for presentation बस इसने अजित ने भेजा है हेमा ने भी नहीं भेजा अच्छा हेमा ने भेजा है
Ja. Anupam.
let's search his name what's his name
Mudruga. Venkat.
Veng
Musturi Venkatraman.
Venkat
हाँ ये
okay let's see yes and something here okay
नहीं ये नहीं है ये नहीं है एयरपोर्ट पिकअप एंड प्रेजेंटेशन स्लाइड्स
this is the guy but he has not sent anything
लास्ट मेल वाज ऑन 21st September
Yes,
so
just
just
send
check this
इसको ऑफ करना पड़ेगा ऐसा अपना दूसरा करेगा
hey खुल नहीं रहा है
just a second RNA meeting ये RNA meeting में अटेंड किया था क्या how do I search now dude
मोलेक्युलर वायरोलॉजी करके कुछ होगा
Peptide vaccine.
hey
This is what I work
hey
on. Okay, let's go back a little. Star immunology. No, dude.
just look at the date
No. Just modify this. Yesterday. This is the one. So now we have to take it to desktop. Session 2. I'll keep the pen drive for some time. I'll come back.
just check if this is only the
Yeah,
one
that's the one. I'm really interested for the other one.
yeah
No,
I collect
Thanks, sir. Just wanted to see what he has over here.
Hey.
Wait. No, it's not visible, that's why. Okay, this is something related to COVID. But T-cell response, nice. just remove it and then take it. Otherwise, I'll be tempted to just copy it.
yeah okay I'll just remove no directly
Yeah. It's copied, right?
yeah just check once session 2 में जाके desktop
ये ना ये
मंजुला
This is-
नहीं
Yeah, this is the guy. 61 MB. I like this fellow who's at 6 MB. She
Oh.
made very old style presentation. I hope she's not here.
चाय पी लो अब बस 5 मिनट है क्या चाय पिए
When will they come back? We'll have to push them also.
she announced the break
Yeah, no, but then once you have to come back also, वहाँ से push करना पड़ेगा. वहाँ पे भी scoring session भी चल रहा है क्या?
नहीं अभी नहीं चल रहा अरे सबने पोस्टर लगाया थोड़ी है
Oh, is it?
नहीं
There's only for coffee. They have
yeah
to. Then we
poster
can push them.
presentation in the evening
Then we
that
can
is
push
1
them.
hour
What?
break
This is the only slide she has. Dude, don't tell me.
no
Additional slide है क्या? Yeah. No. It's gonna be little tough. ये क्या है भाई? ठीक है जो
ये
भी है.
prof है ना
Something
यही वाला
in
है
that method I feel that Shashank is good in terms of making it.
बंद
This
कैसे
connecting
करना
to- Please close it.
Mark हम लोग कुछ खाने गए थे
Yeah.
शुरू हो गया तो
No, it's
भूख लग
okay.
रहा
We can go. अभी सब सारे presentations आ गए ना. वो पहले उसका टेंशन.
रखना है पहले
No.
दे
Keep
दो।
it. Yeah, that's all. I just have this one more person's. What is his name?
least RCA GSTI molecular protein. That actually helps. Later on, when this is going on, then some private party has to check. If you hold that model, so it's institutional, that you can later that will go up to 3 to 4 times. That way, and then you raise samples for asking all the money is already taken up, so it's very difficult to get back. That's what I'm saying exactly. Like they Chinese, these are all they have given because they're saying same year you can't do it. Like the Calcutta was completely Calcutta together. I'm glad that there's a rainstorm. Bengaluru, you cannot really predict. It's very scorchy sun and suddenly it starts raining. So you can't get anyone to carry umbrella. Unless you get way, I think it will be nice for forest. So ICGB, I don't know what's happening. Only Naveen is there and Swami.
Who's the first person? इसमें नाम क्या है किसका पहले? मंजू। उसका नाम पहले है। ठीक है।
Yes.
Yes, ma'am. Sure. I'll just try to go and see that. We have tried to reduce the temperature for the room, but that side it will be really cold. If possible, those who are feeling really cold can move this side, but it will take some time to set it up. So we'll start. I'll just put the presentation first. Okay. We're already presented.
Molecular Immunology and Virology at the Banaras Hindu University on deputation by ACBR. Throughout his illustrious career, he has contributed immensely in the understanding of the molecular pathogenesis of different vector-borne viruses. He has been associated with many prestigious international journals as an editor and editorial board member. He has also published many books in the area of virology to further our understanding of viruses. And sir, we are honored to have you with us today. So Dr. P.N. Rangarajan is currently a professor at the Department of Biochemistry at the Indian Institute of Science, Bangalore. Although his current research is focused on the study of regulation of carbon metabolism in methylotropic yeast, his past research has focused on vaccine development against viral diseases like rabies, hepatitis B, and the Japanese encephalitis virus. He had helped develop a DNA-based vaccine against rabies, which is currently marketed by the Indian Immunologicals Limited and under the brand name DNA Wrap. He's a true asset to the Institute and we are delighted to have him here today. Thank you for being here. I now request the chairs to begin the session.
Thank you. First of all, I'd like to thank the organizers for asking me to join this session. As mentioned, I do all work on IISc syndrome. I was working. Also in correction, the DNA-based vaccine for rabies is not in the market. It went up to clinical toxicity studies, then did not move forward. Small correction. So first speaker is Mamta Chawla Sarkar from IISc Kolkata, and Dr. Sarkar's lab focuses on enteric viruses, particularly rotaviruses, and she has been exploring therapeutic approaches to reduce the disease burden. And studying the role of small RNAs like link RNAs and mRNAs in non-lipid RNA virus infection. And today she is going to talk about the framework of 4 emerging role of non-coding RNAs in regulating virus infections.
First, I would like to thank the organizers for giving us opportunity to be here, not only for presenting but also to meet all the virologists from India, which usually we do not get a chance to do so. Today I will be actually focusing on a topic which we just started a year and a half ago in our lab, and that is emerging role of long non-coding RNAs in regulating viral infections. So we all know that only approximately 2% of our genome is transcribed into proteins and mRNA, and rest of the 70 to 80% genome was considered as non-coding RNA. Non-coding RNAs are of two types: small non-coding RNAs, which are actually studied more extensively. They are some, siRNAs and microRNAs. And long non-coding RNAs are more than 200 nucleotide long non-coding RNAs. They are transcribed by RNA polymerase 2 and 3. They are 5' capped, and they also have, some of them at least have a poly A tail. Previously, these LNC RNAs, especially the long non-coding LNC RNAs, were considered as dark matter or junk in the genome. But in the last decade, it has been found that they are actually very important physiological regulators of cell homeostasis, growth, and differentiation. Most of the studies which have been done are on cancer, but there are studies in viral infection also. So based on their location in the cell, they are actually divided into two types: nuclear LNC RNAs and the cytoplasmic LNC RNAs. The nuclear LNC RNAs regulate gene transcription by either acting as enhancer or a repressor. They also have been implicated in regulating splicing and also form scaffold. The cytoplasmic LNC RNAs do not have a direct role as enhancer or repressor, but they act as miRNA sponges. So if a LNC RNA has a complementary sequence with some miRNA, it will actually quench the miRNA so that miRNA cannot go and emulate the function of its corresponding mRNA. And they also not only interact with the microRNA, they also directly have been shown to interact with some of the proteins. They are also known to inhibit stability and also have been implicated in cell-to-cell communication. So in case of viral infections, many studies including our lab and the studies which we heard in the morning have shown there is rotavirus. And so what we did was we used rotavirus and did a time course study in HT29 cells and did RNA-seq at 12 hours and 24 hours post-infection. And after doing the RNA-seq, there was huge amount of data, as you know, is generated. The in silico bioinformatics analysis were done, and it was divided into many families of proteins or pathways. Some of the pathways which are actually known to be modulated during viral infections, such as inflammation or apoptosis and interferon signaling pathways, we also validated that by doing a specific plate array, which is available in QIAGEN, just to do validation. And we found that there was a 90% agreement between RNA-seq and these PCR array experiments. And then we took few subset of LNC RNAs and tried to identify their function in the rotavirus infection cycle. So what we did is we either knocked down those LNC RNA upregulation, or we overexpressed those LNC RNA transiently, and then did viral assays, titer assays to see their functional significance. So this was a heat map of the RNA-seq, in which we found that almost 1,000 LNC RNAs were differentially either upregulated or downregulated. The cutoff was twofold change, and most of them were found in both 12 hours and 24 hours post-infection, but there were few which were only induced at the late hours of infection. So what we did is we picked up few LNC RNAs to study further. The basis of picking up was either if they were very significantly or very highly modulated, highly significantly upregulated or downregulated, or if they were already known to alter a pathway which is reported in regulating virus infection. So we did KEC pathway analysis, which actually identified pathways like apoptosis, ferroptosis, MAP kinase signaling, mTOR signaling, JAK-STAT pathway, and cell cycle. These are the ones which are already implicated in viral infection in one or the other studies. And there were some new pathways also which we have yet not confirmed, but which will be interesting to study in future. So first siRNA. The first LNC RNA which I am going to show today is SLC7A11 AS1. It is a antisense LNC RNA for the gene SLC7A11. It was induced more than 500-fold at 12 hours and went up to 10,000-fold at 24 hours post-infection. And the real-time PCR also confirmed that in a time-dependent manner, there was a significantly high upregulation of this LNC RNA along with the upregulation of as virus replicated. So what is the role of SLC7A11 which is known? So we did literature search, we found that it is a cis-acting LNC RNA which suppresses its cognate gene XCT and is involved in ROS production. It was already known to have a role in maintaining cisplatin resistance in carcinoma and also in male infertility. XCT is a regulatory light chain constituent of the assisting glutathione antiporter, and it is expressed on the cell, and it helps in uptaking cysteine, which is required for the production of glutathione. And if we have this antisense LNC RNA upregulated, it stops this, it downregulates this XCT system, which resulting in low production of glutathione, and which ultimately inhibits the cytosolic ROS or the lipid ROS induction. So we were really surprised, because we always thought that cytosolic ROS or induction of ROS is an anti-pathogenic function. So how rotavirus modulates, or what is the significance of this LNC RNA? When we did knockdown of this LNC RNA using siRNA, we found that it actually significantly inhibits rotavirus replication, as we see by expression of rotaviral protein or also the viral titers. This which suggested that it has a actually pro-viral role in rotavirus infection cycle. So next we look whether it has any, it regulates ROS or it has any other role in case of rotavirus infection. So we first looked at XCT expression to see whether it is targeting its XCT gene during rotavirus infection or not, and we found that yes, in a time dependent manner we see down regulation of XCT expression in the cells. And if we use the siRNA against the LNC RNA, we see that XCT gene is upregulated back. So this suggests that indeed in rotavirus infection also it targets the XCT pathway. So as XCT pathway is known to be regulating the glutathione synthesis and also lipid peroxidation, we saw that what happens during rotavirus infection, and we found that yes, indeed with the time dependent infection, we see there was a decreasing intracellular glutathione in the cells, and also there was an increase in MDA, which is a marker for lipid peroxidation. Furthermore, we also looked at the release of lipid ROS by using a dye, body P, which is a lipophilic fluorescent dye, and it changes its fluorescence from red to green in presence of ROS. And we can see here that as we go for increasing time points, we see that there is an increase in fluorescence at this dye at 24 hours post infection in SE11 infected cell, and elastin is a positive control which activates lipid ROS. So, but what could be the significance of this lipid peroxidation was still not known, and then we looked at whether it induces ferroptosis or not. So we used MDA marker and checked in presence of the siRNA against the LNC RNA, and we saw there is decrease in lipid peroxidation, and we also saw there is decrease in lipid peroxidation in presence when we overexpress XT. So the significance of ferroptosis was still not known. So what we did is we used a specific ferroptosis inhibitor called ferrostatin, and also lipostatin, which I have not shown here. And we saw that with a time dependent and in the concentration dependent manner, we see there is a decrease in virus yield, specifically the release of virions in the media in presence of ferrostatin. And this ferroptosis has a role only in the late hours of infection, because even if we add the inhibitor at 1 hour post-infection or we add later at 12 hours post-infection, we saw similar level of inhibition of rotavirus release of virions in the media. So in summary, we can say that the siRNA SLC7A11 inhibits the XCT system to actually induce ferroptosis, and ferroptosis is required for rotavirus infection for dissemination. So next, interestingly, we also during in silico study we identified 2 differentially expressed LNC RNAs which targeted the PI3 kinase pathway. And PI3 kinase pathway has been shown to regulate rotavirus infection positively both by our group as well as other multiple studies from other parts of the world. And these 2 LNC RNAs were actually, one was upregulated and one was downregulated. So we wanted to see how they are manipulating PI3 kinase. So we first looked at MEG3. MEG3 is a maternally expressed gene 3 transcript variant 1 which is downregulated almost 50-fold during rotavirus infection. It has a multiple spliced variants and all of them actually act as a LNC RNA. There are several studies reported in cancer that this LNC RNA controls expression of tumor suppressor genes and oncogenes, but we have not seen any in case of virus infection. So real-time PCR also confirmed the downregulation of this LNC RNA during rotavirus infection. So since it was downregulated, we had to overexpress to see its function. So we overexpressed this in pcDNA and transfected in the cell, and we found that overexpression of MEG3 results in downregulation of rotavirus infection. So it actually acts as an antiviral thing and during the rotavirus infection. So to look at its role in PI3 kinase pathway, we infected the MEG3 overexpressing cells with rotavirus strain SA11 and looked at levels of PI3 kinase, phospho-AKT, AKT and VP6. We did not see any change in the basal levels of AKT. However, phosphorylation of AKT was seriously downregulated. And to know how this RNA can regulate a pathway or can inhibit a phosphorylation of phospho-AKT, we did RNA immunoprecipitation experiments in which we used PI3 specific antibody to immunoprecipitate the cells which were overexpressing MEG3 followed by infection with virus. And after immunoprecipitation, we separated the RNA and did real-time PCR for MEG3 specific primers and saw that yes, MEG3 gets precipitated always with PI3 kinase. So when MEG3 is expressed, it binds to PI3K directly and PI3-AKT signaling is downregulated. However, the next LNC RNA which was upregulated was NEAT1. NEAT1 is the most common upregulated LNC RNA in many virus infections. There have been studies in HSV1, HCV, adenovirus, dengue virus and many other viruses. And it is a nuclear perispecial assembly transcript 1. The function is that it is known to translocate the transcripts and the DNA or the protein to the nuclear perispecial bodies during when it is activated. In addition, it has been shown to have both proviral or antiviral function depending on the virus in which it has been studied, and it regulates interferon pathway acting as either an enhancer or repressor, and it has also been shown to directly bind to STAT3 and regulate STAT3 activation and IL-8 induction. So when we looked at the role of NEAT1 to see whether it is proviral or antiviral, we used siRNA against NEAT1 and we saw that it actually has a proviral function, because if we downregulate NEAT1, we actually inhibit rotavirus infection. And knocking down of NEAT1 actually had no impact on MEG3 expression. So it is not that they are both inter-regulated. So how does differential NEAT expression helps virus to replicate? Actually, I have not shown details over here because we are still doing these studies. It does not directly bind to PI3 kinase. However, it actually activates many microRNAs, and some of these microRNAs have a role in regulating PI3 kinase. So, so it actually activates PI3 kinase, and we see that during rotavirus activation, phospho-AKT is indeed activated at 12 hours post-infection. And if we inhibit this PI3-AKT signaling by using any inhibitors, chemical inhibitors, we see there is a inhibition of rotavirus infection. So both upregulation of NEAT1 and downregulation of MEG3 regulates PI3-AKT pathway, which facilitates rotavirus infection. We are still not sure how both of them are involved, because if we inhibit or overexpress one, we see there is an inhibition. So whether they are working in parallelly or whether it is the ratio of these two which work, these studies are still in process. So I think we may be able to tell this in future. So in addition to the in the lab data, we also saw that many of the studies have shown that the LNC RNAs may have may be used as biomarkers or may be present in the patient sera or patient clinical sample. So we also looked at NEAT1 expression and MEG3 transcripts in the rotavirus positive and rotavirus negative sample. These were done in stool sample. The data is preliminary because I only have 30 each at this stage, and we see similar expression, that the NEAT1 is induced and the MEG3 transcripts are actually down regulated in case of rotavirus infection. We could not see the LNC RNA SLC AS11 in the patient samples. So in summary, I would say that LNC RNAs play a significant role at the host virus interface in modulating virus infection. They may also play a role in differential response of the host, what we see when they are infected with the same virus. And this needs to be explored in future. In addition to the host virus interface, these can also be exploited for either as a diagnostic biomarker or can be used for identifying pathways which are regulated by viruses, and we can design specific antiviral therapeutics. So I would like to acknowledge the support of ICMR and DST SERP, and this work was initiated by the postdocs like the ex-students Rakesh Sarkar and Arpita Mukherjee. However, it has been mainly done by Shreya Banerjee, who is my current PhD student. Thank you.
Discussion. Nice. Our question from the first part, the SLC 7A11, the transport.
Yes.
That is transporter of many amino acid, right? I guess cysteine or.
It's mainly cysteine. It is mainly implicated for glutathione synthesis, and glutathione we know is a antioxidant defender of antioxidant pathway, like it defends the cell from ROS. So it is mainly.
Check whether the host, the amino acid host acid, like cysteine, that affects the viral propagation or the pathogens.
We haven't seen that, and in the in vitro cell culture media, it is not very easy to see amino acid alterations very consistently. We have tried for other amino acids also, and we haven't got much success because we do not get consistent data by just depleting media with particular amino acid or adding amino acid helps. But depleting is a very difficult situation because serum and other media constituents are there, so we can actually never deplete it completely. So in vitro it is not that easy to do.
May I ask a question? So in your L3 overexpressing cells,
Oh, okay.
so you saw that there is a decrease in the level of phospho-AKT. There is also a decrease in PI3 kinase protein level. That transcription.
It is not level transcriptional. It is actually activation of PI3 kinase. So phosphorylation is less. It is not as drastic as AKT, but it is post-transcriptional change is there.
So you think it is at the level of phospho-AKT?
Yes. Yes.
Mamta, it's very interesting. I was still stuck with these reciprocal relations with the NEAT1 and the NEAT3. Do you think that they interact with the same sets of microRNA?
So I'm not really right now convinced, because we have just been able to identify 3 microRNAs which interact with this, yes. But we have not been able to see that which one actually is impacting the PI3 kinase pathway. And NEAT1 has multiple functions. We also see interacting with STAT3 also we found.
I was also wondering, with the course of infection, do you think this reciprocal relation changes or reverses? Do you make 3 up and make
No, at least up till 24 hours we did not see. And beyond that, the cells start undergoing apoptosis, so it is very difficult to actually conclusively say that it is happening because of induction of cell death or it is happening because of the virus. But up till 24 hours, it is not that. And surprisingly, what I was initially thinking is that if I regulate NEAT1, maybe the MEG3 will be upregulated, but we did not see that. So it looks like they are independent to each other.
Yeah. So initially the RNA-seq data, so you mentioned that 12 hours time point of post-infection and then 24 hours of time point. And the data looks like exactly stark. And later on this slide, when you show that in 12 hours the virus, after the virus infection was something like 50% and then 24 hours something 80% or something. So how that, because like...
No, actually I understood what you are telling, even I noticed that, but it was too late now. It is not the data is reversed, it is only it is like when they are giving the data, it is like first in the 12 hours we have infected cells in the first few lanes, and then we have the mock infected controls, but at the 24 hours it is opposite. And the first is the mock infected controls and then is virus infected cells. That is why it looks like it's a mirror image.
Because in the 6 hours, like it looked like the uninfected cells kind of thing. In the later stage, 6 hours post-infection.
The 6 hours, it is just beginning to change. Yes.
So I was thinking that probably the 6 hours instead of 12 hour post-infection profile looks like similar, or 24 hour profile is like different because virus already. RNAs play a role in
more than 75% of the RNAs were common in 12 hours and 24 hours. Only difference was in the level of induction or reduction. 75 only there were like few or maybe 20% of the LNC RNAs which were exclusively induced at 24 hours. They were not present at 12 hours. So.
Okay, just a small clarification. Do you know who discovered that NEAT1 is upregulated during virus infection?
I read the name, but I can't remember. Oh, okay.
For the first time, that NEAT1 is upregulated.
Oh, right. I think I saw someone's else paper.
We named it as a big virus.
Yes.
So please quote our paper.
Okay, sure. Thank you for pointing out.
But nobody talks about big.
No, thanks for pointing out. Even I did not know that. I will do that, sure. Thank you. Thank you.
Anyway, thank you very much for a very nice talk. I invite now the next speaker, Dr. Milan Sujit from IISc. Dr. Milan Sujit works on hepatitis C virus, host-pathogen interactions, and is also trying to develop a recombinant vaccine for it. They are also studying the biology of SARS-CoV-2 and identify novel antiviral agents and are trying to develop an mRNA vaccine. The laboratory is also interested in evaluating the significance of endogenous retroviruses, and as they are integrated in the ancestral genome of living organisms, including humans. So today he is going to talk to us about understanding those pathogen conflicts in experiments with SARS-CoV-2.
It is in presenter mode. Yeah, it will be presenter mode.
It's not moving.
Okay, just a second. Can I just check from the content? They are, but not here.
Here it is working.
Okay, just a second.
I can use this. This is fine.
But it's not moving over there, that's what I'm saying.
Ah, okay.
Something freeze. Okay.
Okay, so in the meantime, I can just start. So thank you, Dr. Angarajan, for the kind introduction. You have actually elaborated more than what I do. And thank you, Professor Das and Sasank as well as Kesav for giving me this opportunity to speak here. It's not visible here.
Yeah, no, just a second. Yeah, it's like a little heavy, that's why. Yeah, that's fine.
So as you can guess from the title, we are going to discuss about particularly my experiments with SARS-CoV-2. Before that, I used to work on hepatitis E virus, and that is also an RNA virus. So we had interest in both basic as well as vaccine development side of hepatitis E virus, and we used some of our previous expertise in hepatitis E virus to dissect out the different mechanisms in SARS-CoV-2 as well. So one of the core areas that we work on in the lab is studying a host pathogen interaction and identify possible conflicts in which can be used for developing specific antiviral therapeutics. So I have tried to keep the atmosphere light here. So as you can see, the viruses are non-living outside the host, and they are always looking for the interaction partners which make them living organism. And that is why it is very important to identify the interaction partners of the viruses inside the host cell, and those knowledge will be useful in designing specific antiviral therapeutics. And again, you can imagine from the slide that although the interactions look familiar and they use the friendly gestures to hijack the host machineries, but in both ways, the host as well as the pathogen, they are trying to outsmart each other, and they are looking for opportunities to terminate each other. So in that perennial fight for survival, they are also evolving. And if you have read about the Red Queen hypothesis, here probably you can recall from this cartoon that both virus and the host, they are continuously trying to adapt to the selection pressures imparted by both of them. And in the case of the virus, it is easy because it is a small organism, so you have short lifespan, and you can evolve mutations to retain the interactions with the host, which is beneficial for your survival as well as proliferation. And you also want to establish new interactions, and at the same time you want to lose non-profitable interactions. All these help in your survival inside the existing host as well as in expanding the host range and getting into new species. So that is how the viruses has been working all through these selection process. At the same time, we are highly evolved and sophisticated human beings, so we have a lot of defense mechanisms in terms of physical barrier as well as the different layers of the immune function, and we also have adopted the evolutionary fitness to selectively defend ourselves against more life-threatening pathogens than less life-threatening pathogens. For example, the common cold, we don't have a very good protection mechanism, but for dangerous viruses that used to infect us in the ancient time, we have evolved mechanisms to avoid them and to get resistant to them. So in many of these cases, we have acquired also the beneficial elements from the virus, which are commonly known as endogenous retroviral elements. I think Ajit has already shown a slide on the family of the retroviruses, and many of them are actually integrated into our genome, and they are now part of our genome. And in this graph, you can see that 8% of our genome is actually represented by endogenous retroviral elements. And earlier, there was no clarity of their function because mostly they are in the non-coding region. But now we have a emerging body of evidences which suggest that they are playing important roles in our development as well as in different disease conditions, which I have highlighted here. So we will not go into these details because of lack of time, but I'll straight go into the studies that we have done. So all our study was based on this slide. I'm very much motivated by this slide starting from 2020. All we could do is run away or hide from the virus, and there was no way that we could fight with the virus. In fact, SARS-CoV-2 is a very simple coronavirus, and it resembles, the life cycle resembles all of these RNA viruses. The virus has a RNA which enters the cells with specific receptor mediated entry process, then the polyprotein is processed and you get all the subgenomic as well as genomic RNA, which again get assembled into new progeny viruses which go out of the cells. In all these steps, you have lot of RNA protein interactions, and they guide the viral replication as well as progeny virus assembly process. So our idea was that we should look at something which is very essential for the survival of the virus, and in terms of RNA protein interaction, there is involvement of lot of host proteins in controlling the virus replication process, and particularly the 5' and 3' ends of the virus contains cis-acting regulatory elements which interact with not only the viral proteins but also a lot of host protein and assemble a complex which mediates the RNA protein complex for replication process of the virus. So we wanted to identify the proteins that interact with the 5' and 3' end of the research stopped. Okay, so the idea was to identify the proteins, host proteins that interact with the terminal ends, so that we could establish a RNA protein interaction network, and we could deduce some of the potential conflict regions which could be used for designing antiviral strategy. As you might be aware that there are a lot of papers now which emphasize on the genome level RNA protein interaction, and all those studies were not present at that time, and considering the limitations and resources available with us, we chose only the terminal ends. So we did a RNA protein interaction detection assay. It is a genome level, cellular level study with mass spectrometry based proteome-wide identification of RNA binding proteins, and we identified 48 proteins that interact with the 5' UTR and 14 proteins that interact with the 3' UTR of the viral genome. And from that we constructed the network, and we could find expectedly proteins involved in mRNA metabolism as well as cap-dependent translation process. And as I told you that there were a lot of studies ongoing at the time. So the paper, very interesting paper in Nature which showed the protein-protein interaction network of the SARS-CoV-2, it came out, and we redrawn the network here, and we could see that there was already enrichment of some of the host processes here. And when we merged it with the RNA protein interaction network that we generated, we could see further enrichment of the network, PPI network, and that again suggested that probably there is some physiological importance of the network that we developed. And in the meantime, the genome level RNA protein interaction network also came. So people were already working on the replication, translation, host proteins, all those things. So we selected to study on some other protein which was not directly involved in the viral replication process. Interestingly, one of the proteins that we found is LAMP2A, which we found as an interaction partner of 5' UTR. This LAMP2A is a receptor for chaperone mediated autophagy in the cells, and that plays a very important role in maintaining cellular homeostasis. So we decided to study the crosstalk between SARS-CoV-2 infection and the cellular autophagy machinery, and To do that, we first validated the interaction between 5' UTR and LAMP2A using a fluorescent in situ hybridization technique, and we could see the interaction is there. Then we used siRNA specific to LAMP2, and we could see that the absence of LAMP2 could improve or enhance the viral RNA level both in the intracellular and cell culture media supernatant. And we also used different LAMP2 isoform specific siRNAs, and we validated that it is specific to LAMP2A. And again, we did some overexpression study. These are all routine studies of molecular biology. I'll not go into the detail for the sake of time. And we used a control LAMP1 to show that this is specific to LAMP2. And again, we did some of the biochemical inhibitor study of autophagy. And all these collectively told us that there is a increase in autophagic flux in these infected cells. And then we also tried to monitor whether there is an increase in autophagolysosome in these cells or not, and that we did with co-localization of LAMP2 and LC3 antibodies, and we could not find any increase in the autophagolysosome level in the SARS-CoV-2 infected cells. So we concluded that that although there is an increase in autophagic flux, there is no increase in autophagolysosome formation, and probably LAMP2A is keeping the host RNA away from degradation process, and that way it might be either it is inhibiting the viral replication indirectly, or it is protecting them from the cellular machinery. Now, I also told you about the role of acquired retroviral elements in the protection from disease, and in that context we were doing a parallel study in the lab to identify the crosstalk between SARS-CoV-2 and any of the human endogenous retroviral elements. So these are independent studies that we started in parallel, and thankfully in the summary slide I'll show you some correlation. So again, without going into, so this has also been studied and you can published, and you can find the details here in the publication. So we monitored the RNA level of a panel of endogenous retroviral elements in HUH7 cells infected with SARS-CoV-2, and we found that there is a significant decrease in the level of HERV3 envelope level in these cells. And there was increase in some of the HERV elements, and that we ignored because it is known in other viruses that these envelope protein levels increase because of viral influence through the indirect pathways. So we focused on the HERV envelope level, and here I am showing you the genomic locus of HERV envelopes. So this is present in downstream of ZNF117 locus, and it is possible that the envelope protein is produced as a byproduct of ZNF117 transcriptional activity. So to rule out that, we monitored the transcriptional activity of ZNF117 through real-time PCR using a primer specific to exon 1 of the ZNF117 locus, and there was no change in that. So it's suggested that the downregulation is specific to HERV envelope only. And I'm not showing you all the data. We validated it in multiple cell lines, and we also validated it in some COVID-19 patient PBMC samples, and you can see here clearly that in the infected samples, here we are showing that they are infection positive, and there is a very clear-cut downregulation of HERV envelope level, and there is no change in the level of exon 1 of HERV. We also, because there was a lot of data available in the public domain, transcriptome data from COVID-19 infected lung sample, as well as some cell line data where different temporal studies has been done. We screened for the level of HERV transcript in those data sets, and we could see that there is a decrease in the independent data sets also. So all these collectively suggested that there is probably some importance of this downregulation in the life cycle of, or it is influencing somehow the life cycle of SARS-CoV-2. So then we did the routine molecular biology study, and we overexpressed HER-VAR envelope, and there was a decrease in the level of SARS-CoV-2. And when we further depleted the level of HER-VAR using siRNA, there was an increase in the level of SARS-CoV-2. Now, I'm not showing you all the data, but based on the literature and as well as our understanding, we looked at the possible mechanisms which could control the level of HER-VAR envelope. And we found that the phospho-ERK MAP kinase is the major regulator of HER-VAR envelope expression in normal cells, and it also activates HER-VAR expression in a feedback regulation mechanism. And when you infect with SARS-CoV-2, there is a downregulation of phospho-ERK and increase in phospho-P38. So consequently, when you titer the level of either of them, there is a change inverse correlation between their expression. So we also validated that using a biochemical inhibitor of the phospho-ERK, which is U0126, and we got the similar result. Then we looked at the possible elements which could be driving those MAP kinase activity in the HER-VAR LTR region, and we could find presence of multiple AP1 DNA binding sites. One canonical site was present and two non-canonical site was present, and we cloned the LTR and did luciferase assay to validate whether these AP1 elements are actually playing a role in controlling the expression of HER-VAR envelope. And we could see that if we delete the AP1 DBS containing region completely, then there is complete lack of the envelope LTR activity. Whereas if we just mutate through site-directed mutagenesis the canonical AP1 DNA binding site, there is partial inhibition of the LTR activity. So which suggested that again it's all very well known in the transcriptional field that these DNA binding elements they are redundantly involved in controlling transcription in the absence of a canonical site. So now it suggests that these AP1 elements could be involved in controlling the transcriptional regulation of HER-VAR envelope. Then we did a SARS-CoV-2 infection study, and we could see that the LTR activity was downregulated when you infected with the virus. And again, if we use U0126, we got the similar inhibition effect. And we used a panel of inhibitors for other MAP kinases as well, because we all know that the survival MAP kinase phospho-ERK and the stress kinases P38 and JNK, they inversely control the activity of each other. So yeah, it was important to test whether only phospho-ERK downregulation was responsible for controlling the envelope activity, or crosstalk between ERK and P38 and JNK was modulating indirectly the level of P38, the envelope expression. So to verify that, we did a series of inhibitor studies, and we verified that using luciferase assay as well as by measuring the viral RNA level. And I will not go into the detail due to lack of time, but in summary I can tell you that what we could find that there is a increase in P38 MAP kinase level in upon SARS-CoV-2 infection, which is because of stress. This has already been demonstrated by multiple labs, and that leads to decrease in the activity of phospho-ERK, because they are known to counter each other's activity, and that indirectly leads to, so if I have a summary slide here. So that indirectly leads to inhibition of the envelope activity, either by depleting the AP1 first June AP1 transcription factors from the AP1 with DNA binding site, or by recruiting the P38 MAP kinase activated transcription factor ATF2, which is acting as a repressor here to the AP1 DNA binding site. So in either case, they are inhibiting the level of HER-VAR, and HER-VAR is now unable to feedbackly upregulate its own expression. So that way it is not able to serve its role as an antiviral factor during the late stage of infection. Now one important point that you should note here is the activation of JNK pathway. Although JNK pathway is activated here, but we did not see any crosstalk between them, and in literature also, although there is one paper which shows the activation of JNK pathway, but there is no further studies on that, and that is why we were interested in studying the influence of JNK pathway during SARS-CoV-2 infection, and to do that we did a series of Western blot for different stress pathways as well as the downstream modulator of the stress pathways like the autophagy and the caspase pathway, and as you can see here, there is some difference in the activation of different stress mediators if you monitor here. So in early time points you see some of these ER stress markers activated, but and P38 is very early activated, but if you look at JNK, it is activated at a late time point. So the ongoing effort is to further investigate the crosstalk between SARS-CoV-2 and JNK signaling pathway, and we have some leads that this is playing an independent role outside the autophagy and caspase activation, and we are working on that. So the overall take-home message is that, as I told you in the beginning, host-virus interaction studies are very important for finding out crucial information to decode the mechanism of viral life cycle and pathogenesis, and we can identify possible conflicts and then design novel antiviral strategies. And with a very small example, we are again supporting the emerging body of literature that retroviral elements integrated in our genome is likely protecting us against a lot of pathogens, including SARS-CoV-2. Thank you.
Can I ask a question as a layman?
Yeah, absolutely.
It looks as though on this in the field of virology, you do experiments when you're always interested in at the genome level, the protein, deal always with proteins. Do you consider the structural aspect, the fluid mechanical aspect, and also the, of course, because the way the virus interacts with the cell, it depends on how it gets into the cell and interacts with the nucleus. And that, of course, do you consider the conservation of species, conservation of mass, conservation of energy, production of energy, energy balance, all those things as we do in chemical kinetics, for example, in fluid mechanics, the combustion, all those areas. Do you consider those things? And the other question is, do you normally do any experiment with the individual cells and observe it and do the energy balance, species balance, or chemical kinetic balance, or all those things?
Very valid and interesting points, and obviously I would also be interested in doing these studies, but unfortunately I personally don't have the expertise either in the chemical or single molecule studies. We also don't have the resources for the single cell based studies, but there is a lot of studies ongoing in that area, and a lot of literature is available. So those are very interesting areas to explore.
But particularly the atomic force, all these microscopes
Yeah.
available. Yeah.
People are doing that.
Thank you. So my question pertains to pathogens. You have shown very evidently the link between HIV expression and an apparently antagonistic relationship with SARS-CoV-2 replication. But if you actually examine the correlates of pathogenesis, things like lymphopenia, things like inflammation, have you been able to draw a link between what you have demonstrated here and how this interaction may govern those sequelae?
Yeah, but actually the study that we did that was done in the last 1.5 years, I would say. So the future plan is in those directions. It's just the beginning.
Thank you, sir, for the presentation. I wanted to ask once that restoration of heartbeat protein imposed brings down SARS-CoV-2 replication. So I wanted to ask that does it really interact with any SARS-CoV-2 non-structural protein, or what is the basic mechanism of bringing down SARS-CoV-2 replication when it is being restored?
Can you repeat the question, please? You can just speak.
Yeah, I wanted to ask that how the restoration of heartbeat protein, it brings down SARS-CoV-2 replication. So actually, what's the basic mechanism? Does it interact with any SARS-CoV-2 non-structural protein, or?
No, what we know from other studies, from published literature, is that when there is an increase in P38 MAP kinase activity, there is an increase in overall stress in the cells, and that is helping in virus replication in some way. We don't know the actual target. Some of the targets are known from the phosphoproteomic study, but the actual pinpointed mechanism is not known. And what we showed is that HER-VAR is activating the opposite of the P38 MAP kinase, which is the ERK MAP kinase. And that way it is down-regulating the P38 MAP kinase. So the overall stress level in the cells is going down, and whatever is the target of the P38 MAP kinase, which is helping in virus replication, is now not functional. So that could be the mechanism. The actual mechanism that remains to be tested.
Thank you, Doctor.
Thank you.
Thanks to my co-chair, Professor Bengaluru. Now, next two parts of the session are focused to the vector-borne viruses. And first one is going to be on Japanese virus by Professor Bengaluru from IISc. And she has done wonderful work and in the area of molecular resistance to understand how these very viruses, especially Japanese encephalitis virus and dengue, interact with the different parts of the immune system, so on and so forth. And now today's talk is going to be focused on linking virus pathogenesis to host membrane trafficking, autophagy, ER-associated inflammation. Thank you.
Thank you. Thank you.
Yeah, just save the slides.
So thank you so much. It's a great pleasure to be here. And I also have a small story to share. My career trajectory also was shaped by the first molecular virology meeting, where I presented a poster, and I was lucky that THSTI was looking for virologists at that time, and Dr. Vrati came to my poster. So that's how my journey with viruses actually took off. Yeah. So also I thank Professor Das and Shashank and Kesav for the excellent organization. So the focus of my group has been the Japanese encephalitis virus, which everyone in this audience knows. It's a neurotropic virus. Causes encephalitis, endemic in several regions of Southeast Asia, with a huge mortality rate, especially in the pediatric population. And it is maintained in an enzoonotic cycle between the vertebrate hosts, that are pigs and birds, and transmitted by Culex mosquitoes. Now, the disease is prevalent in several states of India, but the good news is that over the years, the incidence of JE-caused acute encephalitis cases has gone down in states like UP, thanks to the vaccination program. However, we still hear of outbreaks. This is from a recent report in Assam, where about 11 people have died.
And also because of the expansion of the geographical range of the virus, we now see that cases last year, several human cases were reported in Australia. So the pathogen definitely has potential for global spread. So my group has been engaged in understanding the cell biology of virus-host interactions, and for the longest time you've heard me talk about ER stress, autophagy and innate immunity. But for today's talk, I'm going to focus on our work on the host factors which are involved in the biogenesis of the virus replication complex. Of course, Dr. Vijaya and her students have done very landmark studies on actually the first description of the JE virus replication complex, and the biochemical purification was done in their lab. So the Japanese encephalitis virus basically encodes for a plus strand RNA, and this is a capped structure which gets translated into a polyprotein, and this polyprotein has 3 structural and 7 non-structural proteins. Now the structural proteins of course are responsible for formation of the outer envelope and the capsid, and the non-structural proteins play different parts in the virus replication process. The NS5 is the polymerase, NS3 is the protease, and NS1 is a multifunctional protein which is both necessary for the formation of the virus replication complex as well as another secretory protein, and is also used as a biomarker for flavivirus infections. Now the virus life cycle starts with pH-dependent release of the virus particle, uncoating of the nucleocapsid, and virus replication takes place in close association with ER-derived membranes. And the egress of the immature virus particles takes place through the secretory network, and ultimately these are released. So this is the the modification of the ER membranes is one of the very critical events for the formation of the virus replication complex. Now, classical electron micrograph studies have shown that virus replication basically takes place on very well-defined structures which are called vesicle packets, which you can see in this EM as very well-defined spherical structures. There are also a lot of convoluted membranes which are present in close proximity, and these are basically believed to be sites of polyprotein production and the processing of the polyproteins. And if you look at the topology of the virus polyprotein on the ER membrane, you'll see that the NS3 protease and the polymerase are actually facing the cytosol, whereas the viral envelope protein and NS1 are in the ER lumen. The viral replication complex results in the formation of this negatively charged, negatively invaginated membrane vesicle, which buds into the ER, and this topology is achieved by the action of the NS2A, 4A and the 4B proteins. And this you can now look at the virus replication complex. The NS1 protein is in the ER lumen, and this particular region is where the NS3 and the NS5 will replicate the viral RNA through the double-stranded RNA intermediate. And this is also connected by a pore to the cytosol, and this is the pore through which exchange of materials, nucleotides takes place, and also for release of the viral RNA. Now, other recent studies have also shown that 3D electron micrographic reconstructions have shown that the viral replication complexes are very closely associated to the site of the viral nucleocapsid encapsulation. So you can visualize this as the virus replication complex, the pore through which the viral RNA is released, and the actual nucleocapsid assembly site. So our studies basically our studies on autophagy, which started long back in about 10 years ago, we were interested to look at how the cellular autophagy pathway basically intersects with virus replication. And in this study, apart from looking at the role of autophagy, we made 2 very interesting observations. The first observation was that, so autophagy, as you all know, is a cellular homeostatic process which ultimately culminates in the fusion of the autophagosome with the lysosome and gets upregulated during pathogen infection. and under cellular stress conditions. And if you look at the hallmark for studying autophagy is the protein called LC3. Now LC3 is basically a cytosolic protein, which is the LC3-1 form, but in the autophagosome, this protein undergoes a lipid modification. It associates with phosphatidylethanolamin, and LC3-2 is what is incorporated into the autophagosomal membrane. And on the Western blot, LC3-2 can be seen as the lower band and LC3-1 is the upper band. Autophagy is also studied through expression of GFP-LC3. You can see this is a cytosolic protein, and under starvation condition, it gets associated with the autophagosomes. Now, what we studied, when we were looking at immunofluorescence studies for the virus replication complex, what we observed was the viral NS1 structure showed very good co-localization with LC3. And now this would appear to suggest that virus replication is happening on autophagosomes, but that was not the case. Because if we look at a genetically deficient, autophagy deficient cell line, that is the ATG5 knockout cell line, you could again see the LC3 structures will beautifully decorate the virus replication complexes. And autophagy deficiency in the case of GAV does not affect replication. In fact, we have more virus replication when we knock down autophagy. We did several other experiments to show conclusively that these structures are not autophagosomes, and these are not marked with GFP-LC3, these are not acidic in nature. So suggesting that the virus replication complexes contain the non-lipidated and the non-autophagosomal form of LC3. The other interesting observation was that these structures also contained an ERAD protein. ERAD is the ER-associated degradation pathway, and another ERAD protein, EDM1, also showed very good co-localization with virus replication complexes in different cell types. And through subcellular fractionation, sucrose density gradient, you can see that LC3-1, which is the non-autophagosomal membranes, associate with the virus replication complexes, whereas the autophagosomes, which are lighter and can be seen in this fraction 1, the LC3-2 form, and along with LAMP1, that is the autophagosomal autolysosomal fraction. Now, moving on, we were then looking at other localization of other viral proteins, and then we were, very excellent antibodies for the capsid protein were made available. And when we looked at the immunofluorescence studies of the viral capsid protein in virus infected cells, we could see good overlap with the replication complexes. But the confocal microscopy is not, will not give you that sufficient resolution. And when we went to super resolution SIM imaging, what was very interesting was that we could see that the capsid structures were different or distinct from the NS1 positive structures. And they were very closely juxtaposed, but they were distinct structures. And the capsid was also localizing very robustly with lipid droplets. So this is Bode-P staining, and the capsid formed these beautiful structures on lipid droplets. We went on to show that JAV capsid again interacts with the non-lipidated LC3, that is the LC3-1 form. And this is both in wild type MEFs. We could immunoprecipitate capsid to pull down the non-lipidated, the non-autophagosomal LC3-1 form of the protein. And even in the autophagy deficient cells, we were able to immunoprecipitate, and through a reverse IP again, we showed that the non-autophagosomal LC3 associates with the capsid structures as well. And this localization was also present on lipid droplets, and this was basically our model, where we kind of identified that the virus replication complex, which is marked by NS1, contains EDM1 and LC3, and the lipid droplet, which is decorated with the capsid protein, is closely juxtaposed to the replication complex, but it's actually a distinct structure. And so this is all data that was published, and now I'll come to the main story, which is our recent work on the role of the ER-associated degradation pathway. Now, all of you are aware that protein quality control is extremely essential and takes place in the ER. So the correctly folded proteins are trafficked out of the ER, whereas the terminally misfolded proteins undergo your glycan trimmings, and these get threaded into a pathway which is known as the ER associated degradation pathway. So this pathway basically involves recognition of the terminally misfolded proteins. These terminally misfolded proteins undergo the glycan trimmings through the ER mannosidases and EDM1 proteins. These are recognized by lectins like OS9, which diverts them to the retrotranslocan complex. Now this retrotranslocan complex is the most critical part of ERAD, which contains the cell 1 adapter protein and the HRD1, which is the ubiquitin E3 ligase. And with the aid of other proteins, the misfolded proteins are threaded out and are ultimately degraded through the proteasomal pathway. And the VCP protein, or the valucine containing protein, is another very important ATPase which kind of extracts the proteins from these membranes and directs them towards the degradation. Now, just about 2 years ago, we were also interested in looking at the role of VCP in the life cycle, because this came up as a major host factor in an siRNA screening study that we performed. And one of the students from the lab very beautifully showed an essential role of VCP in the formation of the virus replication complex, where we were able to IP VCP along with NS5 and with NS3. So we were able to add another protein to our model of the replication complex along with NS3 and NS5, the valucine containing protein. So since we had some preliminary information regarding the role of some of the ERAD proteins, we again performed extensive immunofluorescence experiments to see if the ERAD effectors co-localize with the components of the virus replication complex. So here we looked at OS9, which is a lectin, which binds to the misfolded proteins, the cell 1, which is an adapter protein, HRD1, which is the E3 ubiquitin complex, herpioid1, which is another transmembrane protein which plays a role in ERAD, and DERLIN1. And what we saw was that the viral double-stranded intermediate, that is the double-stranded RNA, showed very excellent co-localization with OS9, cell 1 and herpioid1, whereas there was no co-localization seen with HRD or DERLIN1. So suggesting that the components OS9, cell 1 are sitting with the virus replication complex, whereas the E3 ubiquitin ligase HRD1 and DERLIN1 are not present in those structures. Similarly, we saw that the viral NS5 protein, which is the RNA-dependent RNA polymerase, also beautifully co-localized with cell 1. Surprisingly, we didn't see too much of co-localization with NS3, and this could be basically because the major portion of NS3 is diverted into what are known as convoluted membranes, and probably the small fraction of NS3 that should be present with NS5 was not detected in our study. The viral NS1, again a very interesting protein, was co-localized with all of the ERAD effectors. It was co-localized with OS9, cell 1, herpioid1, but in addition we also saw its co-localization with HRD1 and DERLIN. And interestingly, again, capsid did not show any overlap with these structures, suggesting again that it was a segregated structure. So our immunofluorescence studies were basically able to demarcate 4 structures in virus replication. The first are, of course, the vesicle packets, which contain NS5 and the double-stranded RNA, and these were marked with OS9 and cell 1. The lipid droplet, which is in close proximity and contains the capsid proteins, the convoluted membranes enriched for NS3. And interestingly, we see another structure here where NS1 sits with the entire E3 ubiquitin complex. Now, what was the role of these ERAD effectors? During infection, the protein levels of these ERAD effectors does not appear to change, alter much. Then we performed an siRNA mediated depletion studies. We knocked down all of these genes and checked their effect on virus entry as well as replication. In terms of entry, HRD1, which is the E3 ubiquitin ligase, was the only protein which showed a significant reduction in virus entry. And this effect could be probably through an, alteration of an essential entry or a host receptor, since this is a very crucial E3 ligase inside the cell. In terms of virus replication, that is when we look at RNA levels at 24 hours post-infection and virus titers, we saw that cell1L, HERPUD, and HRD, all of these caused a significant reduction in virus replication and titers. And if you look at this Western blot, we also see that the viral translation was also hit majorly by the cell 1 depletion of the cell 1 protein, suggesting that the cell 1 was one of the most critical factors for the replication, for the biogenesis of the JEV replication complex and for its virus replication. Now, the entire E3 ubiquitin complex basically exists, the stability of the complex is highly dependent on the formation of or the interaction between these proteins. So normally what happens is that in control cells, you need to have the interaction between the HRD1 and the cell 1 for stability. And the HRD1 is completely essential to stabilize cell 1L. So when we did the knockdown studies, and you can just probably just focus on these, the red boxes, we see that when we deplete cell 1, we do not have any alteration in the levels of HRD1. And inversely, when we deplete HRD1, the cell 1 levels are completely lost, suggesting that HRD1, the E3 ubiquitin ligase, is very critical for cell 1 stability. However, in the virus infected cells, the condition was absolutely reversed. What we saw was that cell 1 was completely stabilized in virus infected cells, whereas HRD1 became unstable. So this is again clearly suggesting that a fraction of the HRD1 is completely recruited away from the E3 ubiquitin ligase complex and is sequestered into the virus replication complex. We also checked the half-life of these ERAD effectors, and through a cyclohexamide chase assay, we saw that in virus infected cells, the cell 1 protein, which was normally inherently stable in uninfected cells, became highly stabilized, whereas the HRD1 protein, which is here, which is quite stable in uninfected cells, was completely destabilized in virus infected cells. We've also checked for the stability or the half-life of these viral proteins to basically see how their levels are maintained in the cell. And NS1 being an ER luminal protein, which also gets glycosylated, we see that NS1 is stabilized under conditions of MG132 treatment. So here we've done cyclohexamide chase assays and treated with MG132, which is a proteasomal inhibitor, and eristatin, which inhibits the translocation of proteins from the ER, and with bafilomycin, which is a lysosomal inhibitor. So of all the proteins, NS1 appears to be the one which is maximally stabilized through or degraded through the proteasomal pathways, whereas all the other viral proteins seem to have a very strong dependence on the lysosomal system for degradation. So this is my last slide. So I think I just want to summarize that our studies have shown that the cell 1, the ER adapter cell 1, the lectin OS9 and HERPUD1 are critical for replication complex biogenesis, and this is negative for the E3 ubiquitin ligase HRD1 and the DERLIN1 protein, and a pool of cell 1 is disengaged and gets stabilized in an HRD1 independent manner upon infection. And we believe that this potentially regulates the sequestration of the viral non-structural proteins to form the virus replication complex independently of the HRD1 E3 ubiquitin ligase complex. So, of course, I like to acknowledge Professor Vrati because the first few studies were initiated along with him. So lab alumni Manish, Renu and Sapna initiated the study, but all this was part of the PhD project of a student, Riya Sarkar, who's moved on, and Simran, who is helping us see this paper through. Yeah, thank you.
So your cyclohexamide experiment, that would essentially knock off viral transcription also.
So it will. So we allow viral replication to proceed till about 6 hours by which time the proteins have accumulated, and then we allow, then we add cyclohexamide. Yeah.
Are there any more questions? Yeah.
You showed the degradation of the misfolded protein. What percentage of protein will be misfolded? Is it a minuscule amount?
In a normal cell?
Yeah, in the infection process. Why I am asking is, after expending so much energy for making the molecule, targeting them all for degradation, rather than trying to go through some chaperones, because there will be always in short proteins, etc., induce consequent viral infection. So they don't help to refold them to be more economic. And
No, so the viral proteins are not really, so they are always made in excess. The viral proteins are always made far more in excess than what are required, because they are all translated as a polyprotein. But we are just checking the half-lives to see how the protein would get degraded, but in terms of the percentage, I don't think a significant portion gets misfolded.
maybe very small amount.
Yes.
Otherwise, after
Yeah.
expending all this energy to make the proteins.
And but physiological ERAT appears to be compromised, but it's still functional.
That
So
would be refolding also, isn't it? That we chaperone.
absolutely, absolutely, yeah.
Because actually the model which you showed, I thought you were going to say they are going to be chaperones and coming out of refolded rather than targeting for degradation.
So definitely the replication is proceeding. So we are having a significant portion of the proteins folding and playing their specific roles. Yeah, but the proteins are far more in excess. Yeah. Indraneel had one question. Yeah.
Interesting one, so I have a question from the first part of the talk. So you showed that the LC3 co-localizes with the division complex NS5, I think, right? So
No, NS1 and capsid, yeah.
NS1 and capsid. So how is LC3 recruited there?
Yeah, that's the big question, we don't know.
What happens if you deplete?
If we knock down LC3, we've done that. Virus replication is completely blocked. So LC3 is very essential for virus replication. So you would also.
What are the key partners, if lab or Lando?
Yeah, we are trying to get into those. I don't have much to share at this point. Yeah.
Thank you.
Thank you, Manjula.
Yeah.
And if there will be more questions, you can discuss with them. Thank you. Now I would like to invite Dr. Masturi Venkatramana from the University of Hyderabad. Her interest is on dengue and chikungunya virus, and her laboratory has done excellent work to understand the molecular pathogenesis of these viruses. And her group carries studies to characterize dengue and chikungunya strains circulating in South India, to analyze the role of virus encoded structural and non-structural proteins in disease manifestation. Dr. Venkatramana.
See the slides are moving. This one please too. Just check if the slides are moving.
Okay. Slides here, huh? Here, huh?
Yeah. No, we can try with both. It should also help. This is the one. Just a second. Try it out. Yeah. Okay. And you can use a pointer also.
Thank you, sir, for introduction. I thank organizers, Professor Soumitra Das, Dr. Shashank and Keshav. Since I am from Dr. Soumitra Das lab as postdoc, any credentials of my research will be shared to sir also. My presentation will be on the dual subcellular localization of dengue virus protease, and cleavage of key cellular proteins. This is the one of the aspect being carried in our laboratory. Regarding the host virus interactions. Professor Milan Surjit showed the very good cartoon. My presentation will be a leaf out of that cartoon. Just before going to the protease, I just want to bring to your notice, these are the red among the 4 serotypes of dengue virus. Red one are indicating the which are circulating in India, and this virus is very much dynamic in changing its serotypes or the dominating serotypes over the time period, and these two showed the couple of years back very much mixed infections with all 4 serotypes, and recently what we observed is this is the one of the only serotype dominated very much. So then regarding the protease, this protease is conserved protein of among all 4 serotypes. It is works efficiently with in presence of another protein called NS2B, which is 14 to 15 kilodalton protein, and it attaches present at the N-terminal end of the NS3, and this is the structure of the NS3, which is having the helicase and the protease with the linker here, and this forms the catalytic triad with 3 amino acids, and NS3 is itself is 67 kilodaltons. It possesses the protease and the helicase domains. NS3 interacts with NS2B to form the NS2B NS3 active protease, and NS2B acts as substrates and so on. Here the question is that there is a possibility of this protease existing in multiple versions, which is mainly NS3 and NS2B NS3, and there is confusion whether really these two forms are existing. If existing, where they are localizing in the cell. If they are localizing in the cell, whether they have any substrates. If they have the substrates, whether it account for the disease pathogenesis like thrombocytopenia. So in this direction, you can see dengue virus 2 protease NS2B NS3 forms a stable complex, and targeting the intramolecular protease gives the another clue that it may be existing in 2 forms. And importantly, here you can see internal cleavage sites here between the NS2B NS3 and within the NS2B NS3 and 2-3 cleavage sites are there, which gives the suspicious that it exists in the multiple forms. So what we try to see is if these 2 forms are there, to analyze their localizations in this study in their cell, whether these proteins is having any localization signals, for example mitochondria or the nucleus. See, this is complete NS2B NS3 from here to here. This indicates the NS2B region. This is the NLS localization signal, and this is the mitochondrial localization signal. Similarly, we found the multiple NLS sequences across the sequence. This is only NS3 where the MTS is present and NLS sequences also present, which are represented in green. So keeping all these things in mind, we have generated some clones like here NS2B NS3 with linker, then NS2B NS3 with some mutation, and only NS2B NS3, only NS3, and so on, and carried some of the experiments. Like we asked the question that since the NS3 possesses the mitochondrial transport signal, whether NS3 localizes to the mitochondria. See here, and compared to the vector, it clearly shows the NS3 localizes to the mitochondria in presence of the which localizing along with the mito-RFP. So along with the and another construct which is naturally existing NS3 also we generated, that is also showing the localizing into the mitochondria. So since it is localized to the mitochondria, we asked the question that any substrates of this NS3 is there in mitochondria. So in this direction, some of the proteins we selected, like here shown here, on the basis of couple of characters. One of them is that whether these, if they are proteins, these are all proteins of mitochondria. if they have the mitochondria protease cleavage site or not. If the cleavage site is there, that proteins were picked and analyzed by Western blotting in presence of the NS3. So one protein called GRIP11, which is the co-chaperone of the mitochondrial HSP70, GRIPL2, another co-chaperone of the mitochondrial HSP70. So that showed some kind of less compared to the vector, but all other proteins, even though they have the cleavage sites, they did not show any down regulation. So then same in the presence of NS3 in the infected cells, also we saw from the transfected conditions to the infected conditions, the observation was clear, and it was confirmed that the GRIPL1 may be the one of the substrate of the protease. Both components were purified and subject to the in vitro cleavage assay. As the increasing concentrations of protease, this GRIPL1 gone down. So the next step, in the GRIPL1, the cleavage site was identified, and then it was subjected to the mutation here. So as a result, in the mutated version, that was not cleaved. So it was confirmed that the GRIPL1 is the substrate of protease. So we proposed in this way, compared to the normal situation, in the dengue virus infected conditions, the protease is cleaving one of the co-chaperones of the mitochondrial HSP70, that is GRIPL1. GRIPL2 is another co-chaperone, and as a result, any of these functions or all these functions may be affecting, since mitochondria is very crucial role in the cell and hence accounting for the cellular homeostasis. Then what about the another version called the NS2B NS3? Here also similar experiments we carried, vector and the NS2B NS3 mutant and the full naturally existing one. Compared to the vector, the green expression, here we can see the merged conditions also. Compared to the vector, all other versions, it is showing that it is NS2B NS3 is localizing to the nucleus, where NS3 localized to the mitochondria. In vector, this is the GFP and the blue, it is host strain, which is not merging. All other conditions is merging, supporting the localization of NS2B into the nucleus. How about the reverse localization of both? Since NS3 was shown to be localized to the mitochondria and NS2B NS3 was in nucleus, how about NS3 goes to the nucleus or NS2B NS3 goes to the mitochondria? So we carried one experiment like this, seeded the cells and individually incubated some time and then mixed with the lipofectamine and transfected along with the mito-RFE to show the mitochondria. After that, the cells were stained with host stain to see the nucleus also. So in these observations, here we can see this is the vector and the merged versions, and here this panel represents the whole cell, means mitochondria. So in vector, the mito-RFP is not merged with the GFP, and also here it is NS2B NS3 is not merged with the mito-RFP, which suggests that it is not NS2B NS3 is not localizing into the mitochondria and NS3 is localizing into the mitochondria. This represents the nucleus, where the GFP is not merged with the blue, not expressed, and the nuclear region, means blue region, at that region the high GFP level is there, that indicates the in the nucleus also, in the nucleus only both versions, both NS2B and NS3 are localizing, means it is more or less clear at this stage that NS2B is localized to the nucleus and NS3 localizes to both nucleus and mitochondria. Further, to support these studies, we have carried some fractionation studies where this where the cells were scrapped, isolated, and made this thing. And this is the left side shows the whole cell lysate, and then pellets started and take the nuclear extract and the cytoplasmic extract, which represents the mitochondria. So the all these fractions were isolated, means analyzed in the Western blotting, and here you can see in the nuclear fraction only, the NS2B, NS3 is there, and these are the in the whole cell lysate also there, and in the broken products of the NS3 internal cleavage site because of that. And in case of NS3, this is present in all the fractions. That means NS3 is one more time it is confirmed that NS3 is localizing into the nucleus and mitochondria. NS3 is localizing only to the nucleus. The other these things purity of the fractionations. So since NS2B, NS3 localization also confirmed, is there any substrates for this NS2B, NS3? So here in order to identify any substrates of NS2B, NS3, we just purified the protein, and then did some pull down assay, and some of the suspected proteins were eluted and sent for the mass spec. The mass spec results shows that one protein called EDRF1, erythroid differentiation regulatory factor 1, is likely to interact in protein, and the same elutes we subjected to the corresponding antibodies, EDRF1 antibodies. It's confirmed that elutes containing that band is the EDRF1. So EDRF1 is very big protein, and then we looked for the protease cleavage sites. The red ones indicates the protease cleavage sites. There are 5 protease cleavage sites. Which one among them couple of them are interacting with the catalytic side of the protease? That was the question. So both proteins we superimposed, and this showed that the amino acids numbers like around 1000 is coming in contact with the catalytic side of the protease. So hence what we did, we cloned, it is 130 kilodalton protein is not possible. C-terminal end where that amino acids are coming close to the catalytic side, that part we have cloned, and then did some in vitro and all these experiments. Here I show one experiment, that is what we have done, some co-transfection analysis. Means we have co-transfected with both EDRFN and protease by following the method, and here what happens, this analysis shows that where the co-transfected one is there, EDRF1 is disappeared. At that place protease is expressing. So this confirms the EDRFN is being cleaved, and EDRF1 is one of the novel substrate of the protease. So then we asked another question, what is the role of this cleavage, what is the meaning of this cleavage? The literature such indicates that EDRF1 is one very important protein. This protein is required for the expression and activity of the GATA1, and transcription factor GATA1 in megakaryocytes is very important, and this importantly, this GATA1 regulates the expression of another proteins called the spectrins, and spectrins are required for the platelet and proplatelet formation. That was the link. So this is the hypothesis we made, This is the normal situation. EDRF1, GATA1 expression and activation, and spectrins, and the normal number of platelets. And if the dengue virus infection is there, ETRF1 is being cleaved, where the non-expression of the GATA1, and hence no spectrins and less number of platelets. To test this hypothesis, we checked these downstream proteins along the EDRF1 in the transfected conditions. We saw that EDRF1 is gone down, and the GATA1 and spectrin also, they showed a little down regulation in the transfected conditions, and which was shown efficiently in the, which was shown clearly in the presence of mutant and the normal natural constructs. Further, we have checked this kind of effect in the infected conditions, where it is more clear that all these proteins were affected. Then, so what is this situation is existing in the case of infected dengue virus infected samples also. So we collected some of the clinical samples of DF, DHF and DSS, and we saw that the proteins called the GRIPL1 and their platelet numbers were correlating, and here also we can see that in the different samples the EDRF1 is going down, and where EDRF1 is affecting, GRIPL1 is also affecting. This confirms that these proteins are having some role on the thrombocytopenia, and protease is doing something of that, and and then since the megakaryocyte derived cells are the K562 cells, we try to check whether the number of these cells goes down over the days of infection. Yes, it is done. At the 7th day, the number of cells is completely gone down, and we confirmed on 5th day the presence of the virus also, and and then to confirm these studies, we are presently planning in the animal models. These are the team I work with, and the work mostly carried by the Lekha Gandhi, and support of the Deepthi and Deepika and other students also. And Deepthi's poster is here, which is completely differently. Please have a look at it. Thank you very much.
So Ramana,
Sir.
this GRPL1 and the EDRF1 cleavage,
Sir.
can you correlate with the pathogenicity? I think I asked you before also, type 2 versus type 4, do you see that one of them cleaves, other one doesn't? Or the one that goes to DSS and the patient who doesn't go to DSS but goes back to plan A, do you see any differences between them?
Sir, all this work was done with the DEN 1, sir. DEN 1. Oh, yes, sir. DEN 2, we don't have the. Thank you, sir. We'll look into that.
Are there any more questions, please? If no, then before I declare the session is over, I would say that this molecular virology forum has provided a chance to our new investigators as well as senior investigators to share their findings. And this is the brainchild of Professor Soumitra Das along with his friends. Few of them are here. Professor Vati is there, Professor Rangarajan is around, and few of them are not here. Professor Shahid Jamil and Devashish Mitra, and we should give a big hand for their effort. Now this session is over. Thank you.
So thank you chairs, thank you speakers. We will now be breaking for lunch at the main guest house where we had breakfast. Please wear your ID cards. Thank you. No.
just pull this out. Yeah, these are no session 3 we have a printout. You can ask her. Session 3 we have a printout, right?
Priya.
Oh, acha, okay. Sorry. So this is the one, right?
I have only one file.
Okay, this is for the session 3.
Yes, I'm the third speaker.
No, I don't have it, sir. If it is in the mail, I'll retrieve from there. Okay. I just checked. I think Hema, right? Okay, sorry. Yeah, Hemchandra. Yeah, it's here. Yeah, sure. I'll do it. I'll load it, and before the presentation also, I'll just go through.
Can you just open and check here in this computer? Everything is okay. Should be okay.
It's fine. Okay, good. Yeah, just connect it. Thank you, sir. Thank you. Just a second, I'll just put the live screen off.
Manager expecting himself came
Okay. So, do you have the list ready already? For the speakers? Yeah, which one, sir?
Press 7.
Okay, let me see the last updated one. This one?
Yeah.
Session 3. Yeah. Do you have the next session speakers list?
Yeah.
Sure. So, Amit Singh I have not got. Apart from that, I think I have for everyone. I'll just check. मेरे पास उनका नहीं है. I'll just check if it is there.
One change. I want to do it.
क्लोज हो जाएगा. लो हो जाएगा. ठीक है.
So, Control S or Function S?
No, Command.
Command, okay.
One second.
Eject.
So edit has happened over here. I think this is fine.
Right,
Thank you, sir. Chandra. टू मिनट्स बस अमित सिंह सर का नहीं है. सर है क्या यहाँ पे? ओके ठीक है. रेस्ट ऑल थिंग्स आर डन. जस्ट सी लाइव स्ट्रीम में क्या जा रहा है. ओके कूल. एक बार लाइव स्ट्रीम में Screenshot. Yeah. That's all.
I think we are live. That's why it's coming like this. I'm not sure उसने save कहाँ किया हुआ है. I'll call him. He'll come then. वो आपको दिखा देगा.
Okay.
कौन करता है?
अरे बॉस दिखा
Yes, sir.
You're running, huh?
Yeah, I was.
Calm down, have water.
So, have you loaded it yet? Yeah, this is the one, right?
Come out.
Stand copy.
You have to close PowerPoint.
It does not have it.
Just open it once, then after removing it, just
This is the one, right?
yeah.
Okay. While the start the session, you can start your intro. Intro start. Yeah.
Good afternoon, everyone, and welcome back. I hope you had your lunch, enjoyed your lunch, and are ready to shake off that food coma. We are about to dive back into the fascinating world of virology. In the afternoon session, we will dig deep into the intricate realms of viral pathogenesis and evolution. These discussions will illuminate the ever-evolving strategies viruses employ to infiltrate and adapt within their host, shedding light on the perpetual arms race between viruses and their host. Without further ado, let's start with our next session on viral pathogenesis. This session will be chaired by Professor Sudhanshu Vrati and Professor Shoma Banerjee. Professor Vrati is affiliated with Regional Center for Biotechnology, Faridabad. He held the director's position previously and is currently a professor at RCB. He is also a visiting scientist at Pasteur Institute, Paris. His lab focuses majorly on understanding the various phases of the life cycle of flaviviruses, Japanese encephalitis virus and dengue virus, and the development of viral vaccine candidates. Professor Banerjee is currently an associate professor at Department of Hepatology at School of Digestive and Liver Diseases, IPGMER, Kolkata. Her lab studies tumor microenvironment and their role in hepatocellular carcinoma induced by hepatitis B and C viruses, as well as telomere biology in HCC development. They have also worked on identifying biomarkers for early detection of HCC in Indian patients. I would now like to request Professor Vrati and Professor Banerjee to start the session. Thank you.
So this is the viral pathogenesis session, and we have four speakers. First speaker is Professor Chandra Jha, Associate Professor, IIT Bangalore. His lab is mainly working on EB virus and Helicobacter pylori associated co-infection in cancer progression and involvement in the pathogenesis during various cancer conditions. He has recently started working on SARS-CoV-2 to identify biomarkers for COVID-19 associated neurological symptoms. And today he is talking about understanding the association of virus with neurological manifestations.
Thank you, ma'am. And happy good afternoon to all of you after the good food. And thank you, organizers, Professor Das, Dr. Tripathi, and Dr. Keshav, for giving opportunity and having interaction with a virologist. So today I am going to speak one of our project, like understanding the epistemine virus. This is the first human tumor virus discovered in 1964 in neurological modality. So earlier it is mostly with the Burkitt lymphoma, PTLD, Hodgkin lymphoma, but last couple of years we are looking in the some role in the neuroinflammation also. With that instinct we are going to discuss. So what is the EBV? As most of us know, this is the double standard DNA viruses, 184 kb, and it is a gamma herpes virus family, and it has etiological agent with various diseases, some of them like Burkitt lymphoma, nasopharyngeal carcinoma. Even it is known co-infection with the bacteria like Helicobacter pylori, with the EBV in the gastric cancer, and then the neurological disorders like various clinical study and some of the on the therapeutic level also they have seen the multiple sclerosis. This is one of the immune disorders and the neurological disorders. And some of the studies has been for the Alzheimer also, but the mechanistic has been known, not known that much. And just a graphical representation what is basically happening in the multiple sclerosis. So basically this is the normal conduction, and here the lost myelin sheath. This is the made up of lipid as well as the cholesterol moiety and other many things, and that is lost. So basically then this conduction get disrupted. So this is one of the property of the multiple sclerosis. And Alzheimer, most of us know that this is the usually amyloid beta 42 aggregate as well as the some misfolding in the tau protein. Yeah, so some possible routes that we have reviewed based on the literature as well as some of our initial work, that EBV can be entered by three ways, either by the direct entry or through the infecting the epithelial cells and then basically transfecting the B cells, transform them, and then basically doing the lot of cytokine storm as well as astrocyte and the microglia and the neural cells. So some of the hypothetical stuff that what could be the possible, we have hypothesized three routes of the EBV entry that published in the one of the journal Virology and reviewed here. But here we are going to talk more some of the mechanistic part. So before starting this in my postdoc lab, University of Pennsylvania, under the supervision of Earl Robertson, we started this. So we have selected 4-5 cell lines of those are available for the neural cells like SHSF5Y and TERA2 cells, and then we have seen then here U87MG in our here IIT Indore lab and the LN229 cells also, and then we are able to see is that really EBV is able to infect, and then their transcript level as well as protein level we can get it or not. But before that one very important study how we started, this is the 6 to 8 week fetal dead fetus that we collected in the one of the NIH center in the Philadelphia Temple University. So they aborted fetus 6 to 8 week, they are growing on the slides, and then we infected with our EBV that has the GFP tag, and then we are able to see the EBV infection as well as the lytic and latent infection also. So basically this is the primary neuron where it looks like that EBV may infect, it can infect. Then we move to the cell lines studies. So this is just the proof of concept that yes, EBV may infect or EBV can infect very efficiently the neural cells as well as many other glial cells also. So now I am starting the possible mode of virus mediated Alzheimer's disease pathogenesis, how EBV can have, and that we have driven. So starting of this work, one of the graduated student, Dikshati Wari, she has started 3 years ago. So basically all possible EBV proteins, more than 100, we collected in vitro, in silico, and then we checked the aggregation properties, because EBV can cause directly or it can maybe just a mediator, bystander. So that for checking that, we just had all proteins, and then we used these two softwares, Tango and IISc. And then for that aggregation evolution, and then proteasomal cleavage that has the software, the NetChop. So basically it's seen several peptides, and then here we took the gold standard as the A beta 42. because A beta 42 is already hallmark for the aggregation. So we compared with that, and then we found one of the protein, glycoprotein M, that is the 12-mer. It is showing somehow in silico prediction, relatively compared, or even it is higher than that. So that further those aggregate, then we synthesizes taken from the companies, and then did some in vitro assays like convoluted assay, these are the biophysical measurement to just assess that yes, whatever it is showing in these Tango and AgriScan, it is really happening in the biophysical manner also or not. So then we did the convoluted assay, thiaflavin assay, secondary structure prediction through the Raman spectroscopy, atomic force microscopy, and just check that it is only the aggregation or it has some cytotoxic effect also, as the A beta 42 has it. So that we have done, and that paper published 2-3 years ago. But some of the key results that yes, it looks like that with the various concentration, this has some shift, bathochromic shift, and who's showing the properties like the aggregation properties, and it has on the neural cells cytotoxic effect also compared to the scramble control. And then we have also checked through the AFM, atomic force microscopy, the property for the aggregation, it follows in that. So till here we think that maybe this is one of the peptide can have the aggregation property. But then in our physiology and in our brain, there are lots of other factors also. And one of the proven factor is A beta 42. So then we are start seeding this with the A beta 42. Is that it is conjugated or it making some more synergistic effect with the A beta 42 or not? So that these experiments we did in the concentration as well as the time dependent manner. And some of them we have seen that yes, A beta 42 is making, but when we are mixing this A beta 42 with the GM peptide, this 146 to 157, even the time duration for this aggregation is less. So basically it is somehow enhancing the seeding capacity of the aggregation property of A beta 42 is enhanced. And that we have checked again through the AFM, atomic force microscopy also, in presence of the A beta 42 and GM, our EBV peptide. But then these are the again biophysical, and we need to see these stuff in the cellular level also. So then we had some in vitro validation where we are putting EBV by virus also as well as GM as well on the neural cells, and then we check the inflammatory moieties because we know there are some patterns for the Alzheimer as well as other neurological disorders like multiple sclerosis. Then mitochondrial potential also, like because mitochondrial biology is also having role in the neurodegeneration, and it leads to the like apoptosis in the S1 phase. And then the calcium, because during the neurotransmitter, this calcium ion channel or calcium ion signaling is also one of the crucial factor. So that we are also included in our study. And then amyloid precursor protein and the apolipoprotein is that in the cellular milieu, although these are the 2D culture system, but then it has some effect or not, that we have also assessed it. And then one of the key protein of the myelin basic protein MBP in the multiple sclerosis, because this is one of the very crucial for the cholesterol associated markers in the case of chewing of the myelin sheath in the multiple sclerosis. So that we have also took in the consideration to check. So as these are some of the inflammatory and the pro-inflammatory and anti-inflammatory, some of the markers it looks like. So here if you see control cells, then EBV 12 hours. EBV 12 hours means our virus, purified virus 12 hours, purified virus 24 hours, purified virus 48 hours. And then this is GM peptide 12 hours, GM peptide 24 hours, GM peptide 48 hours. So these are the some responses that we have observed, that yes, IL-1 beta, or we can say if IL-6 in the case of even GM 24, 28, and in the, some of the cases in the later time point like TNF alpha and the TJ beta also. Although some more work needed to do, this is the one set of the result we can say. The initially it is showing that yes, there are some responses with the EBV as well as the GM peptide also. Then we have also seen is that something with the transcription factor. It looks like yes, in some of the time point NF-kappa B is also regulated here in these studies. Then to understand the mitochondrial association, because in the neurodegeneration, one of the postulates is that G1 to S and then those phases, it is going to the apoptosis because of the mitochondrial disbalance. So what we have seen, mito red, that is the basically action potential, and the green, that is the basically mass. So somehow it looks like the mass is not that much affected in these cases, in these time points. but the action potential is significantly downregulated. So further to understand more, and we talk with several our collaborators those are working in the mitochondria biology, we have looked at some of the genes also, because we know that the fusion, because in the stress condition, this is one of the property they can mix together, like 2 or 3 or 4 cells, they are mixing together and having the bigger cells to avoid the stress condition. So fusion genes as well as the fission genes also. So some kind of stress condition, it is also known that they are breaking out. So what kind of stuff is happening in our cases? So it looks like that in the GM as well as in some case of the EBV also, the fusion genes like mixing mitochondria, overlapping mitochondria is significantly higher compared to the fission genes. So fission genes is relatively less responsive compared to the fusion genes. And this we have seen at the transcript level, the protein level that also we have seen it, but not, I'm showing the data here. And so here it looks like that OP1 is one of the crucial compared to the other one, and this now we are going to delineate for more studies either by the knockdown as well as the inhibitor or other pathways. And here also if you talk about the down regulation, so mid 49, 51 and DRP1, it looks like has some contrast or significant effect. So that also in our future study to delineate how exactly it is going on. And then as I just mentioned that the calcium ion is one of the crucial in the neural signaling, is that really also have one of the factor in this case. So that we have also seen as the total, although I can say it is not that much, but then it in the 2D system, in the cell lines, it looks like yes, there are some thing going on with the this calcium ion signaling also. The more detail that also in our future scope to understand exactly what pathway and what mechanism it is going on. But then all these are the molecular marker, but as these are the related with the disease marker also not, so then we have seen APOE4, APP and for the Alzheimer, and it looks like that EBV 12 hours, 24 hour, 48 hour, and then the GM, it looks like that it had enhanced the APOE4 also, because we know that APOE4 the degradation is not happen properly in case the aggregation started, because the energy get lower and they have that. So it looks like that in this case also this degradation is halt or I can say reduced, and similarly APP also looks like higher in some of the time points. Yeah, and then because this then we have seen this APP and APOE4 in the like this microscopic level also. And one of the hallmark for the MB multiple sclerosis, that is the myelin basic protein. As I mentioned, this myelin basic protein is one of the important component for the cholesterol biosynthesis and in the myelin sheath. And that has been known one of the hallmark, like APP and APOE4 hallmark in the Alzheimer, MBP is one of the hallmark in the multiple sclerosis. And in the temporal manner, some of the time point, this MBP also seen. And recently then we have, because this is one of the question asked by the reviewer also, and we also asked, not much study, but there are several studies saying that these multiple sclerosis and Alzheimer are coexisted also. Although one is very high at a time, so second one may not be that much diagnosed, or we can say, but it is not like that. There are some studies or some kind of clinical importance that they may coexist as well. And then, so as we got the MVP level high, then we also try to understand exactly, because this is the cholesterol biosynthesis pathway that we need to understand, and we delineate that. So with all these pathway, we have seen that some of the important parameters like FDPS and this one, the thiolases are one of the crucial components. So now we have to go more detail in the enzymatic level and then the protein level also, that exactly they are having crucial role or they are just a bystander or working as a cofactor. So till this in vitro study that published and done, but then we followed this in the in vivo model also. Although making in vivo model for the herpes virus is one of the very tedious, and it need B cell, T cell negative one. Last 4 or 5 years we had several failed attempt, but then last couple of years we are adding some immunosuppressive some doses, and then for 5 doses in alternate days, and we are able to see the immunosuppressive mice, even these are the Swiss albino. And then we are asking these questions, adding the EBV also as well as the another group with proper control, peptide also. And then we did this, so basically infection in the IMR32, that is the neural cells, and with the 5 million cells in the 3 doses every week. And after that we did 35 days observation. And then we see the behavioral assays like a man with Morris water as well as the elevated plus maze, and then we also collected the brain. There we did the tissue as well as IISc and Western blot and the serum with the inflammatory markers also. So here what we have seen, the inflammatory markers IL-6, TNF-alpha and NF-kappa B via ELISA and IISc and Western blot, and then also I can go the more slides. In the result part, the time is coming out. So in Morris maze water, at least in the group 3 or 4, that is group 3 is like a mice exposed with the EBV, and group 4 is the mice exposed with the GM. So somehow the neurological behavior or the we can say the disorientation has been observed more in group 3 or 4 compared to the others. Even they are having only IMR cells, or this is the controlled one. Some of these behavioral studies, as well as this is we collaborated with the pathologist in the hospital, because we don't have much idea about that, and they after the validation they said that this G4, G3 and G4 exhibited disorganization of the cells in the hippocampus and dentate gyri region. This is one of the clinical features for the neurodegeneration. And then the cell shrinkage was also observed in the olfactory region of the G3 and G4 in the IISc. Further, we did ELISA with the serum, and then for the TNF-alpha as well as the IL-6. And then we have seen some of the elevated level in the G3 and G4, significantly for TNF-alpha in the mice brain, as well as IL-6 mostly in the G4. That is the only GM, not the EBV one. That has been again find that with the MVP also. So there are something different for the GM as well as the EBV also, but more detail we needed to go. And from the mice brain also we have did this NFKB, and we found the significant difference even in the IMR also for the NFKB, and only IMR, EBV and EBV IMR and EBV GM. So yeah, and then Alzheimer's associated diseases. Sorry, this is something. So till now, what we have hypothesized that in vitro, when we are having infection with the epistemic virus and glycoprotein M, basically there are some properties for the aggregation, and that we further look into the mice, and there are 4 different kinds, we can say, when it is going to the mice. The first EBV may go inside, like we are giving the nasal spray for the EBV, the doses, and going to through the endocytosis, and basically it enhances these pathways to the NFKB, and then base 1. This is one of the markers that also we have seen. This is the one of the critical in the Alzheimer's disease, and through this basically it is making the activating of these secretase, and then the APP cleavage occur, and this further leads to the making the ApoE4 aggregation, fibrils, and the making the plaque. That is the one of the chain that is usually it follow, and ultimately it enhances the kind of, we can say, the neurotoxicity or cytotoxicity in that compartment. So I would like to acknowledge my postdoc PI, where this work started initially, and then my international collaborators. Some of them are contributing in this work, and my national collaborators. And some of the grants for this, like we had earlier CSIR, and now SPARC we got with the Switzerland, University of Basel, and some other grants also. So thank you. And yes, most importantly, my group here, the Anu is presenting poster also. Nidhi also presenting one of the poster with the kinases and cancer, and Buddhe Baral is presenting poster in SARS-CoV-2 E protein channels and RIPK1 pathways. And this is just a general cover letter. Thank you.
Interesting talk. So I was just wondering that upon exposure to GM, the ATP level goes up to 24 hours. But you showed that after 48 hours, it's sharply decreased.
Yeah, because these aggregation properties are not biological, they are the biophysical. So after a certain peak, that is the biophysical property, they may get a quench through these dyes.
So I wasn't clear about the lipid cholesterol metabolic pathways. You see a down-regulation or an up-regulation?
And some of the paths are upregulated and some of the paths are downregulated also. But then we have to go in more, because that is one of the factor where GM and EBV acting little bit separately also. Like group 4 has a separate indicator compared to the whole virus. So we have to go in more detail. I can say this is just a yeah, please, please.
It's a little off track.
No.
So recent studies have shown that EBV infection is also linked to chronic fatigue syndrome. You've seen this.
So EBV is one of the omnivirus, and believe me, when we started some cells to take as a control that may not infect with EBV, most of the time we saw those cells also get infected with EBV. So, and then as they are the kind of opportunistic, like it get induced, like if there is H. pylori infection, if there is SARS-CoV-2 infection, or if there is some other kind of infection, they get induced, they get active cycles. So a lot of other diseases also has been associated and clinically reported, but mechanism-wise, mostly B cell mediated are delineated till date, others are not as much.
I must have missed it. What is the natural route for infection of EBV?
So it has been known the saliva. Yeah.
You are giving it this. You are giving it through the nose.
Yeah, nose. Yeah.
That has the same effect.
Yeah, it has been known that, so yeah, this question actually coming earlier also with the reviewers as well as the collaborators. So herpes viruses had one route, they are going through this PNS system. And then in one of our study, I am not showing here, there we did what? We simply doing the direct infection, and then second we did the PBMC infection, and then incubated with these neural cells, different compartment. And third, infecting the B cells like PBMCs, and then transferring the supernatant, because we know these days enteric nerves also they are transferring. So, and we saw 3 different mode of the pathogenesis. So all these ways like get control brain and many these axes. So they are transferring their secretome, and these viruses are taking help of them. Yes, please.
Okay. So in your behavior studies, you had shown that if you compare EV
the cellular mechanisms which take place in which you are explaining, either going to the DNA structure, genetic structure, or to the observed pathological structure.
Yeah, thank you sir for this question. So we started with like need control, and then only cells, and then viruses as well as the GM also. So we are first checking the behavioral differences, and then we take out the cellular stuff like serum as well as the particular tissue, and then we have seen that also. And then definitely these are the pathological features, because in the brain tissue when we are collecting from the group 1, group 3, group 4, and then we just have simply slices like immunohistochemistry, and then we are consulting the pathologist. Those are like 30-40 years doing these neurological assessment in the hospitals actually. And then they are basically as a unbiased manner they are telling, okay, these could be the one of the features and these are features. So definitely these studies need to go further more detailed, because there are so many things we need to understand. But these are just one stuff that looks like at the behavioral as well as at the cellular level also, and the biochemical level also something is going on. It is not completely bystander. It looks like there are some responses.
But I think I did not explain my question properly. When you take out the cellular tissue from outside human body, then it's complete behavior, its complete structure changes, its chemistry changes, And how do you know, what is the rate at which its chemical structure changes, its physical structure changes, and so what can you make out if you study it after 6 hours, 8 hours, or whatever it is? See, you should study it in situ actually, in the brain, which is not impossible, particularly for human beings.
Yeah, so
I've seen a lot of neurologists talking about putting EEG, taking EEG, putting probes and trying to get correlation of behavior with the signaling, how the signal travels in the neuronal structure of the brain. But it is not easy to...
yeah, definitely this is a little complicated phase. That's why, sir, when we started this, I just wanted to mention it here. We have started when in UPenn, we got 30 autopsy of the patients, 6 groups, 5 each. So 5 completely, we can say the age matched control, and then early Alzheimer, early MS, and then late Alzheimer, late MS, those brain autopsy. And then we first study that embryo paper we had. We have checked the expression of these markers as well as the EVV presence also. And there we have seen that somehow those are in advanced stages and the early stages of the Alzheimer as well as the of the MS. They may have some significant correlation. But then, as you are saying, definitely organoid models are one of the important factors that can be seen at the real-time manner. And in the future, that we are planning to see these viral infection and in the disease model, in the organoid model.
Thank you, sir.
Sure.
So, our next speaker is Professor Amit. He is an associate professor at IISc Bengaluru. His laboratory is mainly working on gaining a comprehensive understanding of redox signaling and its role in governing the virulence traits of two notorious human pathogens like human immunodeficiency virus, HIV, and mycobacterium tuberculosis. And today he is talking about
Just a second.
hydrogen sulfide modulus HIV latent.
Thank you. So before I start, I just met during tea time Professor Sudhanshu Rathi, and before I could say hello to him, he said, so you are also virologist. So that pretty much sums my contribution in virology. Just to make it clear, but I actually thank all the organizers for giving me this opportunity to talk about some of the work we have done on HIV related to metabolism and how metabolism can actually control HIV latency and deactivation. So I'm going to tell you about hydrogen sulfide gas, which most of our cells produce, and how that is critical for this virus. So this is a favorite slide for all HIV virologists, where you would see that in an HIV patient, if you look at HIV plasma viral load, it increases in acute phase, and then because of CD4 T cells, it's going to come down a bit. And then with antiretroviral therapy, we have a very highly effective antiretroviral therapy. Here you would see that viral load goes below detection limit with occasional blips. And as long as you are on antiretroviral therapy, there would be no reactivation, virus would remain latent. However, if antiretroviral therapy stops, viral rebound happens uniformly in most of the patients in a matter of weeks to months. So this is a major problem in the field where virus persists in a latent but replication competent state. And that is the focus of some of our work, where we wanted to identify host pathway and viral pathways that are actually working together to contribute towards latency or reactivation. However, this slide was actually not the reason why I got into HIV. I got into HIV because of the person here, Len A. Heisenberg, whom probably you would know as the father of FACS. He had done tremendous studies using flow cytometer and developed a lot of FACS-based techniques. Len and Lee, seen here, were lifelong partners, and both of them collaborated, husband and wife collaborated on each of the papers Len published. So he published quite an interesting work in 90s, and not even 90s, later 70s and all, in 1997, where he showed that glutathione deficiency is actually associated with impaired survival in HIV disease. And this was really a landmark finding, where glutathione, which is a major antioxidant in our body, which is a tripeptide made up of glutamate, glycine and cysteine, they have been able to develop a technique wherein you can actually measure glutathione level inside the cells by using alkylating agent monobromobimene, MBBR, which is cell permeable. It gets into the cell, conjugate with glutathione, and you get a fluorescence signal. So you get a very nice fluorescence signal, as you can see here. So this is the technique which Len has developed, and what he showed that if you take HIV patients and you look at the glutathione levels in the CD4 of these patients, CD4 cells of these patients, they would identify a threshold value, and below that threshold, if glutathione is lesser, you would see that within 2 to 3 years, the outcome of HIV is very different. Survival is quite less as compared to those patients who have a CD4 glutathione levels higher than the threshold. So this was one of the key findings wherein actually glutathione levels of CD4 T cells can be taken as a yardstick to predict survival of HIV patients. And based on this argument or this data, this is a health supplement which we probably all aware of, N-acetylcysteine, which is a source of cysteine in the cells, and cysteine actually get incorporated in glutathione. So you can actually reverse glutathione deficiency by treating patient with N-acetylcysteine. So they have done an elegant experiment, although this experiment was with a very low number of cells, with very low number of patients. They have been able to show that if these patients were treated for about 2 to 3 years, for about, with N-acetylcysteine of about, 3,000 milligrams per day, there is a clear difference between the survivability. Those patients who have taken N-acetylcysteine, they survive much better as compared to those patients who have not taken. And also, as compared to placebo, glutathione levels were restored in those patients which have taken N-acetylcysteine. Yeah. So this was really a very interesting finding, and that sort of motivated us to work on redox aspect of HIV. However, there were some shortcomings, and one of the shortcomings was that MBBR dye is actually not very specific to glutathione. It also is stained in other thiols, so you really don't know whether you are measuring glutathione or you are measuring a collective thiol status of the cell. Also, the relationship between glutathione and viral load was never investigated, so we didn't know whether a glutathione level going down mean viral load going up or otherwise, right? And mechanism of NAC, N-acetylcysteine, although it is a source of cysteine, it also does a lot of other things. It also produces hydrogen sulfide, it also modifies protein by as per sulfidation. Therefore, we thought that there is much more than what this paper has shown. So we started with all these 3 objectives and tried to fill knowledge gap in the field. And one of the first thing which we did is that we used a glutathione specific biosensor that measured glutathione inside the cells without lysing the cells. So it was a non-invasive genetic tool which we have used, and we started humbly, with cell lines without going into too much of a primary cells and all to start with. And we use U1 cells, which is a latent cell line model of HIV, where HIV genome is integrated in U1 monocyte. You can actually reactivate virus by PMA or TNF alpha, and then you will see that in a time dependent manner by GAG transcript, virus nicely comes out of latency. We also measured oxidative stress using GRX1 ROGFP2 biosensor that measured glutathione oxidation inside mitochondria or cytoplasm, and you would see that as virus comes out of latency, you also see glutathione getting oxidized. And this is a glutathione pathway wherein cysteine can get into glutathione. So we wanted to understand whether this is cause or consequence. Would it be possible that virus reactivation is leading to oxidative stress, or oxidative stress actually reactivates virus? So we can actually oxidize glutathione by a simple method of treating cells with H2O2. If you look at glutathione peroxidase, which is a major enzyme in the cells, which convert H2O2 into H2O using cofactor glutathione, and as long as glutathione is replenished by glutathione reductase, you are able to keep this cycle going. So we thought that we can actually oxidize glutathione by hydrogen peroxide and then ask the question, can we reactivate virus? So we did this experiment at a low concentration of hydrogen peroxide. We did not see much of a virus activation. Perhaps cells are able to take care of oxidative stress. So we combined hydrogen peroxide with BSO, which is an inhibitor of glutathione biosynthesis. Rate limiting step is inhibited here. And when we combine with glutathione inhibitor, we could see virus reactivating nicely. So what it tells you that glutathione oxidation is important, and as long as they are able to take care of oxidative glutathione by converting it back to glutathione, you will never get virus reactivation. But if you combine it with the inhibitor of glutathione and overwhelm the system, you will see virus getting reactivated in this model. We increase high concentration of H2O2 also, 1 millimolar, 2 millimolar, which is going to sort of overwhelm the system. Here virus comes out, and if you then treat cells with N-acetylcysteine, which will replenish glutathione, we are able to suppress virus. So it was very nice correlation wherein glutathione oxidation sort of promotes HIV reactivation and supplementation of glutathione suppresses it. However, genetic evidence was important, and genetic evidence in this case was very difficult because if you tweak around with glutathione level genetically, cells invariably undergo apoptosis or necrosis. So it was really very difficult to prove this model genetically. So we went around and we then collaborated with Professor Mukesh, who has these beautiful nanoparticles, nanoenzymes, which are actually mimicking the activity of antioxidant enzymes without disrupting the level or activity of the natural enzymes. So these are the enzymes like vanadium pentoxide is a nanowire which has a surface chemistry so that it mimics the activity of glutathione peroxidase like natural enzymes. So we use these nanowires, we treated U1 cells and tried to ask the question if the reactivation is suppressed because we have augmented the glutathione peroxidase activity, we have boosted glutathione peroxidase activity, and you can see that in U1 cells when you give PMA there is reactivation, when you combine PMA with a nanoparticle you are able to suppress reactivation. Similarly also in human monocytic cells from one of the donor, we are able to suppress HIV replication when we combine with this nanomaterial. So this is just a proof of concept of how you can manipulate a redox balance to control HIV. Another study which is ongoing in the lab is with another nanomaterial which is cerium vanadate, where these nanozymes actually mimic mitochondrial superoxide dismutase, and mitochondrial superoxide we have seen goes up as virus comes out of latency. So we collaborated again with Professor Mukesh and with some of the brilliant students in the lab. They have been able to show that if you pre-treat cells with these nanoparticles which mimic mitochondrial superoxide dismutase, which reduces superoxide levels in mitochondria, you are able to control virus reactivation as much as NAC was able to do. And you will see also in CD4 T cells, HIV replication was completely suppressed if you pre-treat these cells with cerium vanadate nanoparticles. So this was one of the example of how redox modulators can be used to understand how virus replicate, reactivate or undergo latency. So coming back to NAC, because our original question was how N-acetylcysteine was actually acting, and as I showed you, N-acetylcysteine is a precursor for cysteine, it goes into glutathione pathway. However, we also know that in addition to glutathione, cysteine also goes into sulfur metabolism, wherein it generates oxidized form of sulfur, sulfine sulfur species, and in doing so, it also produces a lot of byproduct hydrogen sulfide gas. Hydrogen sulfide gas is now considered as a gasotransmitter. It has a lot of regulatory role. So we immediately got interested in identifying or finding out whether the NAC effect could be because of hydrogen sulfide variation in the HIV infected cells or when HIV cells are under latency or reactivation. So two of the graduate students started looking into the role of hydrogen sulfide in HIV latency and reactivation. And this is a pathway how hydrogen sulfide is produced in the cells. We take methionine in the diet and that get converted into cysteine. Some of the enzymes CBS, CTH, MPS3 does the job, and in between they also give out hydrogen sulfide. So we looked at the expression of these enzymes in our U1 model. We have tested in many models. I'm going to give you an example of U1. Here you see that virus comes out of latency, and you would see that at least one of the enzymes CTS, cystathionine gamma lyase, as whenever you add PMA, you see a decrease in the level of CTH, not only at the level of protein, but also at the level of transcript. We directly measured hydrogen sulfide levels by a dye, which is very specific to hydrogen sulfide. Here you would see that the latent cells have more level of hydrogen sulfide as compared to the reactivated cells, and measurement is here. So with this, we wanted to at least ask a question that could it be possible that CTH enzyme, which is responsible for hydrogen sulfide production, also regulate virus latency. And one way to check is that if you can knock down CTH and then ask the question what happens to virus latency. See, we created knockdown of CTH by shRNA, and here you would see nice knockdown. H2S level also goes down, and very surprisingly we found that if you knock down CTH, virus spontaneously reactivate. You don't need TNF alpha or PMA to show reactivation. If you combine it with PMA, you see much better reactivation. So by knocking down a single protein of sulfur metabolism, we could reactivate HIV from latency and much more in the presence of PMA. One could ask that if you look at the activity of this enzyme CTH, it converts cystathionine to cysteine. So one could ask that this effect, what we are seeing, could be because there is no cysteine and perhaps there is lack of glutathione, and therefore you see virus reactivation. But we measured both cysteine and total glutathione. Both of them were not changed in CTH knockdown, which means that this is happening independent of glutathione levels. And hydrogen sulfide, if you look at the literature, it is known to donate electron to mitochondrial respiratory chain. So we thought that if you don't have hydrogen sulfide, you would have aberration in mitochondria. And we measured by seahorse, and you could see that in the CTH knockdown cells, a lot of mitochondrial bioenergetic parameters are derailed, like basal respiration, ATP production was down, spare respiratory capacity was down. And also in these knockdown cells, along with PMA, they see a lot of mitochondrial loss. So what is happening in this knockdown cell is not only H2S is not there, mitochondria is also derailed and mitochondrial loss is also increased. And that contributes to HIV reactivation. So we wanted to then ask the question, can we reverse all these events? Do we have H2S donors which we can then use and reverse this whole cascade? So we did this experiment using a slow releasing H2S donor, GYY4137, which releases H2S in a slow manner where entire course of duration you see a slow release of hydrogen sulfide. If you use millimolar concentration of hydrogen sulfide inside the cell, it will be around micromolar level for quite a few days. So we use this donor, and then we ask the question, can we reverse CTH mediated virus reactivation? And if you look at here, this is how virus reactivate in this CTH knockdown cells. If you look at H2S donor, it comes back. Also in case of PMA treated virus reactivation in these knockdown cell lines, we can reverse virus reactivation by just adding hydrogen sulfide donors. These donors were not really toxic. And also, in a normal HIV cell lines, U1 cell lines, latent cell lines, which are not knocked down for CTH, we can activate virus by PMA, but with hydrogen sulfide treatment, virus reactivation was completely abolished, equivalent to NAC levels. So just by adding hydrogen sulfide donors, we were able to keep virus silent. We wanted to understand the mechanism, so we did RNA sequencing analysis under these conditions, and we identified that antioxidant response seems to be highly induced in hydrogen sulfide treated cells. Okay, so it appears that hydrogen sulfide sort of induces antioxidant response in these cells, and the way it can do it, that this antioxidant response is actually under control of a master regulator called as NRF2. NRF2 is generally sequestered along with KEAP1, which is a part of ubiquitin ligase, and that promotes, actually under normal growing condition, it promotes NRF2 degradation. So NRF2 does not enter into nucleus and does not activate antioxidant response when it is not needed. But in case of hydrogen sulfide treatment, KEAP1 gets persulfidated at 16 thiols, and upon persulfidation, it is no longer able to allow NRF2 to undergo proteasomal degradation. NRF2 accumulate in the cytoplasm, and then it goes into nucleus to induce antioxidant response. So we asked the question, would it be possible that hydrogen sulfide treatment is actually doing this? And we identified that in response to hydrogen sulfide donor treatment, NRF2 is accumulated. Not only it is accumulated, it is also inducing the genes it was supposed to induce. One of the markers is heme oxygenase, antioxidant gene, which is going up in hydrogen sulfide treated cells. We also identified that some of the transcription factors, which are also acting as the host restriction factors, are induced in response to hydrogen sulfide, like the inhibitor of NF-kappa B, STAT1 pathway, IRF, and epigenetic regulator YY1. So we checked also them, and NF-kappa B phosphorylated level is seems to act as an activator of NF-kappa B activity, and the level of phosphorylated subunit of P65 NF-kappa B is down when treated with hydrogen sulfide donor. And we also checked its actually enrichment on HIV LTR. HIV LTR enrichment is also going down, which is consistent with the fact that NF-kappa B is one of the main activator of HIV from latency, is going down in hydrogen sulfide treated case, therefore HIV would remain silent. Another epigenetic silencer, YY1, which gets recruited more on HIV LTR when it has to undergo latency. And in hydrogen sulfide treated samples, we see that YY1, which is the epigenetic silencer of HIV LTR, is more recruited on HIV LTR. So these are some of the mechanisms we could find out from this study, which can directly link hydrogen sulfide activities to keeping virus silent by some of these mechanistic it is playing out. So finally, we wanted to also connect cell line data with the patient data, and that would give us much more credibility to what we are doing. So we collaborated with some of the clinicians, and we were able to use this ex vivo model of HIV latency activation, wherein you take PVMCs from HIV patients which are on ART therapy. So the viral load is very low. You isolate CD4 T cells and then activate these cells by PHA and IL-2. Virus would come out of latency, and then you treat these cells with antiretroviral therapy in the presence of IL-2. So they would again go down into latency because of the action of ART. However, they are not eliminated, because once they are suppressed, you can again reactivate them with something like PMA or prostatin. So we wanted to use this system to see if we can combine our hydrogen sulfide donor with antiretroviral therapy to keep virus suppressed and does not allow reactivation to take place. So we did this experiment with 5 of the ART suppressed HIV patients, and we are able to show that within 14 days you see virus getting reactivated. By 28 days, due to the activity of ART, it goes below detection limit, and after 28 days you sort of activate that by prostatin, but you remove ART or hydrogen sulfide donor, and you will see that in the cases where you have given only ART, in all of these cases virus comes out of latency and reactivated. But in the cases where you supplemented ART with hydrogen sulfide donor, virus suppression was ranging from 100% to 80%. So hydrogen sulfide addition with ART, at least in this ex vivo short-term model, was able to keep virus silent. So with this, this is my summary slide where we were able to show that hydrogen sulfide is important for keeping virus latent, and it does so by activating NRF2 pathway and antioxidant response. It also stimulates mitochondrial respiration. It stimulates the activity of YY1 epigenetic silencer of HIV LTR, and also blocking NF-kappa B activity. And we hope that in longer term, hydrogen sulfide donor could be combined with ART to see if how long we can keep a virus in a deep latency and not allowing it to reactivate. With this, I would like to acknowledge my group, a bunch of very enthusiastic students. Some of them are here, and this is the track we recently went near Bangalore, and I hope with the kind of money Shashank is generating, next meeting can be in Coorg or somewhere. We can also go for tracking, and with all the funding agencies. Thank you very much for your patience, and I'll be happy to answer any questions. Yeah.
I was curious, I would have thought the general trend in the field has been that you activate the latent viruses and try to get rid of them, because that's a more permanent principle you are showing that you can suppress it.
Yeah, so there are two thoughts. One is that you can shock and kill the virus, shock and kill approach. The problem with that kind of approach is that reservoir is so diverse. You don't really have enough latency reversible agent to reactivate virus from all the reservoirs, and that remains a major problem. You could never reduce the reservoir size even with the clinical trials using latency reversible agent. So we think that rather than doing that, maybe it is good that you block and lock virus rather than shock and kill. And block and lock has actually a natural paradigm. If you look at your elite controllers, which are able to control virus without ART, they are doing with some of the epigenetic mechanisms that allow virus to remain silent or get integrated in some desert, genomic desert, where it does not get reactivate. So we think that these kind of strategy which can induce deep form of latency may be beneficial, but this is something we need to test. It is available, commercially available, but we are also collaborating with some of our chemistry friends from IISc Pune to come up with donors of hydrogen sulfide that can directly deliver only in mitochondria, not in cytosol. So some specificity can be achieved in that sense. Yeah.
Nice talk, Amit.
Yeah, please go ahead.
Yeah, so the concentration of H2S, that was not mentioned. So is it?
So I think it also depends upon cell line to cell line in primary cells. In most of the cases in micromolar level, only in monocytic cell it goes to millimolar level, but it is a slow releasing hydrogen sulfide donor. So inside the cells, it would remain always in a low micromolar range.
But are there any collateral effects of H2S going on that you have?
So that's an interesting question. H2S has two effects. In high concentration, it is going to inhibit respiration, it is known. In low concentration, it is going to have beneficial effect where it would act as a stimulant of respiration and also anti-inflammatory. So as long as you keep concentration low, it's going to work.
Here, on the right.
Yeah.
Thank you for the talk. Hydrogen sulfide signaling has been integrated in a lot of diseases and for maintaining health status. My question is, this ART treatment is known to reduce on a global scale
protein persulfation in
I don't know that. I don't know the question, answer of your question. We need to check whether ART treatment alter persulfidation level. But my thinking is that a long-term ART treatment, because you are on therapy, it is going to reduce the level of glutathione and cysteine, perhaps because it is going to, chronic oxidative stress is going to happen. Therefore, the persulfidation level can be altered. This is something we need to check. So at this point, it would be very, very difficult for me to comment on persulfidation level. Although I think there are clinical trials going on in US where they are doing ART interruption and trying to see how much time it takes for virus to rebound. And in different patient, it takes very different trajectory. In some patient within few weeks, some patient months, some patient it takes years. So we don't know what kind of metabolic changes are happening. At least in that study, it suggested that TC cycle intermediates and pentose phosphate pathway are disrupted in the patient which rebound virus quickly. So we think that some of these mechanisms might play out there also.
So with the H2S effect on HIV1 and HIV2 is different. If you consider Lugar's work of NF-kappa sites varies between the 1 and 2 and the mutation frequencies, then H2S should have a differential effect?
It's possible. It is possible. We have not tested it, but this was something, a very interesting question actually. Even B or C subtype may have differences, because B subtype have differences in NF-kappa B thing, and also TAT is different. So all of these questions are there in the lab, and students are probably going to address at some point. Yeah. Thanks. Yeah.
Talking about association of host factors and immune response with clinical outcomes in
Thank you, sir. I thank Dr. Das and all the organizers at IISc, sorry, not PHSD, at IISc, who actually have taken care of us and given us good lunch. I hope everybody is still awake. There's still one more talk for the tea break. So, yeah, so I'm going to discuss some of the work that actually we were planning to start before COVID, and we were not able to really take them forward because of the last 2-3 years of other activities with COVID-19, and things that we have been able to actually again put things back on track, and we have started really working on this. So most of this is work in progress, and these are, as most of you can understand, these are clinical studies. So most of this is observation, which will lead to generation of hypothesis. So I'll not waste time in introduction, because we have already heard about flaviviruses and dengue virus also in the talks in the morning. So we'll directly start with the crux of the matter, which is dengue vaccines. So most of you know there are 4. Dengue is not just one virus; it is actually a group of 4 different viruses with different antigenicity, related viruses but different antigenicity. And to really get protection from dengue disease, you need to have immune response that can act against all the 4 serotypes. Therefore, all the vaccines that you can see here which are under development are tetravalent vaccines, and that's a major challenge, because you need to develop a vaccine that can generate immune response for all the 4 viruses. And okay, I can use this slide. So what are the vaccines that are currently that are either licensed in other countries or have reached phase 3 in other countries are listed here. So this is the Sanofi Pasteur live attenuated vaccine, which has a yellow fever backbone, and the PRME protein here is from dengue 1, 2, 3, 4. This is Takeda vaccine, which is licensed in some countries in Europe and Indonesia, and here the dengue 2 backbone, and then 4 PRME of DEN 1, 2, 3, 4. This is the NIH vaccine, which has dengue 1, 2, 3, and then dengue 4. So dengue 2 PRME is expressed in dengue 4 backbone. Therefore, all these 3 viruses were designed to generate a response for all the 4 vaccines. And if you look at the overall efficacy, the Sanofi Pasteur vaccine, the efficacy ranges anywhere between 30 to 60%. And now the interesting part is, if you look at the efficacy in seropositive individuals, it's between 75 to 83%. And interestingly, in seronegative individuals, these are people who never were exposed to dengue before getting vaccine, the efficacy is barely 35 to 43%. So this is a vaccine which probably doesn't protect 1/3 of the people who get the vaccine if you are not exposed to dengue. Now, what is the situation with Takeda? The overall efficacy is pretty much comparable, I would say. So you would get around 62% overall efficacy, and the people who are already seropositive for dengue when they got the vaccine, the efficacy in them ranges between 52 to 83. And here again, if you look at seronegative population, the efficacy can be as low as 43.5 and can be as high as 91.9%. So this vaccine is licensed very recently, last year in Indonesia and also in Europe, and none of these vaccines are in India. The vaccine that currently Indian companies are working on and the phase 3 trial that perhaps ICMR would fund is this vaccine, for which as of now there is no data available in the public domain. So what is the issue with this dengue vaccine and Takeda vaccine? As you can see here, when you look at the serotype specific efficacy here for dengue 1, it's 46, and this is in 2 to 9-year-old people, children. Dengue 2, it's 33.6%, and for dengue 3 and 4, it's given here. Interestingly, in seronegative people, the efficacy is 14.4%. It's pretty much negligible. And the other aspect of dengue vaccine trials that people should realize when they say they are working to make a dengue vaccine is the size of these trials. These are humongous trials, the largest ever conducted for any vaccine development. So anybody who says I want to make a vaccine for dengue should really first look for a commercial partner who is willing to invest in these kind of trials and conduct a trial. And so these are 2 different trials, and again here when you look at seronegative population, the efficacy is really less than 50%. However, when you look at the efficacy of vaccine in preventing severe disease, it's really pretty good. It's over 90% in adults, but in children it's again still only 66%. So we want to actually protect children who are more vulnerable from dengue, and if they are seronegative, they pretty much will have no use with vaccination, and the protection against disease is also really nominal, or at least barely above the acceptable range. And when we look at Takeda vaccine, as I said, so the vaccine efficacy is 64 in seropositives and 53.5 in seronegatives. And when you look at serotype wise vaccine efficacy, in seronegative people, the data exists only for dengue 1 and dengue 2. So we don't even know whether this vaccine will really protect against all 4 serotypes in seronegative individuals. In fact, this data suggests that there could be enhancement of disease, which is what even the Sanofi Pasteur vaccine showed, and that actually led to a big problem and even leading to withdrawal of vaccine in countries like Philippines. And now now WHO has recommended that the vaccine should be given only to seropositive individuals. Now this data from NIH vaccine is available only on the website of Butantan, which shows that they have efficacy data only for dengue 1 and dengue 2. So even for the 3rd vaccine, which we are investing heavily in India to start clinical trials, we don't know yet whether this will protect against all 4 serotypes. So what is the problem with dengue vaccines? So when you vaccinate a person who is never exposed to dengue, so this vaccine is live attenuated vaccine, it will elicit both protective and cross-reactive antibodies. When this vaccinated person now is exposed to natural dengue infection, this acts like a secondary dengue infection, and in countries where this vaccine was given to seronegative people, it led to increased hospitalizations. So this is a big problem with dengue vaccine, that when you vaccinate a seronegative person, the primary infection which they get after vaccination actually becomes a secondary infection. And what happens in a positive individual? So seropositive individual already has antibodies to dengue, and therefore he or she gets a live attenuated vaccine. So this vaccine cannot cause disease. Therefore, they elicit higher amounts of antibodies and are protected for remaining duration of their lives. So with this background, we have multiple questions to really ask in a setting like India, which is endemic to dengue. So we don't even know what is the current situation of serostatus of Indian population, how many people really have antibodies to dengue, how many people have neutralizing antibodies to dengue. Are these antibodies really neutralizing? Are there enhancing antibodies, which is again an issue with secondary dengue infections. So when you have primary infections, you make neutralizing antibodies, you generate T cell responses, but we have no idea how protective they are, how long they are going to last in the system. And when you're then exposed to secondary infections, your memory responses will elicit a response, but it will mostly be against the virus that you are exposed earlier. Therefore, it will be a suboptimal response leading to severe disease. So we don't even know what is the situation in Indian population when people are getting repeatedly exposed to dengue. What kind of memory responses do we have? What kind of T cell responses do we have? What are the viruses that are circulating in India? So the questions that we are interested in the last 3 years, and we would like to really invest our future, is really understanding the seroprevalence of dengue. What is the burden? What is the immune response upon natural exposure? What are the circulating virus isolates here, and what determines the change in serotype dynamics every year? Can we really identify correlates of protection? And if at all there is a vaccine trial which happens soon, what will be the efficacy of vaccine? Can we really contribute to that? And interestingly, and again we will have a session on antivirals. So we talk very less about antivirals for dengue. So can we really also, in addition to vaccine, focus on direct acting antivirals and also host directed therapies? And in general, can we have an ecosystem to facilitate vaccine and antiviral development for dengue? So what we did was we had a cohort of acute febrile illness where we tested them for dengue NS1, IgM, IgG, looked for seropositivity using indirect ELISA, and conducted neutralization assays. In patients where we could isolate RNA, we isolated RNA, we measured viremia and serotyping by nested PCR. And this is our data, where in children less than 15 years, 50% of our children already have antibodies to dengue. If you look at adults 16 to 45, 72% of our adults already have antibodies to dengue. If you take above 45 years, 89% of adults above 45 years have antibodies to dengue. So what do we do with vaccination? Whom should we vaccinate? Do these people really get any benefit of vaccination? We don't know. When we look at the correlation between dengue viremia and antibodies, people who had high levels of antibodies, people who had less antibodies actually showed very high levels of viral loads. People who had good amounts of antibodies showed a negative correlation with viremia. In general, suggesting that antibodies actually are a pretty good correlate of protection. If you have good amount of viral antibodies, you are probably okay with subsequent exposure to dengue. Now, what are the neutralizing antibodies in the cohort that we have established? So as you can see here, we had 149 samples in which we could assess neutralizing antibodies to all 4 serotypes. We could actually detect at least highest amount of antibodies to dengue 1 and dengue 2, and then dengue 3 antibodies were a little lower than that, and dengue 4 was the least. And when we looked at the infecting serotype, most of the people who got infected were from dengue 3 and dengue 4. It again suggests that probably the level of antibodies decide what kind of serotypes emerge during circulation in India. So we now have seen that there's multitypic antibody response in majority of dengue cases and also probably in the population, and we believe that the infecting serotype associates with a low level of antibodies, and the antibodies actually also probably drive the serotype dynamics. So now when we talk about flaviviruses, cross-neutralizing antibodies is a big deal. Everybody is worried. In Brazil, in other countries, they have shown that if you are exposed to Zika, you may have severe dengue. If you are exposed to dengue, you may have severe Zika. So what is the situation in India with the antibody levels that we are seeing? As you can see here, in most of the individuals where we could measure neutralizing antibody titers, we can see a balanced response for the 4 dengue antibodies. They do have cross-neutralizing antibodies to Zika. Lowest neutralizing antibody titers was for JEV, which is barely above background. And to some extent, we do have a cohort of subset of people who have antibodies to West Nile virus also. So when you look at a subset of patients who have antibodies only to dengue, one of the dengue serotypes, but not to any other flavivirus, so this is the average amount or the titers of antibodies as measured by neutralizing titer assays. When you look at people who have antibodies to all the flaviviruses, you can clearly see that if you have really high levels of antibodies to all 4 serotypes of dengue, you probably can also are protected from Zika and from West Nile and also maybe from JEV. But it's not clear whether a subset of these people really were infected with Zika. We don't know because we don't even have any data on the burden of Zika in the country. There could be a silent epidemic of Zika which is actually boosting dengue titers. So this is again something which is very important to understand. But the take home message is, if you probably can have a vaccine candidate that can generate a balanced response for all 4 serotypes, you may also generate response to related flaviviruses and may be protected. So it may be a platform for pan-flavivirus vaccine development. So taking this into consideration, we were then interested to see whether the circulating dengue viruses in India are any different, because the vaccine strains that are currently being developed were actually isolated about 30 years ago. So what is the current status of viruses that are circulating in India? Rahul's group here from IISc recently published that the circulating viruses in India have diverged significantly from the vaccine strains. So we now have made a repository by actually processing about 300 samples. We now have a repository of 80 clinical isolates of dengue, which we have passaged 3 times. And now from the preliminary data that we have, clearly as has been published by Rahul's group, we clearly see that all the isolates from THST actually diverge from vaccine strain, which is in green here. Same is true for dengue 2. This is the vaccine strain. These are Indian strains. These are the Indian strains, and this is the vaccine strain. Again, vaccine strains and Indian strain. So the Indian strains are actually quite different from the vaccine strains. Is it really acceptable? Can we be confident that the vaccines made from old isolates really protect us from the circulating viruses in India? So we have looked at bioinformatic analysis for B cell epitopes from the sequences that we have from virus isolates, and we do see that there are significant differences in the B cell epitopes. When we measure the neutralizing antibody titers, we do see a significant reduction in the capacity of antibodies to neutralize international isolates as compared to Indian isolates. So it suggests that actually, there must be a match with the vaccine strain, that the vaccine strain probably should be closer to the circulating viruses in India, and we have no idea whether after vaccination from an international vaccine, whether the titers would still be good enough to neutralize Indian isolates. So all this work was possible because we have an ecosystem now that we built in the last 3 years. So this is a BioSL lab which is a GLP facility accredited by NABL for all the assays that we are doing. And so anybody who is interested in collaborating can actually use this facility. So now we have an issue with the vaccine, so we know the challenges with vaccine. Can we really think about alternate approaches? So what are the alternate approaches? As we know, that secondary infection is generally more severe because the subneutralizing antibodies lead to increase in viremia, leading to higher inflammation, cytokine storm, hemorrhagic manifestations, and plasma leakage that everybody knows about secondary dengue. So when a patient comes to a clinic, a dengue patient, you have a very short window because most of the people have 7 days incubation, and when they come to see the doctor, it's usually around 4 to 5 days after fever, and most of them probably the virus would disappear in couple of days, and the disease you see is because of host factors. So we have a very short window for direct acting antivirals and probably a significant window for host directed therapies. So can we really focus on both direct acting antivirals and host directed therapy? Can we look at markers of disease progression and markers of recovery? These are some of the things that we are interested in the hospital based cohort that we are looking at. So can the virus really do anything at all to epithelial barriers, because vascular leakage is a major issue with dengue. So if you look at dengue infection of epithelial cells and measure the transepithelial electrical resistance, we don't see any effect on epithelial barriers, but the virus can come out of the basolateral surface, suggesting that the dengue virus actually doesn't do much to the barrier directly. It can come out of the basolateral surface And compared to rotavirus, which is known to disrupt epithelial barriers, dengue virus probably has no effect on epithelial barriers. So using a direct inhibitor of virus to prevent vascular leakage may not be a really good idea at later stages of the disease. So this is one of the again this is an experiment we did with endothelial cells, where we have dengue 1 which can infect endothelial cells very well. Here again virus peaks 24 hours, 48 hours, you do see peak titers, and then when you look at the basolateral titers, you can measure basolateral viral titers in 2D system, and no effect on endothelial barrier. The effect you see is mostly because of cytotoxicity at very late stages. So when we look at serum factors from severe dengue, this is the you take serum factors from other febrile illness, dengue infection or severe dengue, you can clearly see that severe dengue samples actually mediate barrier disruption, and they lead to leakage of endothelial barriers, suggesting that it's the factors present in the serum that cause vascular leakage, not the virus directly. So again, this is shown here for Western blots with a subset of samples where tight junction and adherence junction proteins are degraded upon addition on endothelial cells. This is quantitation of this blot, suggesting that the tight junction and adherence junctions are altered because of serum factors from severe dengue patients. So we have measured a number of analytes over the last few years. We show that reduction in interferons and increase in IL-6 and IL-8 actually associate with severe dengue. When we look at markers that can show association with recovery from severe dengue, we see that IL-8 and IL-8 reduction actually associates with recovery from severe dengue. We have done now quantitative proteomics, taking convalescent samples, dengue, mild dengue samples, severe dengue with fluid leak or without fluid leak, and we did eye-track recently to look for proteins that are differentially regulated in severe dengue samples, and we have identified 54 proteins showing differential regulation in dengue, and out of this, when we do a network analysis, we see two pathways emerge that associate with severe dengue, namely apolipoproteins and complement cascade proteins. These are the two major clusters. We have validated this data now by multiple reaction monitoring using quantitative mass spectrometry, and we can clearly see that some of these proteins, the galectin-3 binding protein, apolipoproteins, and retinol binding proteins are all downregulated in severe dengue. We validated this in a subset of cohorts where we measured the retinol levels in severe dengue, and we show that all severe dengue samples actually have very low levels of retinol and retinol binding proteins. So this is a new marker that we have identified for severe dengue now, and we validated the downregulation of apolipoproteins in a subset of cohort of people, and as you can see here that in pretty much all the severe dengue samples, apolipoprotein levels are down, clearly indicating that dyslipidemia is a characteristic feature of severe dengue, and perhaps this is mediated because of extensive liver damage we see in severe dengue patients. Therefore, one way to intervene in severe dengue is to really address this liver damage. So we have come up with a new set of markers that can be used as used for validation of severe dengue as biomarkers, and there is enough data now to suggest that HDLs, actually the high density lipoproteins which are regulated by apolipoproteins, they regulate inflammation via complement cascade, and some of the other markers also have been shown to play a role in acute phase responses in other dengue infections. Interestingly, the apolipoprotein mimics have recently been shown to actually help people to recover from severe dengue patients, severe COVID-19 patients. So perhaps this is something which clinicians should consider as a pilot scale studies for intervention in severe dengue patients. These are again some of the papers suggesting that the markers that we have identified perhaps will also have relevance in severe dengue. So to summarize, downregulation of interferon response and enhanced inflammatory markers associate with severe dengue, and we have observed that in severe dengue there is dysregulation of complement pathway and lipid homeostasis and retinol transport, and we believe that liver functions are compromised in severe dengue, and perhaps the best way to actually treat dengue patients is to really try to prevent damage to liver and somehow help liver recover. Maybe direct acting antivirals may play some role if there is really tissue resident virus replication, which is difficult to measure in a patient. So we are currently now with our cohorts, we are validating some of these biomarkers in longitudinal cohort studies, and we believe that we can actually use some of these markers for interventional trials. So these are all the people. So we have a separate team doing all the clinical studies. So BioCell lab is extensively involved in vaccine development studies and academic research lab. Some of the data was generated by my earlier PhD students, Rinki, Meenakshi and Aleksha. Balwant, Pooja and Naseem have contributed to proteomics work and also the sequencing work. So thank you very much. These are, so I have two posters, poster 2 and 26, I believe. Two of my colleagues are presenting, and acknowledgements to ICMR team, AIMS New Delhi. There have been constant support for all the clinical samples. Anmol has been a long-term collaborator for immunology and also clinical materials. ESI, IGIB for sequencing. Chitra is helping us with bioinformatics for sequencing data. Proteomics, again, my colleagues at THSTA and also Nirpendra now at InSTEM. Funding by DBT, intramural funds, India Alliance and also BIRAC. Thank you.
Thank you so much for the pretty nice talk, sir. Maybe I missed this thing out. I have a question. Why dengue vaccines have low efficacy in case of children?
So it is not just in case of children. Dengue vaccines have low efficacy in people who are not exposed to dengue. So as I showed, most people in endemic countries actually will be positive for dengue by the time they are about 15-16 years, or maybe most adults would be positive. So when you have a seronegative individual, you give a vaccine, so that elicits some antibodies, but that antibody may not protect against your primary infection, because those antibodies may subneutralize your virus, may not be optimal for the virus that is from a different serotype.
Yeah, go ahead.
Regarding ED, I didn't hear anything right. So I don't think you dwelt on ADE much. So do you think that's something that needs to be considered irrespective of the vaccine? Purely from the immunological point of view, the evolving of ADE specific response. What is your comment?
So the problems with vaccine efficacy is because of AD. So the increased disease you see in seronegative children who got vaccinated is speculated to be because of AD. So that's what we want to do now, because this is something which we have started and we are looking at. We have now a spectrum of people where you have very high levels of antibodies, very low levels of antibodies. So we want to see now, where does AD start? What is really, is there a cutoff for titer which actually says below this you start getting AD?
And just last follow-up, pediatric versus healthy versus adult. Do you see any modulation there in terms of how to respond to that?
Well, our cohorts are all, at least the hospital-based cohort has been a pediatric cohort, because our collaborator is a pediatrician. So we have just started really collecting adult samples this year, from this season onwards. Perhaps I would be able to comment more maybe in the next meeting. Yeah.
So that means that dengue vaccine would require additional parameter measuring the antibodies for ADE, and then give or not give.
It is, but it is something which is an investment before you start deploying vaccines. So I'm discussing with ICMR. We have given this message. They said they will look into all of this. I don't know how much effort is going in this direction.
Sir, Gajanan is actually trying to do an assay.
Yeah.
Distinguishes ADE from neutralizing antibodies.
No, not only that. Before you start your phase 3 trial, perhaps you should know the serostatus of people and.
Yeah. So I think such screening is already going.
I hope so. Yeah, that's what I was told, that they are actually looking into all these aspects. Shashank has a question.
Nice work, Guru. So it was really relevant to the population. These are attenuated vaccines, right?
Yes, all three are attenuated vaccines.
Viral vaccines. So they are supposed to elicit T-cell responses, and that is the mechanism of action for efficacy at all. But you're not got into that at all. You just.
Not only me, nobody actually bothered about T-cell responses with dengue vaccine studies. So in general, people have accepted that if we measure neutralizing antibody titers, that's good enough. But now there is actually more and more interest in looking at T-cell response, and we are also In BioCell Lab, we have now established all the T cell studies. So the cohort that we are conducting as of now will also have T cell data coming soon.
So this data, if at all it says anything, it's a lot of giving a lot of cautionary signals that it's going to be dangerous, because we never know how much the antibodies are in a person before and after vaccines, and it might actually enhance the disease rather than the AD.
Yes, so yes, Rao. Thank you.
I was just curious about, the yellow fever virus vaccine, another integrated vaccine that was made in Japan. So I wanted to hear your comment about why you think dengue immunity vaccines are so poorly while yellow fever virus.
See, again, speculation is that the T cell responses might also be specific, but I don't know how divergent the non-structural proteins are between yellow fever and dengue. There may be some amount of divergence, but at least for dengue vaccine, that's the theory that because you have yellow fever T cell responses, maybe that's a problem. You should have dengue non-structural proteins to get a better response. If you look at Takeda's efficacy, dengue to efficacy is really 80-85%, which is not the case for dengue vaccine. That at least suggests that having dengue non-structural proteins might be better, because you may get both B cell and T cell response against dengue. But the problem of AD and original antigenic sin is for both B cell and T cell. So we really have no idea what will happen with the phase 3 trials here.
Okay, last question from Japan. Quick question, thanks to the group. So this issue of only seropositive people responding to the vaccine, can it be solved by boosting or changing vaccine design?
We have only one vaccine, so even that is not yet in trial in India. Problem is with the tetravalent requirement. So you need to have 4 vaccines. So therefore, even if you change your formulation, it requires extensive validation to show that it is at least as good as what's already there. And I think at least in India, ICMR is very cautious about the other 2 vaccines, and probably there is some kind of faith in the NIH vaccine. You do boost your antibody titers, because people who got vaccine, the seropositive people, were protected for probably the remaining period of their lives.
No, only seropositive people responding.
They are 2 or 3 doses. No, in seropositive people, they are only measuring 4-fold increase in titers. So we don't even know what is optimal response. Here the efficacy is only measure of virologically confirmed cases. So you don't see virologically, RT-PCR positive dengue patient, your efficacy is decided only based on that.
Thank you, Guru. I think there are many more questions, but if we deal with those questions during the tea time.
Okay, thank you, sir.
Please move to the next talk. He is an associate professor at Shivnagari University in Delhi, NCR. His lab uses extensive immunogenicity screens and reverse genetics approach to understand the nuances of virus host interaction, biology of hepatitis virus. He is going to talk about distinct metabolic requirements of hepatitis C virus activity.
Thank you, Dr. Rathi. Good evening, everyone. Under normal physiological conditions, cell utilize extracellular glucose that undergo glycolytic cycle, glycolysis, followed by TCA cycle and oxidative phosphorylation, preferably to produce ATP. G6P then branches out into PPP to produce nucleotides. On the other hand, TCA cycle intermediates serve as substrates for the production of amino acid synthesis. This is in normal conditions. But when in case of virus infected cells or in case of cancer cells, there will be increased uptake of glucose and glutamine from extracellular sources. Then there will be an increase in nucleic acid biosynthesis, amino acid biosynthesis, and fatty acid biosynthesis. And then if there are shortage of glycolytic intermediates or depletion of pyruvate, then there will be two phenomena that will occur in the cells. One is glutamine anaplerosis or aspartate anaplerosis. This glutamine anaplerosis then join TCA cycle in the form of alpha Kg, thereby the TCA cycle sustains and thereby it supports amino acid synthesis and fatty acid synthesis. This is the case of when there is a great demand for energy and macromolecule for viral replication. Right. Viruses have undergone evolutionary adaptations to utilize extracellular carbon sources in order to which enabled them to harvest specific cellular substrates which are required for virus successful replication. For example, HCV and dengue virus increase glucose uptake, and then glucose 6-phosphate goes into PPP, that's where it tunes up utilization of glucose. On the other hand, dengue virus and cytomegalovirus increase fatty acid biosynthesis. HSV and vaccinovirus were found to tune glutaminolysis for their better replication. So in this study, we asked the questions, what are the carbon sources and metabolic requirements for HEV replication? And then what are the specific or distinct virus-induced changes that will happen in host? For this, we need obviously a reliable cell culture system, but there is no reliable cell culture system for HEV. Recently, we developed a molecular method to synthesize full-length mRNA molecules of HEV genome, which contains random mutations in those genomes. Using this, we transfected into hepatomer cells, which showed these RNA libraries a transient virus replication on day 6. But this does not happen in case of wild type. We think that the mutations that are randomly introduced using this method are possibly cooperative interactions among the proteins or between proteins and host proteins that might have increased virus replication. So how to synthesize these? So we have cDNA clone of HEV full genome that we subjected to error-prone PCR. By altering the NTP mix concentration, we can introduce random mutations in the full genome level. Therefore, each genome already would have either at least one mutation in it. So essentially, by transfecting these RNA libraries into hepatomer cells, you can synthesize literally thousands of viral variants with varying replicative capacity. So we transfected these into hepatomer cells. So first we depleted carbon source of those hepatomer cells by incubating them in 10% dialyzed sera without glucose and glutamine. After 16 hours, we transfected with viral RNAs. Post 8 hours post transfection, we replaced the media with replete. That means it contains glucose and glutamine, or either glucose depleted or glutamine depleted media. And then we collected cells as well as supernatant at 8 hours, 36 hours and 72 hours post transfection. At 36 hours post transfection, the amount of extracellular carbon had decreased quite below 50%. That in contrast to wild type. Then the viral RNA levels, intracellular viral RNA levels, and double-strand RNA intermediate levels, which is a replicative intermediate of HEV replication cycle, were high in case of mutant RNA transfected cells, which is possibly the replicative ensemble is undergoing replication. Then we depleted either carbon source, either glucose or glutamine, to see if there is any preference by the cells transfected with mutant RNA. What we saw is at 72 hours post transfection, there was a great decrease in both glucose as well as glutamine, which shows that virus prefers both glucose as well as glutamine for its replication. When we looked at intracellular RNA levels, replete incubated cells had higher levels of viral RNA, and there is a significant difference in terms of viral RNA when we looked at either carbon source availability. So that shows that virus needs both glucose as well as glutamine for its replication. So this has prompted us to look at what possible changes might have happened intracellular. We did mass spec and then followed by analysis. We specifically performed joint metabolite pathway analysis, that means both proteins as well as metabolites, which we have identified total 16 significantly altered metabolites. Among those, the top 3 were pyruvate metabolism, glycolysis or gluconeogenesis, TCA cycle, and amino acid metabolism is highly dysregulated, including glutathione metabolism and purine metabolism. So this is one of the pathways I'm showing among those pathways because we are interested in TCA cycle and glycolytic cycle, including glutaminolysis. So upon the analysis, what we saw is there is significant increase in hexokinase 2. Increase in hexokinase 2 is a marker for cellular infection in terms of metabolism. And then PGI, TPI, enolase and pyruvate kinase significantly decreased. In the TCA cycle, acetyl-CoA levels transiently increased but non-significant. MDH and isocitrate dehydrogenase were significantly down. On the other hand, metabolites succinate and malate had increased significantly. Most importantly, in the glutaminolysis, the 2 enzymes responsible for the lysis of glutamine were not detectable in our data. But in contrast, enzymes that are responsible for converting alpha ketoglutarate to glutamine were significantly downregulated. So we thought to inhibit if there is a role for glutaminolysis or glucose indeed by using inhibitors. We use 3-BP, 3-bromopyruvate that specifically inhibits hexokinase 2 that depletes glycolytic intermediates including pyruvic acid. And then BPTS specifically inhibits GLS, therefore it prevents joining of glutamine into the TCA cycle in the form of alpha Kg by impeding its conversion. So when we added these inhibitors, the intracellular RNA in case of 3-BP was completely down, almost 3 log lower, but if you see this, when we used both 3-BP and BPTES, the levels of RNA were almost similar in these cells. So that tells that possibly glutaminolysis has no role in virus replication. On the other hand, when we looked at succinate levels were also almost 66% in case of 3-BP, and when we used both the inhibitors, succinate levels were similar in case of these 2 cells. So there could be alternate substrates that might be replenishing the TCA cycle. So when we supplemented the media with glutamate, then there was almost 1 log decrease in viral RNA. However, most importantly, the succinate level was little decrease in its levels, which shows that TCA cycle is not replenished because of the glutamine, because of other substrates can also replenish the TCA cycle. This shows that HIV replication is not supported by glutaminolysis. And then this had given us that glucose and glutamine are critical substrates of hexosamine biosynthetic pathway. So we looked at other glutamine associated pathways. We pinpointed at hexosamine biosynthesis pathway, which is responsible for N-glycosylation and O-glycosylation. And we also shown in the literature that glutamine activates HBP in tumor cells. So we then looked at the HBP pathway, what are the metabolites and the enzymes could have been dysregulated. This is the schematic of HBP. So several metabolites and enzymes of HBP were transiently are upregulated but insignificant except glutamine and HSP70. So here F6P, glucosamine N6-phosphate, then acetyl-CoA, and then UDP, all are upregulated but insignificant. Most importantly, the rate-limiting enzyme GFAT that takes up F6P and condensates with glutamine, and glutamine gives amino group to F6P that to convert to glucosamine 6-phosphate. That was detectable in our data. So eventually this pathway leads to synthesis of HSP proteins. That is what been proposed in literature. When we looked at HSPs in our mass spec data, there were several isoforms of HSP70. HSP 70A1B here, most importantly, was upregulated. And then when we saw it at various ISGs, most importantly here, MX1, OS1, ISG15, they were significantly upregulated. So we do not know whether the virus induced stress or inflammation induced stress had led to increase in HSP70. That we do not know. So if it is the HBP pathway that supports virus replication, then mannose should be supporting virus replication, because mannose joins as a feeder pathway with the glycolytic pathway. So we incubated cells along with mannose, including mannose and glutamine. So mannose converts into mannose 6-phosphate, then mannose 6-phosphate joins as F6P. If that is true, then the virus replication should be similar. So what we saw is a little down in terms of viral RNA when we fed the cells with mannose along with glutamine. So on the other hand, when we looked at HSP70 expression, there was a slight decrease in case of mutant cells, but not significantly. So if we inhibit glycolysis pathway on HSP70, if that abrogate HUV replication, that is the question that next we asked. So for this, what we did is, again, we inhibited glycolytic pathway with 3BP, and then HSP70 Kefron activity with JG98 to see if there is any effect. So when we inhibited with both the inhibitors, 3BP and JG98, there was significant cell death, which means as there is no Kefron function of HSPs, there was a lot of cell death when we transfected the cells with unincubated in the replete media. On the other hand, the viral RNA significantly decreased, almost 4 log down when you compare to replete cells. So this is HSP70 and HSP90, which possibly have reciprocal expression, which I do not know the reason for this, but we have seen this observation in our experimental data. Then we inhibited these HBP and then HSP70 activity. So we never able to detect ORF2 protein intracellular because none of the antibodies available do not work. So we have no choice, but we went up to do some Western blots on the extracellular virus. So when we looked at extracellular virus, replete cells producing high amounts of viral RNA, whereas 3BP and JG98 treated cells were producing less amounts of RNA. We concentrated huge amount of supernatant, then we did Western blot on the supernatants, concentrated supernatants. Then we probed with 5 HIV patient sera that is pooled up. Then we saw some detection of ORF2 in the replete cells, but this ORF2 is absent in case of cells treated with 3BP and JG98. Then we also treated the cells with DON, which is an inhibitor of GFAT, that is a rate limiting enzyme, thinking that it will decrease HSP70 production. That had again reduced the levels of HEV RNA extracellular and also did not produce much amounts of ORF2 in the supernatants. We parallelly were interested to see at the same time what is happening in this in vitro data. Can we do something with HEV patient samples? For this we did temporal analysis of serum metabolites and lipids, in acute hepatitis E patients. We had total 65 samples from 14 patients and 25 samples from healthy controls, 5 healthy controls. Samples were collected sequentially after day 3 till day 18, every 3 or 4 days interval from these patients. Almost all patients were negative at the end of time point, at the last time point of collection, and all patients were male because they were collected at Army Hospital. We did mass spec analysis, and then what we saw is HIV samples, all 14 samples data clearly segregated out from healthy control population. And then in the volcano plots, we saw 93 metabolites were down-regulated and 70 were up in the sera of hepatitis E patients, acutely infected. In case of lipids, 165 were down and then 60 were up. And this is the heat map analysis, which was done in a 3 to 5, 6 to 8, 9 to 12 day analysis up to 15 to 8 in 5 classes. As because of there is a differential abundance in metabolites and lipids, there was clear segregation of samples in this data, which shows that clear dysregulation of metabolites and lipids. So these are some of the pathways were dysregulated. If you recall that purine metabolism and glycolysis were one of the topmost pathways dysregulated in case of our in vitro data. We also saw amino acid dysregulation, and all these blue highlighted pathways were indeed we saw in the in vitro data. So glucose 6-phosphate, fructose 1,6-phosphate was high level in case of HIV infected patients. Malate increased and succinate was also increased in case of HIV infected patients, which is correlating with our in vitro data. Ribose has increased, ribose 5-phosphate was increased in the sera, and then there was changes in the pyrimidine and purine metabolism, and there was dysregulation in case of aromatic amino acids, where homogenticate and kynurenic acid were more in healthy controls. There was dysregulation in case of alanine, glutamine, and threonine metabolism. Most importantly, all of bile acid synthesis metabolites, taurocholic acid, glycolic acid, and other bile acids were significantly upregulated. So there are 2 prostaglandins which are down-regulated, prostaglandin D2 and B2, which were down in case of HIV patients, possibly showing dampened immune responses. Most importantly, in these patients, cofactor FAD and coenzymes pantothenic acid and pyridoxal were upregulated, which shows that there is an increased metabolic activity in carbohydrate, lipid, and protein metabolism. Then acylcarnitines increased in case of HIV patients, as well as triglycerols and oxidized triglycerols, which shows that there is an increased beta oxidation in HIV infected patients. Lastly, there are many glycerophospholipids which their expression levels were decreased for many of these. When we looked at the saturation of these lipids, many of these glycerophospholipids were unsaturated. You see, they have 6 double bonds, 5, 6, 5, 5 here, which shows that HIV has really tweaked the membrane fluidity in the patients. All right. With this, I would like to acknowledge my collaborators, Professor Rakesh Agarwal and Amit Goyal, who had given us valuable samples to do the second part of this talk. And then I would like to thank Yashwant and Amit from THSTI, who had helped us doing mass spec. Much of the data has been contributed by Shaheen. Recently she submitted her thesis. Shalini had contributed to some extent. Pooja is presenting a poster today in this event. Lastly, I would like to thank Dr. Das, Dr. Shashank, and Keshav for giving this opportunity to present our recent work in this great platform. Thank you.
related to the HSP-17. If you inhibit HSP-17, my understanding you will activate UPR and that will automatically lead to translational setup. So you don't expect to see wire to translational code.
Yes.
But then how do you think
So when we inhibited with JG98 the activity of HSP70, there was increased cell death, which I agree. Might be contributed due to the UPR, which I agree. That could be the reason. There is some death because of the inhibition of. Don't know whether it is dependent on the metabolites. I do not know, but as far as I see, it is linked up with glutamine at least.
Nice talking to you. So the markers that you show, are they like generic markers of liver inflammation? Because what you measure is in the blood. Right?
No, no,
in any way with, if there is a viral load estimation possible at all in your case.
So markers, you mean to say the metabolites and lipids in the second part. So we have not done the correlation whether virus titers will have any effect on the metabolites, because many studies have not found it. So we just avoided to do the analysis of viral titers versus metabolites amount, let's say. We have not done that. But we don't find any correlation with it. And that too, we are looking at serum, right? It's not liver. Everything happens in the liver. So it does not reflect what is exactly happening in the liver cells. What we analyze is the sera, which is a indirect picture of what is happening in the liver, but not a direct picture.
That's what I was thinking.
Right.
This is a
Generic way of
liver inflammation which can
happen in any hepatitis virus. Even in case of liver steatosis, it may be happening. Yeah. We try to include HBV data in this, but we ran the mass spec in a different run, and then M by Z values never correlated, so we could not include HBV data.
So your cell line data and also patient data seems to suggest that glycolysis is important.
Right.
But you also do see accumulation of succinate and malate. So is it that glycolytic pyruvate is actually deriving this cycle because glutamine doesn't seem right? Do you see any change in the mitochondrial respiration?
So we did not touch upon mitochondrial respiration. Actually, glutaminolysis happens in mitochondria, right? So pyruvate is the one that eventually would join the TCA cycle via acetyl-CoA. So yeah, that will be the main thing. Yeah. So not glutaminolysis, but glucose and glutamine, but not with glutaminolysis. Some other associated pathway, that is HBP, possibly has a role. No, we have not done that. No, we have not studied the flux. Only we have the mass spec data. That's all.
I was wondering whether it would have been ideal to keep one control of patients who have general liver inflammation other than any viral infection.
We sort of did a mistake when we ran analyzing the samples. We are supposed to include at least HBV acute patients, serum samples, but we could not include that. But later we included that when we got the samples. When we saw the M by Z data, it's not correlating at all. So the lesson we learned is all the samples should be run in the same assay. No matter the control or a different set of samples to study the case controls, no matter what, they should be run in the same assay.
Right, then we conclude this session by thanking all the four speakers for their talk. Please join me in
Thank you, Professor Vrati and Professor Banerjee, for chairing the session. Kindly proceed for the coffee break and poster session, and take this opportunity to network and engage with your fellow young researchers, and we'll assemble back at 6. Thank you.
Sure. You are sweetie?
Yeah.
Okay, sorry. Just try to remove this.
I want to see it.
Yeah.
Because I made some changes.
Sure.
You delete the slide, right? After the talk.
Yeah, I do it. I will recover it. It's already too much, so I've reduced the session to all of us.
No, yeah.
This one, right?
Sorry. Yes. I'll just check it here.
This is Nexus session 4.
Hello. Hello. Yes, Sita, I'm in a molecular virology conference, so I will be back on Monday. Oh, okay, you're there. Okay, we can meet. Oh, you are here. So, I'm just uploading my slides. Okay, I will come to the coffee break. Okay. 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 Sure. Okay.
Screening
Second. Keshav. My talk is tomorrow. Yeah, you can. If you want, you can give it now. If not, then pen drive, no? Yeah. But I don't have pen drive. I have a pen drive. I can give it to you. No, that's fine. Actually, morning also it's not in the first session. It is in the first session. It's first session. First speaker. It's better you can give it before you leave today. But or else I can come up and which can we send yeah, through mail also. That's fine. Yeah, that's fine. In the molecular virology email itself, you can just check. Molecular virology 2023. This one? Yeah. Can view, can comment, can edit, assistant. It looks like sent. Okay, sure. I'll just check. Yeah. This one, right? Arup Banerjee. Yeah. I'll take it. It's okay. Just need to see if everything is okay. Yeah, tomorrow when you before the session, just let me know. I'll be able to do that. Because anyways, it will start somewhere around 9, same today. So around 8:30, everyone will be there, so you can just have a look tomorrow. Okay. Yeah. Keshav sir का क्या सीन है? You have to begin. are going for 400 MB. That's a maximum so far.
Is it? I have 70 MB.
That should be enough, sir.
But only thing is just make sure once it is done, delete it.
Yeah, yeah. That's what we plan to do for all.
Yeah. Just
I'll
we
just first
haven't
copy it.
prepared anything. Too time for us to prepare.
This one. Why this one?
It has virus. Do you have pen drive which can work?
Yeah, sure.
Okay. Can I remove this?
Yes.
I'll open my laptop. Sure.
This one, right?
Yeah. Yeah. Can I just try
Let me
wherever?
open it here and then I'll try to.
Yeah. Because since it's a Mac one, I just want to.
Sure.
Yeah, I can
see
see
it over there also.
Okay.
Just
No.
yeah.
There is one video. Let's see.
The video will not automatically play.
Yeah, I think it's correct.
But if you have an animation, that will be.
Yeah, I put animation. There is one video that. This one. Yeah. So they're going to switch off this light, right?
Yeah, they will. So once that will be clear. Yeah.
Okay. Yeah, that's great.
Yes.
I did escape. You can put it at first light and leave it.
Yeah. So yours is first. Okay, that's right.
Yeah, first light. That's it. Okay, put molecular virology meeting. Okay. Yeah. Thanks, Rajesh.
Okay, sir. Thank you.
people came and asked questions. I just left my bag there.
Sure. Not too bad.
So how far are you from there?
I stay close to in Sanjay, not very far from here, like 20 minutes. I moved here from behind 9 months. I used to stay there. I used to, now I don't, thankfully.
So this is Sanjay Nagar, sir?
Yeah, it's very close. It's like Ramaniya Hospital. Then you just go on that straight 80 people, you do the place. At the end of that road, very close to the place.
My brother stays in Virology.
Okay. Nice. So,
Sanger sequencing, I'll just update from our side. So Sanger sequencing, almost everything is done.
Okay.
So
You
that-
sent me something yesterday also.
So Virology is there, he's comparing with it. So he just sent me the, I told him because 5 minutes for something given to sequencing person. After it came back, I told him to label this so that when you see at least you know what is it. And that's what he yesterday sent me and will not discuss. There is maybe another large round of sequencing runs which are going on. Should be done maybe this week.
Anyway, right now we are not looking at the UTRs.
Our analysis we don't need.
But we need some for application fitness, the UTRs are important. Like obviously because the NIH vaccines are, they are application fitness or they are application fitness.
Has a remission and we are actually planning to generate the criteria in case the GPs actually application.
Oh yeah, okay. Oh nice.
For that we need the sequence. That's why I am particular. So here if we measure some difference then we can actually go back and check the sequence. So what we are halfway through now is that we need to look for the resources to shortlisted for in vitro kinetics and so forth.
That is a big course.
In like a couple of hours, we do much better than
I see.
but I don't want to celebrate anything they do it twice and we do it second. I had one doubt in that day because I didn't run our marketing. And you see differences and I wanted to be very critical. I said what if the marketing in one case is planned from MRI, in one case is for GI. So I was wondering whether we should measure the original rates and sequencing before going on to that. So if we do that, and if we say we are 100,000 copies, where there is your 100,000 copies may not have nutrition. There is a problem there also.
But it's better. Otherwise, actually, the way we do is to always do plaques.
That's what we are doing.
So that is the best you can do.
As of now, we have done black assays and we have done infections. I'm just curious to see whether there will be any differences in the amount of effect.
But if you do multiple points, you will get the same thing. And you will also
We will see. Right now we are doing one and more with different for this.
How are you doing? So, this is a list of
using that, sometimes they use it. But I made a list of changes for her. What I realized which is very weird thing. The reason why they were not willing to give qualified positions is because they used the X27 among the samples as the reference, which is not the reference to institute. This then they say in the literature, if you look at it, and they say there is a E1 something 26 VA and things like that, which we were not seeing. And I was like, oh, why not seeing these changes? So that nomenclature is from the H27, which is not a reference to. So I was not getting any actually reference positions. So then I saw, and so this has fixed now. So but the way it will look. So then I understood why people are not willing to commit to the mutation list. So because what you need to then do is say that the amino acid position. So even E2 in the website was 339 to 732. The sequence is identical, but the annotation is not the same. So position 1 is not the same as position 2. So yeah, so unique which is X27, X27 is what they are using to call the nomenclature. Anyway, this is important thing that I learned about checking with you. And so this is how I plan to show. So we will have our NC whatever, we will have ISO, and then what is the position in that reference studio, and what is it in the sequences from the study. We should get that number of sequences that have been within the study, and whether it's present in that genotype or not. So whether it's a cosmopolitan specific thing, or whether it's really specific, or they need to have it, or they need to have it, that can be made from also. I think that this is how we should present it. In earlier we used to have this IISc and those lines, that's what we used to do before, but I think it's good to present it as a template. So we are only going to show not like the ones that only not amino acid right now. We should make a table of the synonymous ones, but initially it will be amino acid. Yeah. So for this thing, this EPRN vaccine comparison, I think it is good to do a table like this, say vaccine. It's a bit of work, but I'll do one, and I'll send it to you. If you think you have to do it, you can try, and I will verify. And that's okay. Same with the mutation list for various regions. That's it. So this is what I plan to do. I will give you the time which will have I got good. Because for the halogenetic posting, this is so showing this. He said E is determined in halogenetic. actually, I'm also with Dr.'s company and working. They are very good. Which is not in Bangalore. They have a remote working company and helping their R&D using UAPC processes and things. So they are doing wonderful people and they're working with them for doing the resistance discovery. They're also running with the genes. So I would see how much the development is like. So right now I'm working with them 8 hours a day. I don't know her. Do you know her? So you know her to what my colleagues do. I see. I see. I think so. It looks like that, but the reality is the feeling is okay, but that's what I told you, 3 months, and I see if they're doing it. If they do it, good. If they don't do it, I don't know. So they get this immunity actually because that is the patient. And art so they are I told them I can't join now, so 3 days a week, each test 1 week. Yeah, it allows me to do everything I want to do actually. Company You know who does the cross-reactive antibodies?
So basically,
Yeah, we don't. Yeah. So that is going to depend on your secondary. What does it mean? That is a reasonable hypothesis, right? With NIH. Yeah, you should. Yeah. And this money about that's yeah, okay, so I'll tell you, I'll tell you determine the rate which never published. If you just put NS1 learning and you have London module, just because it's a huge vulnerability. What if it is permeable? That's what you're vulnerable. Yes. Yeah. Small amount of transfer can be permeable. Large amount should be more permeable. But maybe there's a threshold that you know, but you didn't see anything. No,
everybody was just mild, nobody had any plastic.
Oh, I see. Everybody.
So that's what we like. This one might be acting as a bar, this one antibodies might be this one per se what it does, I don't know.
I see. Because they have these monoclonal presence studies from the predicting mass, which Nora is like really champion.
I know. It's the only group which pushes for this election. So maybe
if somebody here wanted to do, Rama wanted to do it, they let him get his hand. And he wants to do so many things.
So,
I mean, if you can do a nice get for population, not just for somebody, and they volunteer for 5 years. But at least in that, if you don't count on antibody states, you can.
It will be visible over there.
अच्छा उधर दिख इधर नहीं दिखेगा?
No, because वो ऑनलाइन ही है.
अच्छा ये पॉइंटर है.
Yeah.
और इससे मूव होगा?
Yes.
नहीं मूव होगा.
This is again this. Now try. वो छोटी टॉकें ख़त्म हो जाएगी जल्दी से. बकवास करना पड़ेगा. ठीक है चल.
looks like they are high titer.
We know that there are some plant flagging, right? There are some amazing plant flagging. 60, right? That is the antibody that is
So we don't know the polyclonal response. Now
right.
is this something which
Oh, I
happens when you
see.
are infected and for 3 months, 4 months you are protected against all the viruses and slowly With this I have control. These samples don't neutralize which are these samples are very good. So I have 6-7 now trials out of this where the subset of samples only neutralizes dengue, JE, they don't neutralize the one subset has antibodies to etc. but they don't neutralize the other. So it is not really a technical really what is actually happening.
And within this also, it's not like all of them are high for.
So
There is variability.
if you look at this sample, it does not neutralize any food. It has only dengue. It was around 89 now. At 89, it was 88. I was 12, 13, 11, 7. So this we are now done. This is just to show equal distribution of only 1, 2, 3, 4. So we had committed to make 80 So we have been Indian estimate of this.
NIH.
You can see that.
Interesting. For dengue too, are you seeing that for dengue too as well?
Because dengue titer is so high, so if you have high enough titer, the problem is realized.
But dengue too actually
is closer to this. So that is also one.
I'll do that properly, but I was actually very surprised that there are such. I didn't expect E to be. There were very few. Amino acids.
So this is what others
Okay, so you're hopeless. But same here. This only I did for like the 1 year that I was in the UK. Every time putting in and measuring this. But NS1 alone was able to.
But you see, this is what I'm saying. You have epithelial cells infected with dengue virus. This one is coming out, this one is expressed. Thing happens with that. 3 days, 72 hours cells are infected. Here, measurement in epithelial cells and the barrier. But the virus is coming out of this virus. So there is a response.
That does not. Yeah.
So this is epithelial cells and this is endothelial cells.
Endothelial cells also.
Endothelial cells infected with dengue virus years 24 hours to last 26. By 96 hours, 6 hours. Virus is coming out of the body, so that it's so I am really not I am not
Nothing exogenous.
not invest my money on this. But I don't want to create a controversy. My management will go to inquiry, so I don't want to say anything about it. So this made us to say that maybe direct acting antivirus may be useful or may not be useful. We will focus on host factors. So when you add samples from severe dengue patients, you will see that leakage, increased leakage.
So there is something
in the serum,
but it is not being produced by the virus in cell culture.
So we show that tight inches are response to serious problem with liver.
Which we know clinically.
So all this is happening because of impairment in liver functions. If you want to take care of the liver, liver and make sure liver will be fixed. That's what we see. We have a patient show that the liver functions are very so the screens are important in liver function. Giving you multiple pointers for so we have the chemical angle, we have something like biochemical just to see we have proteins which we should actually think of mechanism of action. This is what we students are interested in. This is my everything is after
Makes sense. It's a contained project.
So this is biosimilarity, which does all the vaccine assays. They are basically
Okay.
yes, so
I
came to talk with
No, because he can, she can find this.
they have taken, I have given the samples have been daily.
Okay,
So this has been done. I will ask this. Yesterday also I came book. It's very difficult to reach her. I told her we have written the right in the mail and ask her. She didn't respond to email. Otherwise she has a child. So she comes at maybe 8:30 and she goes back to the and she also has to report to the police. So night from 11 to 1 also lot of meetings will happen. So I don't leave in the morning. But she responds, she replies at 5:30 in the morning, 6 in the morning.
I will message you once I'm done.
You should come this week, so I'll update her.
This is yours, right? Yes. Just a second. Let me check which one. This is Keshav says. This one, right?
Hello.
This is Shweta.
Oh, hi Shweta.
She's in GNC now. And there is NCIDR, but from MBE.
Yes.
Flavy structure and flavy mosquito stuff.
Ah, okay.
RNA.
There's a lot of flavy here.
RNA biology in mosquito.
Okay.
So she is from Sumitras.
Okay, okay.
Then she went to Peter Sarno's lab in Stanford.
And so from there you're back here. Okay. Nice to meet you.
I think she took a long time to decide if she wanted to come back. Something like that.
Yes.
But now that you're here, they support system.
Okay. Should be fine.
Yeah.
go and call these people? Should I go and ask them? मैं बोल देता हूँ. Should I call them?
Welcome back, everyone. I trust you had an energizing break filled with stimulating conversation and interesting discoveries at the poster session. Our virology conference journey continues, and the knowledge exchange promises to be even more enriching in the upcoming session. We have had the chance to access the fascinating research presented during the poster session, showcasing the diverse and innovative work in the field of virology. Now let's transition back to our scheduled talks. Our next session will unfold exciting discussions on viral evolution, offering us fresh perspectives and insights into the ever-changing landscapes of viruses. The session will be chaired by Professor Uday Ranga and Professor Shamitro Das. Professor Ranga has been associated with Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, since 1997 and has been working with HIV for 26 years now. He is also a member of the Trainee Selection Panel, AIDS International Training and Research Program of the Albert Einstein School of Medicine, New York. His lab's focus is on the molecular evolution and biology of HIV-1 subtypes, especially HIV-1 subtype C, immunoactivation of HIV-AIDS, and HIV broadly neutralizing antibodies. They are also working on DNA vaccine engineering and optimization. And Professor Das needs no introduction. Our lab works with so I am one of his students, and our lab works with the role of various host factors in different RNA virus infections. So now I would like to request them to begin the session. Thank you.
Good evening to you all. We have four presentations in this session. I invite first Professor Keshav. His laboratory focused on intracellular RNA sensitive mechanisms and activation of functional higher order complexes and how they orchestrate cell information and virus host interactions. This group also explored the evolution of immune evasion traits in RNA viruses through a combination of protein engineering and structure-based rational approaches.
Thank you, Professor Uday. Can you able to hear me? Yes. Now? Better, okay. Very good evening to you all, and thanks for keeping your energy at this time. I hear you better. Yeah. It's okay. Can you hear me now? Yeah. So today my talk is going to be about why there are many SARS-CoV-2 variants have been evolved, but not all of them have turned out to be pathogenic. For example, in this case, Delta variant, and compared to all the variants including Omicron, Delta variant turns out to be having higher pathogenic potential than any other variants that's been there. So first of all, we try to find few clues and see whether we can come up with some kind of molecular clues that can explain why they're highly pathogenic, and can use this clues for identifying new variants, whether they have higher pathogenic potential.
Before I go into my talk, I just want to say the people who are involved in this talk. So 4 of my students, the lead one is IISc, then Sanchita, Dishan and Antika, which were involved in this work, and we have active collaboration with Prasad Tamineni from University of Hyderabad, because the work that I'm going to show now, we are not much expert in that, but we're trying to dwell into this kind of work. And we have multiple collaborators from chemical engineering, Narendra and Maipal Ganji from biochemistry IISc, and Shashank from CLDR, and another guy, Arinjay from University of Saskatchewan. Let's look at the virus evolution. The goal of virus is the moment it has its host, it's tend to start mutating. I mean, the reason it gets mutated is to tend to gain the advantage of virus fitness or higher viral loads with the time, because it'll help the virus to spread better in the host. Okay? But with this, it has again other complementary properties that it can gain. One is to adapt to the host, either by evading the host or regulating the host immune responses, and which will have impact over virulence and pathogenicity. So virus will keep on mutating itself, but whether it can really cause the pathogenesis or not, it's sometimes by chance or by strong immune pressure that it can expose to that. Okay? And if you look at SARS-CoV-2 virus, and many of the variants that evolved except the Delta one, and they tend to acquire a lot of mutations, but their fatality rate, which is in red color, is not that as high as the Delta variant that you can see here. We don't know the reason why there is sudden peak in the Delta variant mediated fatality response when compared to the transmission rate. But the reasons could be many. One is, for example, there's by chance that SARS-CoV-2 can evolve into less pathogenic or highly pathogenic. If it is directly highly pathogenic, then we know that how it's going to. But if it is evolving into less pathogenic strains, that doesn't mean that it can suddenly turn into a pathogenic, because it can turn into a pathogenic strain at any time. Not only that, now along with the humans and bats that we know, but the SARS-CoV-2 is sampling many other hosts where when they keep accumulating the mutations, that can be problematic for us when they jump back to the humans. So the question can be like, can we predict the pathogenic potential of variants so that if any new strain that comes into humans, we should be able to predict that, yeah, it can become pathogenic with time. Okay? So we know that most of the SARS-CoV-2 variants acquired mutations in spike protein, because that's the huge literature that's available so far. But the problem with spike protein mutations is that it will help us to understand that how well this virus can transmit among hosts or in the humans and all. But it won't explain how it got the pathogenic potential or replicative potential inside the cell, which is dominated by rest of not 29 proteins, which is encoding. So I think it's a time, I think many people are already looking at it. What's the role of non-spike mutations that this SARS-CoV-2 is attaining, and how do they determine the pathogenic potential or the less pathogenic potential? Okay. So if you look at the SARS-CoV-2 trajectory, this is the trajectory, like it starts with Wuhan strain, then you can see that alpha, beta, and delta, and Omicron right now is happening. This is from the covariant website. So we would like to understand, because this entire phylogenetic tree was made by the complete genome sequence analysis, but it will only tell you that how one variant is evolved from the other strain. It is nothing to do with the pathogenic potential of the virus. It's just the linear trajectory, how a virus from starting point 1 to starting point 10, how is it evolving. So to understand whether we can classify these viruses based on their pathogenic potential, what we did is that we tried to take all the non-spike mutations in all the proteins of SARS-CoV-2, and we listed them out, and we tried to make some kind of plots where you can see that this particular protein is mutated in this particular variant at that particular position. Then we also mapped onto the structure to see that it can really impact the structure. Okay? With that kind of analysis, we came to a few proteins which have shown unique kind of pattern of mutation evolution, especially with respect to delta and any other strains. Okay? And those are like NSP2, 3, 13, N protein, ORF3 and ORF8. And it turns out to be that now there are a lot of papers showing that these are the proteins which have a bigger role in antagonizing interferon responses, even other host cellular functions intracellular. Okay? So now we don't have a data, but with the help of Professor Narendra Dikshit in IISc, we would like to reconstruct these phylogenetic maps using only the sequences from this only, particularly with NSP2 protein or NSP3 protein from Wuhan strain to all strains. Now, if you do that, can this phylogenetic tree changes? Can we figure out whether it can, this phylogenetic tree will help us to understand how we can segregate this pathogenic potential of the viruses, which is under construction. But we became interested in one particular protein called ORF3A, which has unique mutations in delta strain than other strains because of multiple reasons, which I'll come to that in a point. And it has a big role in even inflammation regulation and cell death regulation that we are interested in this point. So this ORF3 is a membrane-bound protein, and it's been considered as a viroporin or channel protein, but there is one or two new studies which are debating that it cannot be a channel protein, but there's still a confusion over it. Okay? So, but one very well-known function that is established is that ORF3A inhibits autophagosome lysosome fusion in the cells which it gets expressed primarily by targeting Hobbs complex, which is required for the fusion. And it's also known that it facilitates lysosomal segregation towards the membrane. But why does it need to do all this function? And now later there are several studies came up and showed that this ORF3 is important for facilitating lysosomes towards the membrane so that the SARS-CoV-2 can use lysosomes as an egress or for virus budding. SARS-CoV-2 cannot egress itself or bud out itself through the classical ways that other viruses do. It uses lysosomes as a carrying machinery which tend to take them towards outside the cell. Okay? So now we started asking how ORF3A mutations from Delta variant impact either virus replication fitness or autophagosome lysosome fusion. So these are the mutations that the red color mutations are the ones which are from the Delta strain, and the yellow color mutations are from the Omicron strain, which we are trying to show that these mutations are accumulated at different hotspots of the protein. And we tried to model these mutations from Delta strain, and we could figure out that some of the mutations, they did not actually affect the structure, but one particular combination called ZY, I'll come to that point, that Z188 and Y21, they actually altered the protein structure locally. Okay. And also we established a simple technique that everybody uses in autophagy field. You have a LC3 protein which is tagged to ZFP and mCherry, where you can monitor the autophagy activation. So when you express this in the cells, and if you induce autophagy, and if autophagosomes fuse with lysosomes, you tend to lose the GFP signal. Okay? If they don't fuse with the lysosomes, and then you tend to see this yellow color or the merged signal. So this kind of assay that we use, and it's been reported that only SARS-CoV-2 ORF3A can do autophagosome lysosome fusion inhibition, but not the SARS-CoV-1. Okay? And that's what we see. We use this method and established in HeLa cells, and we could see that SARS-CoV-2 ORF3 expression increased autophagosome accumulation, but not the SARS-CoV-1. Then we introduced multiple Delta variant mutations into ORF3A, and then we did the same assay. And we could figure out the two particular mutations, DW and L108F, they tend to see increased autophagosome accumulation, that means increased inhibition of autophagosome lysosome fusion inhibition. But not all the mutations. These mutations also from Delta, but they didn't inhibit. The reason is when we express, somehow these mutations led to the intrinsic ability of losing the protein expression in ORF3 itself, and that could be the reason why they weren't showing anything. Okay? And then we also were wondering, is it only for the Delta mutations, or it could be for any mutations that have occurred in alpha or gamma or anywhere. So we took two particular mutations from Omicron, T223I and L106F, and then we did the same assay, and we could figure out that only Delta variant mutations were increasing the autophagosome accumulation, but not the the Omicron variant mutations. But one of the mutations, as usual, it has the less protein expression. So that means viruses keep on sampling the mutations. They can be beneficial for the virus, but sometimes they may not be. So, but we were wondering why Delta mutations, some of the Delta mutations, particularly L108F and L106F, which are there on the same helix, but one could able to facilitate ORF3A function, one cannot. If you guys know about a helical package, because this helices package at very particular fashion called heptide repeat interaction fashion, where they tend to have these interactions are dominated by hydrophobic interaction. If you look at the L108 position, which is facing towards the helical interaction that you can see, That's why when this leucine was replaced with filanin, this helix interactions might have been stabilized. But in case of Omicron mutation, sorry, it's written delta, but it's Omicron, this particular L10F is facing towards outside of the helix, which may not be facilitating the stability of the ORF3. So overall, what we would like to say with this preliminary data is that the delta mutations in ORF3A are actually enhancing its function by facilitating inhibition of autophagism lysosome fusion. Perhaps that might be helping them to be beneficial for better virus binding or virus egress. Okay? And we wanted to also see is this ORF3A protein actually sits on in the lysosome membrane, which can inhibit autophagism lysosome fusion. But how does it affect the lysosome dynamics? We wanted to understand that. And you can see in this video, the green color one is the lysosomes and the red color one is ORF3A. ORF3A nicely sits with the lysosomes. So what we observed is that when we expressed ORF3A and stain the lysosomes using LAMP1, the LAMP1 without ORF3, you can see that it's very, very nicely located in the perinuclear space, which is a conventional way of their localization. But the moment we express ORF3A, like people have reported before, the lysosomes start spreading out towards the periphery. Okay? And we also did high resolution imaging with wild type ORF3A along with the delta mutations. What we could see is that, particularly with two delta variants, the lysosome distribution has been very well enhanced compared to the ORF3A. And this is qualitative, but we wanted to actually establish a quantitative method to really see that it's indeed a real phenotype. And we adapted this technique from this paper called cell analysis, where you tend to grid the cell with particular size micron or particular size orbits. Then you can score the signals or lysosomes in them. And then with this, you can plot the granules or lysosomes which are close towards the perinuclear or which are towards the membrane. So we could see that the moment we express ORF3A, you tend to see them going towards the periphery, but and this signal has been increased with the ORF3A delta mutation. And not only that, we also try to monitor the LAMP1 expression on the membrane, because if you take cells and if you don't perimerize them and stain them, if the LAMP1 or the lysosomes are coming towards the membrane, we could be able to stain the lysosomal signal on the membrane. So that's what we did. You can see that marked cells, you have some basal level of LAMP1 on the membrane, but the moment you express ORF3A, the surface LAMP1, which is nothing but lysosome targeting the membrane, has been gone up, and it keeps going up with the delta mutations. Okay? So what we know so far with our observation, we know that ORF3A inhibits the autophagism lysosome fusion. Now with the delta mutation, this fusion process has been even more enhanced inhibition process, and not only that, the lysosomes have been come to towards the periphery. Okay, but why? How does this help the virus? I mean, we wanted to understand how does this help the virus to get increase their viral fitness or pathogenic potential. So for that, what we did is that we took A549 cells which are expressing H2, and we overexpressed these proteins, ORF3 and other proteins, and we infected them with the virus, and then we tried to see in the cell culture, are there any differences in the viral titers. But what we see is that there are no real trend in the viral titer differences, which is indicating that perhaps this ORF3A mutations may not be really affecting directly viral fitness, but they might be affecting host evasion responses or host manipulation responses. So that's where we wanted to look at, since lysosomes are going to the membrane, are they helping the cells instead of putting the virus out? They're trying to put the intracellular cargo outside, because it has something to do with pathogenic inflammatory responses, because any intracellular content comes out of the cell that causes massive inflammatory responses. Okay? So one particular thing which we are interested in is called unconventional secretion. So this pathway is called unconventional secretion because it doesn't go through the classical Golgi complex pathway, because they don't have leader sequences or any kind of signals that can go through there. So, and it's been very well established, and two very well established pathways. One is through the pores which are formed on the membrane, or through the autophagism or lysosomal vesicles which can take the cargo outside the cell. Okay? And in case of SARS-CoV-2 infection or COVID-19, IL-1 beta and inflammasome activation is known to be directly associated with inflammatory pathology and COVID-19 severity. It's very well established. There are tons of studies, even clinical trials have come out, and people are targeting IL-1 beta pathway. In general, in immune cells, this IL-1 beta comes out of the cells through pores called gas-terminated pores because it doesn't have any leaderless sequence. So if you don't have this pore, if your cell is not dying, they stay in the cell itself, they never come out. But many of the epithelial cells are non-immune cells, this pathway is not active. So that means they must be depending on the autophagism or lysosome mediated unconventional secretion pathway. And that's what we wanted to see, that do ORF3A delta mutants drive unconventional secretion of IL-1 beta, which is directly associated with COVID-19 severity. But again, in this case, we were asking whether is it through autophagosomes or lysosomes. I'll just summarize our data in 1 or 2 slides because we try to put a lot of energy to do imaging. But one challenging thing is imaging IL-1 beta because it's a small molecule, about 7 to 8 nanometers in size. But if you use conventional fluorescence microscopy, you only see the hazy signal and all. So this is a DNA paint. We did these experiments with a colleague in biochemistry who is a biophysicist who does super resolution imaging with using DNA paint. Now you can see that when we expressed IL-1 beta and LC3, which is nothing but autophagosome, and you can see that autophagosomes are there, and we do see a little bit collection of IL-1 beta, which we cannot conclude anything. But when we expressed ORF3A along with the LC3 and IL-1 beta, the autophagosome size have been increased like we have seen in confocal studies because the ORF3 inhibits the autophagosome lysosome fusions and the cargo is still inside. But even in this case, we see that IL-1 beta is in the vicinity of the autophagosome, but we could not conclude anything. But when we repeated same studies, the high super resolution imaging studies with LAMP1, which is lysosomes, we could see that without ORF3A, again IL-1 beta signals appears to be spread in the cell, and this is the lysosome that we stained here in cyan color. But the moment we express ORF3A, we observe very peculiar features of IL-1 beta forming like vesicular structure. But again, this is not completely colocalizing with lysosomes, which we are still puzzling why. One thing we think is that perhaps this IL-1 beta is loaded onto either damaged lysosomes or amphisomes, and that's the reason why they are not labeled with the intact lysosomes. So that's what I'm trying to show is that in case of ORF3 expression, this IL-1 beta nicely forms this ring-like structure indicating the vesicular structure that can be cargoed out of the cells, which is causing the heavy inflammatory responses. Okay, so to support this data, and we also figured that during ORF3 expression, lysosomes get damaged because lysosome damage is directly associated with cellular homeostasis imbalance and also partly associated with intracellular cargo delivery outside the cells or extracellular space. And we do see that compared to the wild type ORF3A, the damage in ORF3A delta variant mutations have been increased with it. So with this, what we think with the data, the limited data that we have is that the strains like Wuhan strain or Omicron strain, which tend to be relatively less pathogenic than Delta, the ORF3A tend to facilitate virus egress and especially the lysosomal target to the membrane. But in case of Delta, it is actually favoring along with the virus egress the heavy inflammatory responses that might be associated with immunopathology. But why are we interested in this? Because we want to figure out, let's say, this particular mutational patches that are happening in Delta variant ORF3A. Can we figure out or define the patches on the ORF3A if you say that if this particular patch has been modified, that can cause the pathogenic potential? So that's our goal with that. Almost done. And that's our story, but we also work actively on understanding why the bats could able to handle this pathogenic RNA viruses very well, but not the humans. So we just published this paper, but I just quickly go through in a few seconds. So we figured out that bats have this particular proteins called ZVP1, RIPK3 and RIPK1, which are important for activating programmed inflammatory cell death pathways, but not apoptosis. Okay? And they have a specific motifs of about 15 to 20 residues. Now we figured out that exactly some of the RNA viruses in bats, they mimic these motifs so that they can manipulate host inflammatory responses and host inflammatory cell death. That's what we figured out that SARS-CoV-2 and sp13, it has this rim motifs that I just explained to you that can directly interact with the host cellular machinery and favor cell death, which is indicating that perhaps SARS-CoV-2 has got intrinsic ability to favor more respiratory epithelial cell death because it's already has well been trained in bat immune system. With this, I would like to thank all of you and my funding system, and it's all that. I'll take questions.
So, with respect to viral adaptation, if you would consider that
introductions zoonotically in our population documented. Other coronas, sarbecoviruses, they're common. But the intriguing part is they are common in the pediatric population and not in the adult population.
Okay.
So coronavirus is essentially a pediatric disease except for SARS-CoV-2.
Okay.
So my question with respect to viral adaptation is that ultimately these viruses are found equilibrium. Why? Because the immunocompetent individuals eventually develop immunity. They kill off the older people. And you have pediatric population that is asymptomatic.
Okay.
So my argument is it the host equally or more important to consider in terms of the adaptation? When you are looking at viral mutation, right? That's point one. And point two is with respect to the vaccination confounding effect. So,
Okay.
Ever since April of 2022,
Got it.
sorry, 21, there has been a major confounder through vaccination. And have you tried to focus any of these adaptive pathways that you're looking at using that so-called confounder?
Okay, so we don't per se work with vaccine-related thing, but I understand your question about confounding factor. For example, you could say that the variants that came after Delta, we could not get any disease simply because we are vaccinated. That's not true because it's been there. Many animal studies have been done with Omicron, Delta, and everything. The animals which have never seen the virus, you can see that intrinsically Delta has higher pathogenic potential than any other strain that you can see. The other thing is, yes, adaptation is already happening. That's why, and definitely one of us might be already have SARS-CoV infection over here, but we don't spread our venom because we already get adapted to them. But the virus, the humans are the platform for them, the continuous evolution. As long as they're there, they always have a potential to adapt or they suddenly flare up. And not only that, because humans got infected, we humans have transferred this virus to many other hosts. Like you might have seen many newspapers saying about white-tailed deers. They have tons of SARS-CoV-2 variants. But what if, and they also got accumulated a lot of new mutations that you don't see in any of these viruses. But what if they come back? Do you think, we don't know how they're going to behave.
So just to follow up, sorry, just follow up later. There are two more questions, one here over here.
Yeah.
Yeah, actually, this side.
Okay.
You were talking about reconstructing the phylogenetic tree with respect to considering the non-spike mutations and its contribution to pathogenicity. Now, I just wanted to ask that, when you were considering only the non-spike mutations, did you find any change in pathogenicity or any cluster of virus with a different pathogenicity within Delta?
Yes.
As we know that mother Delta was very highly pathogenic than any other variant, the main Delta. So if you consider omit spike and consider only non-spike, then did you find any change in pathogenicity within the Delta variant?
Yeah, we haven't found any answer for your question because that's what we are doing it. Handling all these sequences, since tons of sequences are there, handling them and making this phylogenetic tree with particular, let's say, I say that ORF3 has specific mutational pattern that can differentiate pathogenesis. Doing that is becoming challenging with entire population of viral sequences. So now, with the help of Narendra Dixit, we are trying to focus on geographical area specific sequences, then tend to see that. But we haven't come to the point where by looking at phylogenetic tree, we can already differentiate them. But that's our goal. Whatever you asked is our goal, but which we don't have an answer yet. We used the Hong Kong strain, the first Hong Kong strain that has come up. Yeah. Yeah, the reason we did that is because if we take any other strains like alpha, delta or anything, because they already, not only the mutations of ORF3A, they also have other mutations. So it's hard to say that what's the effect of ORF3A mutation. That's why we created cells which are expressing this ORF3A wild type or the mutant ORF3A in excess level. Then if you put the virus, its own ORF3A levels are very limited that the phenotype effect can be seen.
Last question. Yeah, nice work, sir.
Yeah.
So one would believe that the immune response per se would be driving virus evolution. In case of SARS-CoV-2, obviously the antibodies are probably contributing. That's why you see a large number of mutations in spike. So what is the selection pressure on ORF3 or any other non-structural protein to mutate? Do you think there are T-cell epitopes that are changing? Have you looked at that aspect?
No, I haven't looked at it, but I agree with you that it could be T cell epitopes. We were initially thinking it's B cell epitopes as well, but I'm sure you must be knowing Jesse Bloom's work and everything who is doing, where now he could figure out that now this Omicron, the new strains that are circulating, they're so very well evolved that they're coming back where they're accumulating mutations in the spike that are reducing affinity to the broadly neutralizing antibodies. So that means it went too far than what we could imagine. So I think that may not be happening with ORF3A. That could be because either it could be through the T cell epitopes because it's a small set of peptides, or some kind of innate immune activity that we don't know much about it.
Thank you, Keshav.
Okay, thank you.
So we move to the second presentation. Now allow me to invite Keshav to respond to. His group is engaged in the examination of host interactions with emerging human RNA viruses with a primary focus on influenza and flaviviruses, including Zika and SARS-CoV-2. His lab is centered around overarching themes within host-virus interactions, including the study of viral strategies for evading or antagonizing the innate immune system and the utilization of cellular machinery by viruses. With an aim to identify potential targets for the development of broad-spectrum antiviral therapies.
Thanks, Professor Uday. So this talk was supposed to be by Professor Anita Shetty, and we were to hear about monkeypox virus emergence. I'm trying to fill in her shoes and bring in some data available in our lab which fits in evolution theme. And it's more of a pathogenesis work, but there is some evolution aspect to it. I'll try to explain. So all of us have suffered firsthand. It's a respiratory virus, SARS-CoV-2, there is no doubt about it. But the motivation for this study came back in towards the end of Delta wave, where colloquially we heard from patients as well as doctors that a lot of people were suffering with GI symptoms, which at that point we had no knowledge of. We didn't look into it carefully. But I thought, what is going on and why GI symptoms are showing up only in severe cases? So the motivation was that our GI symptoms are hallmark of a severe coronavirus variant. And to understand that, we initiated the study. At the same time, the Omicron variant also emerged. So goal was very simple, that let's try to compare whatever BOCs are available in whatever model systems are at our ease, and then see what kind of different pathogenesis we can get, primarily looking at the GI tract infection and tropism of the virus. So I'll give you some background, which when we started working on, we looked into it. So coronaviruses in general, coronaviridae can be divided into alpha, beta, gamma and delta coronaviridae. And majority of animals which are infected, they actually develop GI disease. So other than humans, closest will be, let's say, other mammals, bovine, canine or feline. They get infected with alpha and beta coronaviruses too. They develop diarrhea and other GI symptoms, sometimes hepatitis too. Respiratory symptoms come from gamma coronaviruses and delta coronaviruses, which primarily infect birds. And bats are considered as reservoirs of the three problematic coronaviruses, MERS, SARS-1, SARS-CoV-2. They don't show apparent disease, but they shed a lot of viral RNA, sometimes infectious particles also in bat feces or guano, which is one of the primary samples who do bat surveillance they collect. So there is indication that there also the virus is present in the GI tract. Now all these are potential reservoirs. So overall you can conclude that majorly in the reservoirs, the virus is GI tract infecting. But whenever it comes to humans, it becomes a respiratory virus. So that was kind of odd. So I'll try to convince you that whenever that happens, virus has this latent memory of that it's a GI tract virus, and if it tries to go back, that's an indication that it's going back to a severe phenotype. That's what I'll try to convince you through the data. Now, why that is important to understand in study is that whatever organ is being infected, That will decide what kind of disease will develop. That will decide what will be the route of transmission as well. Respiratory virus, this is SARS-CoV-2, primarily spreads through aerosol or through contact or touch through surfaces. But fecal-oral transmission, whether it happens or not, that was the question when we started the study. Since then, many papers have come. Although there are sewage water surveillance programs going on where you can detect a lot of RNA in the sewage sample, but we are convinced, based on our data and other published reports also, that fecal samples have no contribution in viral transmission as far as SARS-CoV-2 goes, although a lot of viral RNA is shed. Probably not through fecal route, probably through people gargling and spitting in the sewage sample. That's how it goes. But other than that, majority of the coronaviruses, we look at them in their wild reservoirs, they cause GI symptoms, except for the birds. Bats I discussed already, that they don't show apparent disease, but they do carry virus in the GI tract. Now, these are the list of viruses which have infected humans, have established themselves, or having trouble with human population. Out of these 8 viruses, the SARS virus I am more worried about. This is a GI tract infection causing virus. In young piglets, 5 days old, the mortality rates are 90%. All the piglets die. And it's a similar story like H5N1 bird flu. It just needs few mutations to adapt and come to human population. So if I have to be worried about all the other SARS-CoV viruses which are present in adjacent reservoirs, I'll be worried about that. So far, it has infected humans, caused severe disease, but not spread human to humans. But all the other 7 listed viruses, they have caused infection. Many of them are seasonal coronaviruses mentioned earlier. But if you notice, these seasonal coronaviruses, they are coming from a rodent origin through a bovine intermediate. In both these intermediate zoonotic reservoirs, the disease is GI disease, not respiratory. When it comes to humans, it becomes a respiratory virus. Similar story here also. In bats also, the virus is present in GI tract. When it comes to humans, it becomes a respiratory virus. So something is odd about it. Now, there are a lot of studies, probably in thousands, which have looked at patient samples and presence of viral antigen or viral RNA in different organs, in mild versus severe versus fatal cases. If you take a summary of all of that, this is all you get. In a human body, you can detect viral antigen in all these tissues listed, pretty much everywhere. And the statement at the bottom is virus tropism largely is correlated with ACE2 tissue distribution. So when I read this statement, I thought let's look at ACE2 tissue distribution. I'll come to that, but I'll mention that we were surprised that highest ACE2 expression is in the GI tract, actually in the small intestine. So if ACE2 was the driver of the tropism of the virus, we all should get a GI tract disease because of SARS-CoV-2. So it immediately negates the fashion that statement that ACE2 is not the main determinant of the viral tropism. There is something else. And if you look at symptoms also, so there is a lot of sequencing data available, but often happens that patient metadata is not available. So both virus genotype and disease severity, that kind of information doesn't come together. It comes in regional locations. So there is a ongoing effort in Hong Kong. There you can look at the initial SARS-CoV virus and SARS-CoV-2 virus. There are a variety of 26 symptoms actually. Top symptoms are listed here. There is listed diarrhea. In case of SARS-CoV-2, it was about 25%. In case of SARS-CoV-2, it is only 4%, but minor. But we are looking at, is it a hallmark of severe disease? So if severity correlates with GI symptom, then that matches. SARS-CoV, which was a very severe virus, it does have high incidence of diarrhea. And maybe these 4% people who are showing GI symptoms, they are the severe cases. That was the initial motive of the study. But if you look at overall GI symptoms across different variants also, this data is from a very large cohort from England. There also you see that yes, GI symptoms are present at low incidence rate, but there are strain specific variants also. I would like you to focus on this light green one, which is Delta variant, which shows that there is slightly higher GI tract symptoms being observed in a large cohort where both mild and severe cases are mixed together. But we thought probably if you took out only severe cases from this cohort, Delta would look much higher, which again matches the hypothesis that Delta being a severe virus, probably you are seeing the GI tract infection. So Yeah, this was the statement when we looked at ACE2 expression in protein atlas and mRNA atlases, multiple databases we looked at. When you compare in lung, stomach, small intestine and colon, highest ACE2 expression is in small intestine. This is human data. And if you look at TMBRSS2, potentially another tropism determining factor, it doesn't follow any particular pattern. So we quickly tested in whatever GI tract cell lines were available in the lab, CACO2, which is a colon cell line, AGS, which is a stomach cell line, and did a quick serotype particle entry assay. What we see in CACO2, there is very poor entry, and this is a distal GI tract, so possibly the virus doesn't like to enter there. In stomach, you see quite a bit of entry, and there is clearly a stark difference in ability of delta to enter these cells. So again, supporting the initial thought that probably delta is a severe virus, and it is able to enter these GI tract cells better, and which leads to GI tract disease also. We were working on SARS-CoV-2 animal models at that time, Syrian hamsters. We thought whether this model can be replicated, or can it recapitulate this expression of ACE2 what is reported in humans. We did ACE2, TMBRSS2, RT-PCR in healthy animals in different tissues. The highest bar you see here is in the intestine. Mind you that this is a different kind of data representation, 1 by delta CT, and this is some other sort which comes from deep sequencing. But you do see highest ACE2 expression in intestine. So that motivated us to take this study to animal models, because if it is recapitulating the ACE2 distribution the way it is in humans, it should recapitulate the disease also. We'll start here now. Data wise, this was the study plan. We compared the ancestral Hong Kong or closest to Wuhan with Delta and Omicron variants, collected samples either on before infection, day 0, day 2 or day 4, and these were all the parameters we calculated. I'm not going to present everything. Some of the key data will present. There are a lot of negative data. I'll just discuss that in the end. Looking at clinical signs and body weight, few of the things in the beginning are very obvious. Everybody knows about it. There's a lot of published data about that too. So we tested these 3 viruses at 2 different doses, 10 to the 5 PFU and 10 to the 6 PFUs. And you can clearly see that for Delta virus, highest dose, you see more body weight loss. For Omicron, you see less body weight loss. And the ancestral strain is somewhere in between, intermediate phenotype, which is reported by many, and nothing surprising about that. We looked at viral RNA load and infectious virus counts also. Omicron has lower viral load, both RNA as well as infectious particles in the lung, which is considered, let's say, lower respiratory tract, which is also reported. So we were able to recapitulate that data. Delta is slightly higher compared to both viruses. In nasal wash, on day 2 and day 4 when we looked, this is something interesting. On day 2, Omicron has expression of viral RNA almost comparable to Delta and Hong Kong strain, but day 4 it drops down drastically. So probably this early high level was facilitating fast transmission of Omicron, because everybody knows that has much higher level of transmission compared to other viruses. Look at lung gross pathology, summarizing that there is more lung lesions in the Delta, which is also expected. This is a gross estimation of the inflammation. You basically take a ratio of lung versus body weight. For the Delta, you see higher. All of this is expected, reported by others too. Similar for some of the inflammatory markers, TNF alpha and interferon gamma, you see higher level expression in the lungs in case of Delta challenge. Lung tissue pathology, it continues. You see more severe cellular infiltration, loss of bronchial epithelium. Those parameters, if you score them, you see much higher histopathology in case of Delta challenge compared to Wuhan or Omicron. You detect the viral antigen presence, basically infected cells through immunohistochemistry. You see similar pattern. All of this correlates. But I continue to repeat, nothing surprising. All of this has been reported. We are just verifying what is there. Here some interesting new data starts, which we have looked at for the first time in hamsters. This is blood parameters comparing these infected animals, Hong Kong Delta and Omicron. RBC parameters, what are listed here, they basically overall represent what is the oxygen carrying capacity of the organism. And you can see in Delta there is a clear drop of RBC parameters, So in a hamster model, you can actually detect that animal's ability to carry oxygen is dropping down by looking at the whole blood count. And platelet parameters, basically they overall indicate the chances of getting a thrombosis or sepsis or severe blood mediated reaction, and in Delta and in ancestral strain also it has gone up compared to Omicron. So that's also something which hamsters can tell you about. WBC parameters, there is cross immune dysregulation, there is basophil and eosinophils going down, which shows immune suppression, and monocyte count and WBC counts going up in Delta, which showing hyperimmune reaction at the same time. So there is gross misregulation. So this was interesting to learn that you can figure out these things using Syrian hamster model. Now here's the interesting part. We looked at the viral RNA load in the GI tract, starting from the anterior to the posterior GI tract. Expression level ranges from 10 to 3 to 10 to the power 6 copies of the viral RNA. To compare with the lungs, in the lungs we get 10 to the 6 to 10 to 7. So although tenfold less, but there is significant amount of viral RNA being found in the GI tract. Now one might question, how did this RNA came into the gut? While inoculating, the virus was swallowed and then it reached there. So that hypothesis doesn't work, because in stomach you see lower expression. In esophagus also you see lower, but when you go to intestine it goes higher, and especially in case of Delta it has gone much higher. So one might argue that this is because intestine has the highest ACE2 expression. Probably that is why it is more susceptible. But for this data to appear, virus must get there and cause the infection. So hypothesis is that virus is not coming through the oral route. There is viremia happening, and there is some infectious virus in the blood which reaches the intestinal lining through the circulation, and then in that case the infection should appear at the bottom of the intestinal lining, not at the top, not on the apical side. And as you go down the GI tract in large intestine, the virus titers drop drastically. This is actually below detection limit. So this also shows there is a particular preference in the GI tract also for Delta virus compared to other two viruses to infect. And we did look at the fecal matter also of the animals. There was no RNA detected or very poor, and there was no infectious count, which corroborates other studies which have looked carefully whether fecal-oral transmission is possible or not. So that is not there. But small intestine infection was definitely there, and that's we went on to investigate further. So this is looking at sort of pathology in small intestine infected with Hong Kong Delta or Omicron. So the parameters we are looking at, this is basically the basal side of the intestine, which is close to circulation, and here you will find the enterocytes. This is called the crypt, and this is the villi. So if there is pathogenic infection in the intestine, the villi will shrink. Their ability to absorb nutrition will go down, and that's associated with diarrhea too. And that you see in case of Delta, there is much more disturbance. And crypt is basically enterocytes. If they are being infected and dying, there will be more regeneration, so crypt will increase in size. And those things you can count actually. You can see the crypt size increases upon Delta infection, and villi length goes down upon infection with all these three viruses. But the way this pathology is calculated is that you take ratio of crypt length versus villi length. So there is clear evidence that upon Delta infection there is significant pathology happening. There is viral RNA at high level. There is pathology happening too, which you can detect. Then we thought, if you have to categorically show that there was infection and because of that it happened and not just a host response, we need to detect viral antigen being present. That became a challenge because viral antigen is there but at very, very low quantities. Although the viral RNA is at high quantities, although tenfold less compared to lungs, but viral antigen was quite low. Out of 4 animals, only in 2 animals we were able to detect few PFUs, but at least we were able to see that virus got there, it caused infection. And infection was caused in the same site where enterocytes or the crypts are. So it corroborates the whole data so far. And this is data from other published study which shows that small intestine infection is a hallmark of fatal coronavirus infection in human. So relating these two items, you can say that hamsters can tell you about if a virus is progressing towards very severe diseases, especially if you look at its small intestine, if there is viral antigen present. So the conclusions are that SARS-CoV-2 have white tropism, which can vary between different VOCs. They are not just respiratory viruses. They come from a GI tract background, and in humans they can infect a wide range of cells, especially if it is a severe case. In GI tract, the viral RNA load decreases from anterior to posterior GI tract. In Delta, it can actually infect the GI tract, small intestine preferably, and cause pathology. Syrian hamsters actually are a sufficient model to look at infection in the GI tract. Although we looked very carefully, they do not show any symptoms. There is no diarrhea. There is loss of appetite, you can say. Because of that, there is body weight loss too. But otherwise, GI symptoms are very limited. But GI infection can be detected. And viral RNA shedding in the fecal matter either is undetectable or not there. Probably because it's not there in general, in humans too. So fecal-oral route transmission is not there. And coming back to the initial statement, is GI tract infection pathology is potentially a sign of virulent VOC and severe disease? Based on this data, we believe yes, that is true. So if GI symptoms start appearing upon a new wave, one must be taking that seriously, that it is moving towards a more severe phenotype. That's the conclusion. And the study was led by Santosh and Christy primarily in our lab. I thank the financial support from these funding agencies. This is our lab, and thanks to their hard work.
Very nice talk.
Thanks.
How much infectious virus do you see in the wash of the small intestine? I mean, PAQ that you showed, was that from extracted tissue or was it from the wash?
Extracted tissue. We homogenized the tissue and then looked at. So there is no virus in the cavity. It is in the basal lining. So that will not go away by washing.
When you say it's shedding virus in excreta?
RNA, not infectious virus. We did not detect anything. Yeah.
So Shashank, when you look at the here, so when you look at the time course of infection, when is there like you see it in the intestine at later stages or you see it at day 2 when you infect animals?
So for intestine, we have not done time course yet. We are waiting for the reviewers to ask that. But in the blood, we have done that. In blood, the RNA viremia happens at day 2, and at day 4, it drops down. So that matches our hypothesis that there is an early viremia, which allows the virus to go through circulatory system to the intestinal lining, and that's when the infection happens. But you're right, we'll most likely end up doing that experiment. Yeah. I can't comment. We have only looked at day 4, but I don't know what will happen later. But we were very interested in looking at whether there is some viral shedding in the fecal matter and whether there will be fecal-oral transmission, because that has been reported in SARS-CoV. There was a patient 0 story that he went to a hotel and he dumped the virus in the commode, and through the sewage lining it went to all the rooms in the hotel. Everybody got infected. So that kind of stories are there. So that's why we're interested in looking at that, but we don't see any evidence that that has contributed. Yeah.
Great talk. So the GI symptoms were also reported a lot before the Delta wave. You know, they didn't really seem to peak during the Delta wave. And my second point is that they were all considered to be kind of safe. You know, the doctors never worried about, because GI tract patients never really progressed to severe.
Well, that's what we are arguing, that one should look at it carefully now. Especially, I mean, that data I showed in the beginning from the INGLAD cohort, the REACT-1 cohort, 150,000 patients. So there the Delta virus had slightly higher prevalence of GI tract. You see these green things? Diarrhea, slightly up, higher compared to Omicron. And in large cohort, the data don't change too much. Yeah, so that's listed here actually. This is wild type, alpha, all of them are here. So slightly lower compared to Delta. Now this is mixed with severe and mild everything. So if I had to go back and extract that data and look at only severe cases, I'm quite sure this data will change.
What I understood was that Delta was able to infect lower respiratory tract as opposed to Alpha and Omicron. And I also see the same you have higher load in lung, why you do not, wish to continue with the same theory saying that this is because of lung pathology?
GI tract infection because of lung pathology?
No, I cannot connect
the severe disease because of lung pathology.
Because of the lung pathology.
That is very well established. They are, we started with that thought that we could wrap the story there, but that has been reported by at least 2,000 papers.
Correlate, what you are proposing, then you need to, get the metadata as you correctly said.
Correct.
Till that is done, I think it will be difficult to, negate what is already known.
Yeah, so lung disease is the primary disease, that is the driver of whatever you see in patients. We are just claiming that GI tract also contributes if there is a severe variant, not always. So that's the statement.
And you never thought of doing electron microscopy of, tissue to demonstrate virus in cells, etc.?
We did IISc in the small intestine. It showed.
That's not electron microscopy, right? Just to demonstrate viral particles.
Viral particles. I mean, we could do that. IISc is little quantitative. You can actually count how much load is there and you can plot it. Yes. Yeah.
Sir, if you get in mouse model, you try with mouse?
Mouse, they don't get infected as such by wild type virus. We have a adapted strain.
H2 transgenic or the H2 transduced?
I am not a big fan of H2 transgenic. It gets a neural disease which doesn't represent anything. So, but in a mouse adapted strain, we will probably have a look at it. Yeah.
So you have not done a similar study?
No, not in mouse. Not in mouse. We were trying to optimize hamster and trying to see other than respiratory disease, can it represent blood parameters, GI disease and other.
H2 transduced intratracheal administration route. Mice recapitulate the disease severity in a very similar way to
Right.
And that is an easy model to dissect out different.
Right. We have that. We plan to use that for antiviral testing quick studies, but since hamsters are there, they are far superior. So we've not gotten that. Yeah.
So, yeah. What about gut dysbiosis?
Yeah. So that's ongoing. We are looking at the microbiome right now. So the samples are out for sequencing. We are going to compare the gut microbiome status, comparing across VOCs. Again, that's a very rich field now. There are a variety of studies which have reported that SARS-CoV-2 infection causes gut dysbiosis. Strain wise, is there a difference or not? Probably will be the first to look at or report. That's ongoing. Yeah.
Thank you, Shashank, for the challenging work.
Thank you.
So our next speaker is Professor Vidya Mangala Prasad from Indian Institute of Science. I just figured that this session is only by the organizers. Speakers are two conveners you heard. Rita is from IISc, Sweetie is from PHSTI, and our other chair is also from JHC, which is also co-sponsored. So Vidya's lab have been working on the structural analysis of the human viruses, and they are mostly doing high-end microscopy, particularly with the cryo-electron tomography and subtomograms, averaging single particle cryo-electron microscopy, and various biophysical and molecular methods, trying to understand the infection machinery employed by different human viruses. And today she is going to talk. It's actually her debut innings in the virology match, in the home ground. She is going to talk about the structural analysis of the epitope targeting by the broadly neutralizing antibody. Good luck.
Thank you, Professor Das. A very good evening to everyone. I hope everybody can hear me. Before I start, I'd like to thank the organizers for giving me this opportunity to really present my lab's work and to such a specialized audience. So just to start off, our lab is relatively new and I just wanted to kind of highlight what we do. So our lab focuses on structural biology of enveloped RNA viruses. We are primarily interested in enveloped RNA viruses because many of them are human pathogens, and to this audience I don't have to really introduce these pathogens, but these are some of the viral families that we are interested in. all the way from flaviviruses such as dengue, Zika, coronaviruses, coronavirus to a small extent, retrovirus and orthomyxoviruses. And our main method of looking at these viruses for their structure is actually through transmission electron microscopy. So the biggest advantage here of using transmission electron microscopy is that when you're looking at viruses, before cryo-EM or EM came into the picture, a lot of the work was done using crystallography, where you had to actually crystallize the viruses. And in those cases, when you have a non-enveloped virus or just a protein coat, they do crystallize. But when you come into the section of enveloped viruses where there's a membrane involved, then crystallization becomes a huge task. It's actually not possible because the membrane actually gives it much more flexibility. And so transmission electron microscopy, and especially cryo-EM that I'll be talking about in this, has become more of a very important tool to look at these enveloped viruses, and our lab focus is more on RNA viruses that are enveloped. So we do different types of TEM in the lab. We do conventional TEM sectioning, so this is just a cell section, resin section of room temperature virus infected cells. We also do negative stain TM, where it's actually just purified protein or purified virus, in this case just purified protein. But beyond just the staining techniques, we also do cryo-EM, which is actually cryogenic transmission electron microscopy. So transmission electron microscopy, cryo-EM, basically the biggest advantage of doing that is that you actually don't have any kind of heavy metal stain. There is relatively much less dehydration, and so you actually reduce the amount of artifacts that you're going to cause onto, especially when you're looking at viruses that actually have membranes or vesicles or anything that can have a lot of effect due to dehydration. So here you actually most of the time have your virus sample or actually any sample in buffer, and you actually have a thin layer of the buffer, which you actually then flash freeze to actually give you a vitrified specimen. So here the ice basically becomes like glass, which you can then put it into your transmission electron microscope. This is what we have currently at IISc. We have a 200 kV Talos Arctica, and you can insert your grids into the microscope to actually start collecting your data. So within cryo-EM, we have major types. One is single particle cryo-EM, some of which you heard in today morning when Dr. Manideepa was talking. We also do some amount of cryo-electron tomography, and the major advantage between the two methods is that single particle cryo-EM does really well with homogeneous purified samples. So we just have an example of a cryo-EM micrograph of purified virus particles here. And so if you have a homogeneous virus population, or even actually any protein where you can actually average between the two particles, then single particle cryo-EM is the way to go. But if you're looking at samples where that's actually not true, which is the case, for example, in influenza virus that I'm showing up here, where each virus particle can have very different morphologies, then you can't actually apply single particle cryo-EM, because you're assuming in that case that every particle is homogeneous, which is not the case here. So here so this is actually a 3-dimensional tomogram that we actually collected here in the IISc facility here. So this again shows it's actually a 3-dimensional volume that's going through stacks. So you're actually seeing all the way from the outside of the virus, which is the hemagglutinin protein, all the way to the inside, where you can actually see the helical nucleocapsids ordered into the virus particle. And the advantage of doing cryo-electron tomography is that the way the reconstruction is, the way the structure is determined, you're not assuming any kind of symmetry or any kind of assuming that they are homogeneous, which really helps in looking at more pleomorphic viruses at a 3-dimensional structural level. So that's more on the introduction of what we do in the lab. So in today's talk, I'm just going to shift a little bit, and what I'm going to talk about today is on HIV. So HIV virus, again, I'm not going into go into the introductions here, but HIV, it's a very well-known pathogenic virus. It's a retrovirus, and it's also a pleomorphic virus, and its main antigen on the surface is the envelope protein that's on the surface of these viruses. And my talk will be focused on what we learn on the envelope protein here. So the envelope protein is the sole target for neutralizing antibodies against the virus. I just have the structure that is being solved of the ectodomain on the right here, the PDB model of the structure, and it has been very well characterized. And despite this being the sole antigen, it's also a very challenging target because of the immune pressure and the rapidly mutating characteristics of HIV. The envelope protein actually has many different, rapidly evolves, there are many strains where the envelope sequences are different, and though it is actually a whole membrane protein. Recently we have structures of the full protein, but primarily the ectodomain has been characterized very well with X-ray crystallography and single particle cryo-EM. Right, so my talk today is going to be focused on the ectodomain, and in this ectodomain, what I've shown up here is it's actually a non-covalent trimer of heterodimers. So the top part is GP120 and the bottom part is the GP41 in terms of terminology, and CD4 is where some of the very common epitope sites are what I've labeled here. So though the HIV envelope ectodomain is very well characterized, one of the caveats that has happened, and that's primarily because it has been a difficult protein to crystallize and work with, is that a lot of the initial, or if you look into the structural database, most of the structures that have been solved are actually from very stabilized constructs. So they are from very specific stains that have actually been, and in the field, most of them, you will hear the word of SOSIP constructs, which basically means they have disulfide bonds and specific mutations to kind of stabilize this trimeric structure. And so all the structures we have are very rigid in their conformation. And so one of the questions that our collaborator in this work asked was, so the amount of rigidity that you see in these highly stabilized constructs, is that actually true across a panel of HIV envelopes? So when you, so they actually use hydrogen deuterium exchange mass spectroscopy, and very simply what it looks at is that it actually looks at the exchange between hydrogen to deuterium in the sample, and the idea is that if you have a very protected peptide, there will not be enough exchange, and if the peptide is more free or available, or there is any conformational change, then you have a lot more exchange and you will see a larger signal. So based on that, when they look at different isolates, what they see is that there is actually a conformational change at different peptides all across, which shows that compared to all the rigid structures that we actually see, there's a lot more conformational flexibility that's happening in the envelope protein. Right, and in some cases, the peptides that they're mapped suggests that the conformational sampling is actually between the closed structure, which is the wild type structure assumed, and a slightly partially open conformation that keeps switching back and forth, and that contributes to a lot of the flexibility that's seen between different envelope types. So in this study, we focused on a specific epitope. We wanted to look at how that epitope would be affected when it comes to being targeted by broadly neutralizing antibodies. So the epitope that we looked at was the V2, V1, V2 loops, the variable loops at very much on the top of the HIV envelope. And so this is actually, again, a very important site for antibody binding, broadly neutralizing antibody recognition and binding. And on top of it, it generally binds on the top, all the trimers together is how most of the antibodies bind. And so when we looked at it again with hydrogen deuterium exchange, what we find is that this variable loop is again, does have a lot of variation if you look through a global panel of HIV envelopes, which are actually from different clades. The BG505 here is actually the most rigid or the most well characterized HIV envelope, and it falls more on the rigid scale, but there are other other envelope strains that are actually pretty flexible in how the dynamics are happening. So we we selected one specific antibody, which is the PGT145, broadly neutralizing antibody, which is very well characterized, and we know that it binds to the V2 loop on top of the envelope. And we asked the question for an antibody that actually binds a quaternary epitope, how does that recognize, or is there any variation in how it binds to these? So we know from binding studies that it actually does bind most of these envelopes. So the question we asked was how does that binding differ? Does the antibody actually adapt itself, or how does it catch all of them in the same conformation? So when we did, when our collaborators again did BLI, which is actually just binding bilayer interferometry to see how the binding occurs, what they found was the V2 loop dynamics that happen between different envelopes affects how the association of the antibody is onto the on. So it actually doesn't drive the dissociation of the antibody, but the antibody to be able to bind to the envelope, that really depends on how dynamic that V2 loop is on the surface. So the association rates can be low, and that happens. And this binding studies show also that there's an inverse correlation. So the more rigid your envelope strain is, where it's found to be more rigid, then the binding actually happens, the association happens pretty fast. And if you actually have a more flexible, then the association rate is more low. And that's kind of what they were able to see. And also that this binding was also very specific, at least from the hydrogen deuterium exchange studies, they could not see any other effects that it was affecting other parts, which meant to say that if you have a dynamic envelope, the other parts might still have their dynamic activity. When the antibody binds, it actually doesn't locking all the parts together into a rigid, only the local areas are actually getting rigid. So to kind of see how it would look from a structural perspective and whether we see similar changes, we went ahead and selected 2 envelope isolates here. So CNE55, which is actually from the more ordered trimer or more the rigid trimer. So these envelope constructs that we have are still ectodomains, but they don't have any stabilizing mutations in them. And so they are actually more basically what they've used in the HTX data. And so we went ahead and selected 2 of these dynamic, one ordered trimer and a dynamic trimer, and we complexed both of them with the PGT145 Fab instead of the full IgG. And we went ahead and did single particle cryo-EM analysis for both of them. This is just a sample micrograph on how these micrographs look. So you actually, the envelope particles are pretty small, so they are all these small triangular density that you see. And so the data that we collected, we had a large data set for both these complexes, and we actually calculated the 3-dimensional structure for both of them. So the CNE55 plus PGT145 complex and the BJOX 2000 plus PGT5. So on the left is the more rigid trimer, and on the right is the more dynamic trimer. And so we had overall similar number of particles for each of them. And in terms of resolution, also we had very similar resolutions. So this is just a resolution scale on how well the different parts of the structure is resolved. And in both cases, so we have about 5 angstroms and 4.9, so very close in terms of resolution. So we could start comparing how these 2 structures were when they were binding the antibody. Okay, so overall, if you look at it, as you would expect, right, as the Fab has a specific epitope, globally, both the structures look very similar, and it's also similar to the BG505 SOSIP construct structures that have been solved, that the PGT45 overall footprint is very much the same. So the Fab contacts are overall conserved. So the the residue contacts between the Fab and the actual envelope is particularly conserved. But the difference that we see is that if you look into it more carefully, if you look at the smaller differences, you actually see that there's a lot more difference between the BJOX plus PGT145 Fab. So the more dynamic trimer with the Fab has small variations that you don't see in the more rigid antibody complex. And for example, the groove of the CDR loop that actually goes in to bind the quaternary epitope that actually binds to the other protein residues on the envelope, we actually see there is a shift almost about 4 angstroms downwards, where actually it almost feels like the antibody has to go in a little bit deeper to get hold of the envelope, which is not the case when we compare all these different structures together. It is not the case between the more rigid envelope structures, but we only see that with this dynamic BJOX strain. The other differences, for example, are also where the glycan positions, which were not very resolved in others, all the other many structures it was not fully resolved, but in ours we were lucky that all the glycan chains were pretty well resolved, and we could clearly say that there was almost an 8 angstroms difference, which is a large difference in terms of where the glycans are positioned, but the antibody was still able to bind, and the binding contacts at the end between the different antibody complexes were actually still preserved. And the way the antibody is doing that is that there is a slight tilting, which I am not showing here, but there is a slight tilt that it does. So the antibody, actually residue contacts that are happening are still preserved, but the global position is slightly moved in such a way that it still accommodates this dynamic envelope on the surface. So that's when we started looking more into the glycans, and we realized that in this structure, and so it's a very complex slide here, and I don't want you to go through everything, but the take-home message here was when we looked at each of the glycan contacts, so we found that each of these, as a trimer, each monomer has one conserved glycan, which is the N160 glycan here, and each of the glycan actually interacts with the Fab. So it's almost holding on to the Fab at 3 different positions, and that interaction site between the different glycans and the Fab is conserved across the two complex structures that we have. And at the same time, this type of glycan, this is the first time we were able to see that all the 3 glycans were actually involved. And if you look at the surface area that's been occupied by the interactions between the glycan and the Fab compared to the Fab to the protein area, it's almost equivalent, which means that contrary to what we would generally assume, the glycans are equally important in how the Fab is likely looking at or how it's identifying the envelope or actually recognizing it. So in a similar, So, and in a similar way, there was the other glycan that we looked at was N156. So, because our resolution was not very high, we were only at 5 angstrom, so we didn't actually model the whole glycan here. But you can actually see that there was enough density which showed that apart from the N160 glycans, there were also other glycans near that interface that were also interacting with the Fab, which meant that the glycans were equally making sure that the Fab bound in a specific way, and the Fab was actually accommodating for that. So, to summarize what I've been talking so far, what we have done, what the talk that I've given you here is a combination of, say, cryo-EM combined with Hg exchange mass spectroscopy, where we have tried to look at a specific antibody. So again, this is the tip of the iceberg, where we have started out with one specific antibody and one specific epitope that it's looking at, but trying to start asking the question as to how these broadly neutralizing antibodies that are actually successful in neutralizing many HIV isolates, what would be driving factors in terms of structure, where we are trying to design immunogens, then in those cases, what would be the structural features that they're actually targeting and what are they actually localizing on. And we see that there is a very focused epitope targeting that's happening here, where despite the different dynamics natures of the two envelopes here, the actual specific interactions are still maintained, and the PGT145 broadly neutralizing antibody actually accommodates for that very slightly. And as we saw in the structure and as we saw in the HTX exchange data, we actually don't see any long range effect of such a binding. So even in the dynamic envelope, even though at the binding region we see these small variations in how the glycan is and other things, it's pretty interesting that all the other glycans or all the other places which are not near that epitope area are pretty much equivalent between the two. There's actually no more differences. So whatever changes that are happening during binding and such seem to be very localized. And of course that extensive glycan interactions are an essential part of the epitope. So it could be one way how broadly neutralizing antibodies actually are able to span across different strains where there is a lot of variations or mutations in the protein structures, but the conserved glycans are a good way for the antibodies to actually make sure that they still recognize and can still bind and neutralize these viruses. So again, this is a start when we are hoping that we will progress more in looking into more antibodies at different epitopes and see if these kind of assumptions that we're making actually follow through. But so far, this is where we are starting out with. And with that, I'd like to have final acknowledgements. I'd like to thank the current members in my lab, mainly Ananya Chatterjee, who was involved in this specific work. And the initial 10 images were done by Nandini, Ankush, and Shruti. The collaborators in this HIV envelope work, which was with Dr. Kelly Lee at the University of Washington. The data collection, the cryo-EM data collection that I showed for the HIV envelope, the data was collected in the University of Washington Titan Krios there, but all the data processing and the structure determination was done in-house at IISc in our lab. I also like to thank the TEM and the cryo-EM facility at IISc Bangalore, which is where we have now most of our data sets and all the negative stain, the sectioning and the cryo-EM data sets we are collecting here. And of course the funding from the DBT Alliance and Serve Power and the startup grant. And with that, I'll thank you all for your attention.
Nice talk. Why did you choose PGT145? There are several other PNAs. Actually, PGT145 is not that broad as there are other PNAs that targets B1, B2 text.
Yes.
PGTN, Cap256, you have PG9, PG16.
There are many, yes.
Yeah.
Yeah.
PGT145 is generally used as a reagent to unify trimers. I'm not sure what's the rationale of this.
So the initial choice of PGT145 was more because it actually binds to the trimer, so it's a quaternary epitope that it's recognizing. So the idea was to first see how the quaternary epitope, because when you're looking at dynamics between this partially open or closed, it's the central quaternary epitope that gets distorted the most. So the idea was to start with an antibody that actually recognized that quaternary epitope. But yeah, there are other B2 which are more potent. Yes.
The other thing I'm surprised because again 156 and 160 is well known. It's not only for PGT145.
So the glycans are known to actually, so in mutational studies it's been, so the difference here is that in the mutational studies and other things where people have deleted these glycans, they were able to show that it actually impacts binding. But again, the structures that were known before this, which was basically with the BG505, in those you could actually never trace the density. So there was always a speculation of is it a direct interaction or it was an indirect way of making sure. And in this structure, we were able to actually dissolve these specific. We don't know if that was because the purification doesn't have any other specific mutation, like any stabilization mutations, and so the glycosylation is better. We actually don't know why. But in this case, this would be, from a structural perspective, this is the first time where we could actually see these glycans making direct contacts. But yes, the data was there from molecular work that they are important in the binding.
Nice work, Vidya. So in your hydrogen deuterium exchange experiment, where you could figure out that BZ505 was really structured, and it's because the given V2 loop is much.
Yeah. So there were so I mean, we didn't do the work, but our collaborators did look at the length of the V2 loops and its correlation in terms of the dynamics. So in terms of dynamics, there is a slight variation because of the length, but when it comes to PGT145 binding to it, what they found was that the length of the V2 loop didn't actually have a prominent effect, but the dynamics of it, but the length could affect the dynamics, but then there was no direct correlation in terms of, if you just looked at the length of the V2 loop. See, because there are certain isolates where the V2 loop is a little bit longer, but it's still a bit more rigid than others.
So when in your structures when BZT145 binds to a structure and without BZT145, do you see any differences in GP41 structure? Does 145 binding influences GP41?
So no, so what we see is that there is actually no connection. It doesn't actually transmit all the way. So the GP41 is actually pretty exactly, if you overlap them, the RMSD values are pretty common. So the binding it doesn't propagate all the way. So whatever changes are happening, it seems to be a very local binding order that's happening. Yeah.
Okay, thank you.
Thank you.
Let's now move on to the last participant, Sweetie Samal from IISc at Faridabad. Our laboratory has been working on this, characterizing different viruses and next generation vaccines, and more importantly, they are actually doing vaccine evaluations, different candidate vaccines, and also setting up assays for screening a range of antivirals. She is going to talk about the virus envelope protein, enigmatic, multifaceted, and promising vaccine targets. I know all of you are getting restless, and the food is also getting cold. So she is standing between the dinner and the
Yeah, but on behalf of organizing committee, I can assure you, have patience for next 20 minutes. You are going to have a nice dinner. Good evening, everyone, and it's a great privilege to be here. And I would like to thank organizing committee, Professor Das, for this nice intro, and Sasank, Kesav, and other committee members to give me an opportunity to present our work in this forum. So today my talk is divided into two parts. One, I'll be talking about two viruses. Both are enveloped. One is the RNA virus and one is the DNA virus. And I will start with SARS-CoV-2 because our lab mostly work on the respiratory viruses. And so we started with some of the fundamental question, and the setup has already been given by previous speakers, Kesav and Sasank. So we, as a virologist, we are always interested to know the mechanism, and SARS-CoV-2, as you heard, that unlike the sister virus SARS-CoV-1, it has evolved, and now it seems like it's a different virus from the coronaviruses, because people who are already working on respiratory viruses, human respiratory viruses, and with coronaviruses, they always feel influenza is going to be the next pandemic coming out, and they never anticipated that SARS-CoV-2 will come out. When the SARS-CoV-2 come out also, there is anticipation that the virus is not going to evolve that much. But from December 2019, the first evolve has happened in March 2021. So we asked this fundamental question, and gaining the knowledge within 2 years, we have harnessed one of the non-structural protein of SARS-CoV-2 to establish a novel indigenous vaccine platform. And using this vaccine platform, now I will share you some of the data in which we have established the proof of concept for development one subunit recombinant vaccine for monkeypox, which is emerged in last September 2022. It's not moving. No. Okay. Okay. So I think everyone is familiar with this Greek word. So one of the one of the quote that SARS-CoV-2 has made us learn that change is the only constant, and now we are having deficit of Greek words to give the new variant, emerging variant, and the recent emerging variant is the IISc. And as you heard from previous speakers, so if you look at the evolution of the SARS-CoV-2, you can broadly divide this virus into three categories based on their pathogenesis. The original strain, which we are calling the Wuhan, the highly virulent strain, Delta strain, and I will call this Omicron as a moderately virulent strain because there is a lot of argument because of this vaccine and overexposed to the virus, we do have the antibody. That's why we are not getting this infection, at least in India and some part of the Asian countries. And I will say that maybe for Indians we have a very strong innate response. But if you see this figure, and it's from August 2028, from last month, so you can see that the prevalence of this IISc Omicron variant in US, Australia and other parts of the world, and because of which last week only CDC has approved seasonal vaccine for SARS-CoV-2. I don't think we will get into that, but definitely this virus is very much different from the sister virus SARS-CoV-1. So for us, we wanted to understand what are the molecular drivers for the SARS-CoV-2 productive infection, and we mainly focused on different mutations that happen in the spike. As you heard from Keshav, that his focus is towards understanding the mutation in the non-structural proteins, but as we all know, the spike is heavily mutated. So we wanted to understand the effect of any particular mutation on this virus pathogenesis and transmission. And as a virologist, we believe that viruses are very smart. So if they are introducing any mutation, they do it in the purpose. So if you look back to this first variant, which happened in March 2024, the first mutation that virus introduced is D614G, and it completely changed. There are two science papers came out back to back from US, and they say it completely changed the orientation of the spike protein, which enhances at least threefold binding to the HSA2 receptor. Then came the alpha variant in UK, where the 501 has been introduced, and also in the cleavage site, proline is a highly conserved motif in the cleavage site, and I will explain it later. And this proline has been mutated to histidine. Then the virus has mutated this proline 681 to arginine in the cleavage site, and then again the mutation has been reverted by this Omicron variant P618H, and all these viruses still now, this IISc variant also, they are conserving these two mutations. So we wanted to understand whether this two mutant, whether they are playing any role in the virus infection and transmission. So for any envelope virus, whether it's influenza, HIV, Nipah, Ebola or SARS coronaviruses, then the pathogenesis mainly depend on this proteolytic cleavage site, where the virus usually harbor this polybasic arginine residue, and that must be cleaved by host cell proteases. So once the host proteases cleave this proteolytic cleavage site, the HR1 and HR2 repeats, they forms a 6 helix bundle, and the virus enters through the fusion and initiate the infection. And for the SARS-CoV-2 entry pathways, there are two types of entry pathways, because it as compared to the SARS-CoV-1, it has introduced another cleavage site, which we all known as S21 prime site. So it can harness two pathways. One, it can enter through the endosomal acidification route, where it can cleave by cathepsin A-like proteases,
and then it can mediate the entry and fusion, or it can also enter directly by, which is, they said that TMRS2, this serine protease prime this S2 cleavage in the Wuhan, but later on it has been proven that the virus can also enter through direct binding to this TMRS2 pathway. So to understand SARS-CoV-2 entry pathway and the role of this particular mutation in the entry and fusion, so first we have introduced different kind of mutation in the protein label, in the RBD label, and I'm not showing the whole data, but what I'm showing to explain that when you introduce this particular mutation, you can change the binding of the HSA2, binding of RBD to HSA2 receptor, and which also helps the virus for the entry. More, the higher the binding, the virus infectivity increases. So it also can help for a vaccine design. So instead of every time having a new vaccine for new variant, you can selectively introduce mutations which has shown to enhance the receptor binding, and that can be used for an universal vaccine design for the SARS-CoV-2. So as I said earlier, the proline is highly conserved in the cleavage site. So to understand this mutation, so we have developed different pseudovirus mutant, and one of the particular mutation that we have introduced is the D614G, this mutation, which has only two mutation we introduced, so D614G and P681 and other pseudoviruses also we have produced, and you can see that introducing this two mutation only, it is increasing the AC2, not only the AC2 binding, but the cleavage is also increasing as similar to Delta, and the similar cleavage also we are seeing in the live viruses. So these particular two mutations may be along with the compensatory mutation, but this two mutation is very important for the Delta infectivity and then for the pathogenesis. So we have also shown the, on the pseudovirus labels, see the infectivity titer. And another mechanism that we have seen is the fusion mechanism, and we see that this proline 2681R really it is impacting infectivity and the fusion as compared to the P681H which is available, which is present in the Omicron. And the fusion and synthesia in presence of HSC2 is one of the major factor, because it not only helps from virus, from apical spread, but it also helps in the basal cell to cell spread. So the more the fusion, more the infectivity, and it is not only for the SARS-CoV-2, but you can see for other envelope viruses too. So that also we see that it has been increased for Delta as compared to the Omicron and all the other mutants which is having this P681H mutation. And one of the things that is interesting also, we found that even though Delta is highly pathogenic, but it cannot be, it is not inhibited by E64D, which is a very well known cathepsin inhibitor, nor it has been as compared to Omicron, it is not, you cannot use this chemostat mesylate also. It's not showing significant inhibition of the viral infectivity, which is also a TMRS2 protease, serine protease inhibitor. So that means that Delta, there is some other mechanism it is adapting to not only for the virus entry and fusion, but also which is impacting the virus pathogenesis. And we in the Delta live viruses, we also see that as compared to the Omicron in both A549 and Kakoto cell, which is an intestinal cell, and that Sasank has already told that the pathogenesis is high in case of Delta virus as compared to Omicron. And also we see that the stability also going to increase. That means this Omicron viruses as compared to the original Wuhan ancestral, the stability also increases for this mutant viruses, and Delta is highly stable than Omicron. That might also contributing for the virus pathogenesis. So I'm not going to show all these results which I have done, because this papers has been already published. So what is the take home message is that the double mutant D614G and P681 are one of the crucial factor or mutation that has been adapted, that has been
introduced in the Delta, and both these pseudoviruses phenotypically they behave very similar. Omicron has the less fusion ability than Delta, and in the presence of TMRS2, it doesn't increase the fusogenicity in cell. That means it suggests that it is moreover taking the HSC2 pathway, where Delta may be taking both the pathway or some other pathway which we don't know yet. And in another study also we have found the conserved serine residue at S2 site. If you could abrogate this particular site only, you can see the complete loss of virus protein cleavage and virus infectivity. So all the studies give us that maybe we have, when designing an antiviral drug, we have to understand how this virus mutation is affecting the virus infectivity, fusion and pathogenesis, then understanding then designing this antiviral drugs. And one of the things then we came across, because when we were doing lot of studies, then we found that maybe SARS-CoV-2 variants might be exploiting the tunneling nanotubes. So some of the virus, it has shown in influenza and HIV, the virus can cross-talk between cell to cell, and they can spread between the cells if the cells are in the distance by forming this TNT or nanotubes. And we have also found that here you can see that how even between two cells which are very apart, they can form very good tunneling tubes, and it is seen in different variants. But we have also seen that the highest number you see in the Delta, the thick TNT you can see in the Delta, and also Delta is able to do some also able to introduce philopatia. So maybe that is one of the mechanism, and that these studies has been done either in the Viro cells, and we see some of this effect on the A549 cells, and we are now doing some other studies to confirm whether, because these nanotubes are formed because of this actin filaments, and so we would like to understand whether this virus can exploit that route to form this nanotubes. So because of time constraint, I will quickly switch using this non-structural protein of SARS-CoV-2 to establish a vaccine platform. So when we are studying the SARS-CoV-2, we come across one of the non-structural protein which has been reported by one group that they can form self-assemble to form a dodecamer structure where the N-terminal and C-terminal hands are open. So you can put any antigen, it can be different from the same virus, different epitopes, or from different virus and it can be used as a vaccine. But there was no report or no proof of concept where the group has been able to develop the vaccine. So we used that this phenomena, and we started with SARS-CoV-2 variant of concern, because as you know that the virus is going to change, so every time, like influenza, you need to have a new vaccine, and which is not a very cost effective. So we developed this novel indigenous multivalent vaccine platform, which is a very plug and play platform. It has the flexibility to express one or more epitopes domain, but it has a limitation that above from that size you cannot express this whole vaccine as a subunit vaccine, a subunit protein candidate. And we use a very convenient and robust expression system. So we use E. coli system for the expression, but you can use another expression system like baculo or mammalian expression, and it works very well. And it is highly stable because they form 100 nm like VLP like structure. You can store it at 2 to 8 degree. And we have the proof of concept established for SARS-CoV-2 variant of concern, now for the beta corona vaccine, and recently for the monkeypox vaccine. And all these candidates has taken up by the industry for the drug substance and drug product formulation. So I'm just showing two of the data from the SARS-CoV-2 VOC vax, where we have put the RBD epitopes from the Wuhan strain, from alpha and Omicron strain, and these are only 20 more epitopes from RBD, and you can you can produce a 29 kD protein, and the company has done all the drug product formulation, and they gave back the formulation, and we challenge both the Wuhan and BA5, and you can see against both the challenge, it gives you a very good protection. So now the stability study is going on at the company front. But then again, when we present this data, the reviewer always keep on asking, at least in India, that when there are so many vaccines are available for the SARS-CoV-2, so where you will place this vaccine? So that's why when this 2022 with the emergence of monkeypox, so monkeypox belongs to the orthopox virus genus and it's a DNA virus, so it doesn't have a single antigen. So we are lucky we had SARS-CoV-2, but the single spike is highly immunogenic. But monkeypox, it translates more than 100 genes and there are more than 10 antigens which are highly immunogenic. So there is no vaccine is available except the Genos vaccine. But I mean, as an academic scientist, you also have to understand the industry perspective, whether the industry wants to take this vaccine or not. So our market research says that, so industry came back to us and says that why don't you have a monkeypox vaccine using this platform, and even though the market is not good for India, but there is a big market for the US and Europe. So as I told you earlier that this monkeypox vaccine, monkeypox virus, there are more than 10 antigens and it is envelope virus, but it has both extracellular envelope variants and intracellular mature variants, both are infectious. That means when you are designing a vaccine, you have to take multiple antigen and then produce it as a vaccine candidate. So the only vaccine candidate available for monkeypox today is the Genos vaccine, which is a live attenuated vaccinia virus vaccine and which has more than 85% homology. But when this outbreak happened in the Europe and US, this vaccine is not available to everyone. So there is a scarcity of vaccine, and that's why the demand for the monkeypox vaccine in the Western is still good. And for us, also it's give us an opportunity to prove our platform, it can work for other vaccine also. So we have designed around 5 antigens, and I'm going to tell you about this one vaccine candidate which worked for us. And there are different criteria when we did develop an antigen, whether you are getting the yield as per the industry requirement in the lab scale, whether it is stable, whether it is forming this dodecamer structure, all these criteria it's need to fulfill. So as you can see, so we have stapled 2 antigen, and I cannot disclose this antigen name because of the IP issue, but in the bacterial expression system in the lab scale, in the 2-liter batch scale, you can get a yield of 1 gram per liter, which is very good for the manufacturing scale. And you can see this not only binds to the structural protein, but it also binds to the monkeypox antigen. And this is the SEC, which shows that it forms a dodecamer structure, and we have completely characterized the biophysical analysis of this candidate. And then we also have to show the POC to the company before it will take you for the drug product development. So to do this, we have established the vaccinia virus system in our lab. Also, we have established the in vivo challenge model for this vaccinia virus, because we cannot access the monkeypox virus, which is a BSL-3, but vaccinia virus is BSL-2. And as you can see, if you give 2 doses of this protein along with the Adavax or Al-hydrazole adjuvant, it is able to induce very good humoral response, and also it is able to induce very good T-cell response. And this is the challenge study against a lethal orthopox vaccinia Western reserve virus, and you can see in both the adjuvant there is a complete protection in the mouse model, and you can see that there is a complete reduction, there is no virus titer. We have taken trachea, lungs and brain. You can see the clinical scoring parameters, as compared to the adjuvant alone, that it gives you very good protection, and also it able to give you very good neutralization also against the vaccinia virus. So this is now taken up for the drug substance and repeat development by the company. So three of these vaccine candidate has taken up by the company, and I hope before I reach 50, maybe I will have this big gram moon landing effect, because for the academia scientist, if your one product will enter into the human clinical trial, then it's give you a big goosebump effect. So with this, I would like to acknowledge, thank our leaders who believed in some of the very risky and new concept which has been written in the lab notebook, and my colleague, teachers, staff, faculty and scientists. And this is our lab. So the study on the SARS-CoV-2 was funded by Bill and Melinda Gates Foundation, and the vaccine platform has developed by the THSTA core, and the beta corona vaccine we are now developing with the CEPI. And with this, I would like to once again thank you, the organizer, and this beautiful audience for having the presence to listen to me. Thank you. I'll be happy to take any question. A nano cage means it's formed a nanostructure. It's self-assembled. Like there is, you have one vehicle where you attach two epitope. It can form 12 structure come together to form a nano cage. So if you have given one epitope, so in the protein you are displaying 12 epitope. So there is an N terminal, there is a C terminal. So you can display around 24 epitopes. So PCT has been filed. It is available in the website, the patent website. We have filed for the Indian patent, but I don't think the sequence is available.
I don't have a question, but I have a suggestion. So since you have done this monkeypox vaccine, so why don't you go ahead and develop it for lumpy disease?
Sure, ma'am. Our teachers' day mandate is to work with human viral, and working this, and 2 days back we heard, like I think everybody's know that the COVID vaccine came on 356 days. Then CEPI declared you should have a vaccine for 100 days. Now they are telling that there is that failure, rapid failure, that fail early to reach the success. And in India, having a vaccine from academia scientists, it's take a lifetime. So it's very difficult. At least now we have 3 candidates. And I don't know, because when it goes to industry, then there is a lot of regulatory things come up. And I'm telling you, all these proteins are tagless. Usually we express this protein as tag. So all these are tagless. So purification is a big challenge. But you purify, and in the lab now we are getting around 1 gram per liter. So definitely, but we want to prove at least one candidate to go for human trial for the safety, because it's a new platform. Then it will establish the piece.
Congratulations, excellent work, Sweetie. Against nano cases, do you get an immune response, and is there a problem for boosters?
That's a very interesting. Within 1 year we screen 20 antigens for beta corona. Okay, that's a CEPI mandate. When they give you money, they expect you the result. And we find, because it's a very noble platform, so we found when the epitopes are, very well displayed in the nanocage, you don't get response in the nanocage, and that is your candidate to take forward. But if the epitopes are not, because it forms a 32 symmetry structure, it's a 3 by 3. I'm not a structure biologist, but I'm now looking for some cryo-EM. So if it make a perfect structure, then you don't see. But if you see, like the epitopes are not well displayed, you do see a response against the boost. But then again, when we started working with the CEPI, they wanted to check if you give the NSP, because now everybody has exposed to SARS-CoV-2. So we have a memory. So when you are giving a vaccine, whether any adverse effect you are seeing, and that study we have completed, and we don't see any AD effect.
All right. So excellent translational work. Thank you, Sweetie.
Thank you.
And if there is no more questions, I think I would like to thank all the speakers for this session for this wonderful work and exciting and challenging aspect. I think now we have announcements.
Thank you everyone for your active participation and engagement throughout the enlightening session on viral evolution. A big thank you to our esteemed speakers for for sharing their expertise and shedding light on this ever-evolving aspect of virology, and the chairs, Professor Ranga and Professor Das, for chairing the session. As we reach the end of the day, I want to express my gratitude to all of you. Today has been a day filled with profound insights, engaging discussions, and a true sense of community among us virologists. We look forward to more stimulating discussions and exciting sessions tomorrow. For now, please proceed with the dinner that has been arranged at the same place as lunch, that is the main guest house. Enjoy the evening, continue networking, and prepare for another enriching day of scientific exploration ahead. Thank you and see you all tomorrow.
One small announcement. Please carry your ID cards, all the participants.
Please proceed for dinner.